Good morning, everyone. So due to some technical issues, this session is starting 15-20 minutes late. Sorry about it. The first speaker of today's session is Professor Devesh Kumar, who is from Department of Physics, Central University, Lucknow. He was Ramanujan Fellow in CSIR IICT, and before that he did his postdoctoral research from the Hebrew University of Jerusalem, Israel, and Max Planck Institute for in Muhelheim, Germany. He is PhD in physics, and his research interests are computational physics, computational chemistry, quantum chemistry, electronic structure and spectra, and molecular mechanics, biomolecular simulations, and mesogens and biological molecules. These are all in his research expertise. He has done a lot of research work in the area of computational chemistry under the supervision of, in his postdoctoral tenure, Professor Shashank Sheikh and Professor Walter Thiel. When he started his independent career, he continued several of those works and taken them to a good height. He has published more than 20 JACS papers, several Angevin Chemie papers, several chemical reviews are there, and he has published, he had citations of more than 6,000 and H index of 44, and his total impact factor is more than 650. Welcome, Professor Devesh Kumar. Now you can start. Thank you.
Good morning, everybody. I am going to talk about something about chemical and biochemical activity or reactivity. The main point is some stupidity one has to learn. Why one needs to learn the stupidity? The answer is very simple. The system for which we will try to model the chemical reactivity or activity. These all systems behave very stupid. Hence, we have to do some stupid tricks and all those things. And I'm just going to tell all these things. These are my collaborator and members. First, electron. Chemist's notion of crystal reactive based on the distance of two particles, proton and electron. A neutron is just a combination of the two, and the force actually, when two electrons come near one another, they interact by the fundamental electric force law, and this force can be expressed by a simple equation. That is just equal to Q1, Q2 by R square, where R is distance between the two electrons, Q1, Q2 are the charges, and F is force on it. If both particles are electrons, then Q1 and Q2 have the same, and force means a positive. When a positive force acts on a particle, it pushes it away. Problem now suggests that the force of interaction is small if the particle get very far apart, so the R becomes very large. Therefore, two electrons right near one another will push away from one another until they are separated by such a long distance that the force between them becomes easily very. Then next comes atoms and iron. A simple picture of atom is a dense heavy nucleus with positive charge surrounded by a group of electrons that orbit the nucleus and that have negative charge. Nucleus and electron opposite charge active force hold the atom together. Uncharged atom, the number of electrons negatively balance the number of total charge of the nucleus. So 1 electron for every proton. If the number of electrons does not match the charge of the nucleus, the atom has a net charge and it is called ion. If there are more electrons than the protons, the net charge is negative and ion is called negative ion, and if there are more protons than the electron, the situation is reversed and we have a positive ion. Positive ion tends to be smaller than neutral atom. Smaller than neutral atom, whereas negative ion tends to be a bit larger. Then the next term comes molecule. When atoms are brought together in a fixed structure, they form a molecule. These are there figure shows some examples of molecules. To form molecule, atoms bond together. There is variety of chemical bonds, but they are all caused by interaction between the electron of atom and ion involved. The breaking and forming bond is a chemical reaction. Since electrons are responsible for bond, and chemical reactions are just the making and breaking of bond, which follows that electrons are responsible for the chemical properties of atoms and molecules. Then we come to the molecular modeling techniques. Molecular modeling is a set of techniques for investigating chemical problems on a computer. Questions commonly investigated as we all know. First is molecular geometry. The shape of molecule, bond length, bond angle and diagonals. Energies of molecule and transition state. This tells us which isomer is favored at equilibrium from transition state and reactant energy and how fast reaction should take place. Chemical reactivity help us to know where the electrons are concentrated, that is the nucleophilic site, and where they want to go, that is the electrophilic site, enable us to predict where various kinds of reagents will attack a molecule. And then we can also model the IR, UV and MR structure. Next, we can calculate the interaction of substrate with an enzyme. Seeing how a molecule fit into active site of an enzyme, if one approach to designing the better drug. Some physical properties of substance can also be modeled. They depend on the property of individual molecule and how the molecule interacts with the bulk material. We can model the interaction of a drug with protein and DNA, seeing how a drug fit into the active site. Also to verify the stability of drug at binding site and estimate the interaction energy to potent drug. And modeling of the structure of biopolymers. Being a complex structure of biopolymer, it is structured like structure with assay. With help of molecular modeling, several possible structures generated and by projecting and comparing the electron density as obtained by the X-ray crystallography and generated structure of final structure reported. What are the tools available? What are the tools available? So first is ab initio method. This is the equation describe how the electron in molecule behave. Sunday equation cannot solve typically for any molecule with more than one electron. Ab initio calculations are relatively slow. The geometry and IR spectra of propane can be calculated at a reasonably high level in a minute upon QC, but very large molecule like stride could take perhaps weeks. Then there is a semi-empirical calculation. This is also based on some equation, but there is more approximation is included to calculate the very complicated integral and save the sign. This plugging experimental value into the mental procedure to get the best calculated value is called parameterization. It is a mixing of theory and experiment that makes the method scientific. Semi-empirical calculations are slower than but much faster than ab initio. Then it comes the density functional calculation. We all know that it is also based on saturation. DST does not calculate wave function, but it derives electron distribution directly. It is faster than ab initio and its results are reliable. 15 minutes है So nowadays, through internet, everybody is aware of software packages. So I am going to skip this also. These are the functional group and structure which are required for modeling of any chemical reactions or drug molecule, whatever, when it's chemical activity. The first question arises, do we need to reproduce the experimental number? There is several answers of this question. If you are developing a basis state, a method or a software, in that case, answer is yes, because these comparisons are important. But if you want to model reaction mechanism, reaction pathway, or try to understand interaction at molecular level, answer is no. It is important to reproduce experimental pattern rather than the experimental number. Some calculated parameters are necessary energies of substrate taken from the chemistry chemical book as well as the calculated with UV3 all data represented its result after a full geometry optimization in Jaguar and followed by frequency. Jaguar is the quantum mechanical software which comes with and what we are seeing, you will see that the first small basis set number is far different with larger basis set. It is a bit closer to the experiment, but it does not produces exact numbers what experimentally found. These numbers do have no proper match, although the system is studied small. This is just correlation of barrier height of the and the ionization potential. So this is an initial potential and this is very high and we found a very good correlation. Getting good correlation is not much important, but the result suggested substrate epoxidation by Day-4 enzyme is function of ionization potential of substrate. Ionization potential, one of the basis property of substrate, not of the oxidant. But this correlation suggests that the epoxidation, various height of epoxidation is mainly depend on the basic property of substrate, not the of oxidant. Why you want to attend this workshop? Most of the answer were to understand the basic concept. Why to understand the basic concept? Why not feel how other people are working? Is it possible to understand any concept without feeling? It is important to feel the systems to be studied, method need to apply etc. Like, it does not matter on what we are writing. Only important task is to curve some structure and feel it. It is just feeling which makes us to learn the alphabet and complete a full sentence. These are the general alphabet of the Hindi. This is normal alphabet. This is the conjugated alphabet. What about this our modeling? Out of 112 characters, we have given some example of some characters like hydrogen, carbon, nitrogen, oxygen, sulfur, fluoride, etc. How much we know about them? What are their conjugated form and what are the effects on the conformation? These are the basic questions which we need to fill before we start any project. Effect of double bond over single bond. This is the sum. This is a line and this is the hydrogen. You can see here it is doubly bonded, one hydrogen out from here. Because of double bond rotation around this tree will be restricted. Why? Because it is not energetically favorable. The next question always strikes in our mind is choice of method and basis. What method and basis is suitable for my system? Artifact, BP86, B3, M06, etc. Why? For MD parameter, it is always suggested to use 6,3G star basis rate. Is it written in Gita-Ramayana or any other holy book? Answer is no. Then how I will decide? Is there any set rule? Answer is no. Then what we need to do? We need to do some nursery rhyme. By my system will have some thing like below conjugations. It will always be nice just to do some test calculation to benchmark with the different basis state for each atom and compare the computer data with standard one. This is our nursery rhyme. It is always necessary to have a faith on the software which you are using. It does not matter someone else got good result with different programs. This point need to be remembered. That someone with good result with different program does not make sense that the program you are using will not give the good result. All program will provide good results. It is used properly. So proper use of that program is needed to learn. Selection of methods and software for any modeling calculating or calculating your reaction. Initial geometry always play an important role. It is always better to optimize your geometry with 61G based on it is my experience. It is always better to optimize geometry with 61G. Although visually overestimate several times, but optimized geometry is always reliable. Once you have optimized geometry, use any method necessary to complete your project. Never try to optimize with some super basis set and method initially, because initial geometry is very important to start a project. Nasty behavior of the system. This is Crowley and bond dissociation energy calculated for the hydrogen at different position 3, 4, 5. And we found that extracted hydrogen at position 5 is energetically much favorable in comparison to the 3 and 4. But in enzyme, C4 is always targeted. Why C4 is targeted? It is clear that here steric effect plays an important role, that C5 will never be approached by the oxidant. Oxidant will always have access to C4, so it will extract hydrogen from the C4 position rather than C5 position. Boiling reaction mechanism. So this is our, if you see this mountain curve, this is one side is our lectin complex, another side is intermediate and product, and we have to find the barrier. Start reaction curve, path of the barrier height, and you will have intermediate. It seems so simple, but once you reach at the top of the tail, you observe that it is not only flat surface, but it leads to several different pathways with minor barrier height. This is very common in any enzymatic reaction, that after barrier height, there is always several pathways with minor energy barrier, and sometimes that height is not the peak, but it is a flat. A smaller system one can easily compute its water molecule. The calculating the heat of formation for your impression is very simple. Just calculate energy of each species, and this is simple arithmetic principle. One can get it. But is it so simple to model reaction pathway only? Scan along a reaction coordinate, we choose the reaction coordinate, which one will be correct reaction coordinate? OH bond distance or HO bond distance. In this case, for this particular case, both will be okay, but it will not be for every case. Like for this system, we want to extract hydrogen from here. So if we choose HO bond distance as a reaction coordinate, it will try to break the FEO bond rather than CS bond in which we are interested. So for this reaction, the reaction coordinate will be OH bond distance, scanning between OH bond distance. From the previous slide, I am transferring data here. इसके और अलावा reaction coordinate O bond will be given like this, and after extraction it will become like this. But when you try to study the reaction mapping, even with artifoxy 21G, it will not only make you straight from photograph, but also teach you all tricks which are essentially required for modeling any enzymatic reaction. Just perform a very simple calculation like this, and it will make you learn how stupid these reactions are and how stupid tricks need or required to handle such a small problem. Thank you. Okay, thank you.
To Professor Devesh for the presentation. Session is open for questions. If anyone have any question, they are welcome to ask. सुमन ये बोल रहा है it will So due to some technical reason, he is not able to answer the questions and it is disconnected. Now we will continue to the next talk, which will be given by Dr. Priyadarshi Satpathy. Dr. Priyadarshi Satpathy is Assistant Professor in IIT Guwahati in Department of Bioscience and Bioengineering. He was Assistant Professor in Department of Chemistry, Cotton College from 2015 to March 2015 to May 2015. He was a researcher in Uppsala University, Sweden with Professor John Ekquist. He worked there molecular dynamics free energy calculations for understanding accuracy in mRNA translation. He was a postdoctoral fellow in Paris, France with Professor Thomas Simpson. There also he worked on molecular dynamic simulation of initiation factors. His PhD in 2008 from Department of Inorganic and Physical Chemistry, IISc Bangalore, with Professor K.L. Sebastian. And he got several awards and honors that includes DST-ACRB Young Scientist Award in 2015, DO India Professor Fellowship Award in 2020, recognition as a key researcher in Sweden. He was having CNRS postdoctoral fellowship from 2008 to 2009, and several other research and professional awards, including University Rank 2 in his masters. He has published several papers, and they are all highly cited. JBSD and JCIM papers are his recent papers. And let's welcome Dr. Priyadarshi Satpathy. Dr. Satpathy, over to you now.
Yeah, thank you very much, Hemanji. But I was unable to. Can you see my screen? Is it visible to you? Can you see my screen, Hemanji? No, I am unable to hear you.
So your screen is not visible till now.
Okay.
You can go to share screen.
Yeah, that is what I am trying, but
right hand side share screen is there.
Yes.
You can share either your whole screen or only the presentation.
Yeah, so this is the problem in this team. So if I want to share, then I don't see that option.
Right hand side, you see three dots, no? Besides three dots, there is one arrow, up arrow. If you click on that up arrow,
yeah, this
it will
is what
show you the option to share.
This is what I'm trying, but actually this works with my
no, right hand side, you see that mute all and share is written, no?
Yeah, I know that because we use it for meeting this platform, but unfortunately I don't know, this team one is not working. So usually what I do, okay, wait, so files, let me see if I can. No, this is adding to the cloud.
You are having complete right if you go. You will be able to see mute all and share.
Yeah, I know this. Unfortunately, when I you see press that option should come now, so where I can share all my screen or not. So that is not coming. So something is fishy here.
Share and mute all button you are able to see?
Yeah, I can see that.
You are using Apple or you are using any
Mac. Yes.
So your accessibility option is not given to the Teams.
Yes, but I can do it from my web-based browser.
No, web-based browser accessibility. Maybe you can go to the setting. Yeah, you talk to Suman. He will help you to give the accessibility.
You please go to system.
System preferences. Okay.
Yes, sir. Then privacy and security.
Oh, wait, so this is Mac. So just
Privacy is there, sir. System preference. Then
privacy settings.
then left hand side. So what you are seeing right now? General tab. Some tabs are there. General. Then
Yeah, I see all this location service, contact, calendar, microphone, automation, screen recording. These technical issues.
sir, I will try to help you. So if you go to the system preference and then security and privacy, if you click on security and privacy, last tab you will get privacy, sir. Last tab you will get general, then file vault, firewall, last privacy. Under security and privacy, you will get last tab privacy.
Privacy. Yeah, security and privacy.
Click on that, sir. Open it.
Okay, authentication. Yeah, I have opened it.
Now, what you are seeing, sir? Can you please help me?
Yeah, in the left hand side, you see in the privacy. So I'm using Mac. Okay.
Mac. Yes, same, sir. Fine.
Yeah, so on the left side you have location service, contact, calendar, reminder, photos, camera, microphone.
Correct, sir. Correct. Accessibility you can find, sir. There, accessibility will appear.
Right.
So, team is there or not?
Yes, it is there. So I have to click here, you mean, right?
No, sir. Whether team app is added, sir?
Yeah, added. Yes, accessibility. Now it was not selected, so I have selected it. Allow apps below to control your computer. Okay.
Yes, sir. Now you try to share your screen, sir.
Great. Let me see. No, still it is not. Nothing is.
Sir, can you please close this and join again?
Okay, fine. So I'm closing it, huh?
Okay.
Sorry for these technical issues.
This is very, very strange. Yeah, there's nothing is happening.
We are very sorry. So I don't know.
Can you please share your presentation? We'll share it from our end.
So you said that it is not possible to do it in online this web browser mode, right? So then I cannot share.
That is not possible.
Huh?
That is not possible. You cannot share through web browser. It has to be from app. Or we can share your presentation. You share it to us. We will share it, and you can continue from there.
Yeah,
Sir, try with web browser. That means, sir, you have to use this app, but web browser you can share.
Yeah, web browser I can use, right? So let me try that then. There I can use, because I used to do this in the meeting. So this platform I use, but I use actually the web browser. So it goes to the team, but in the web browser. So then there I share actually files. Let me see if it works.
So you are able to share now? Or
No.
if not, then you can share the presentation to us. We will share it from here.
Fine. So that is what. So there is an incoming call. So let me just. Are we live? So we can kind of.
Yeah, we are live.
So. Okay, wait. Hello, Hemanji, can you hear me?
Yes, we got your presentation and we are sharing it.
Okay, good.
Yeah, we have downloaded now. Now we are going to share this.
Yeah. Yeah, so I sent the PDF version so that it becomes easy without any problem. So please go to, full screen mode.
Full screen.
So it's not yet full screen mode. Okay, so thank you very much, Himanji. So, let me start. So today I'm going to talk about biomolecular recognition and the insight from the molecular dynamics simulation. So the cartoon which is shown here is the protein and the ligand complex. Now this complex has to decide that whether it is a correct complex, that means it is a correct ligand or it is an incorrect ligand. Okay. So next slide. So the outline of my talk is, first I will talk about what do I mean by recognition. So how to recognize right versus wrong, okay? And how molecular dynamics is useful in understanding right versus wrong. And then I will give you a basic introduction to molecular dynamics simulation and how we estimate this strength of recognition, okay, using MD. And I will give you some applications and highlight some of the limitations. Next slide, please. So here you see the protein P and the ligand 1 that is represented in a free energy axis. So if the complex is more stable than the free reactant or the free protein and ligand, then if you mix them in a test tube, what will happen is your complex will start appearing in the test tube. Okay. Now a time will appear soon or later that the concentration of the complex and the reactant will not change with time, and that is something which is called equilibrium, and that depends on the temperature that you are working with and in the buffer condition. Okay, so that concentration ratio of concentration is linked with the binding affinity of the protein and ligand, which is represented in this red box. So this delta G bind, okay, binding affinity, is related with the ratio of the concentration, which is represented with the third bracket, and the subscript equilibrium EQM represent the equilibrium concentration. Now this is the first Nobel Prize is given in chemical equilibrium by Van Doff. Okay, he worked with this binding affinity. He has related with the rate of the reactions, which we are not going to describe. Now in this slide I would like to draw your attention that you see the P, that the protein has changed its conformation in response to the ligand binding. Okay, next slide. Okay, so suppose there is another ligand which is L2 and represented by a broken line and red color, and it has the free energy which is a little bit close to the free state. Okay, so then you have two binding affinities associated with ligand 1 and ligand 2, right? And these binding affinities, okay, the difference between the binding affinities is going to be characterized as which. So if you take one test tube and you use same amount of L1 and L2 and your protein concentration is fixed, so if the gap between this red and the blue level, that red is the complex formed by L2 and green is the complex that formed by L1, okay, if the gap is more, then what will happen is in the test tube most of your protein will bound to this green one but not with the red one. Okay, so you can understand that this gap is something which can control that the protein will be mostly occupied with the green or with the red, to what extent the gap will dictate. Okay, again I will just briefly mention again that you see the distinct different conformation that it has attended once the protein bound to the ligand. So please go to the next slide. Okay. Now this delta G binding, okay, that is the holy grail of computational biology. So the interesting thing is, if you know this delta G bind of L1 and L2, then you can tell the experimental people that you try L1 because you know that it is more downhill. Okay, but it is a very, very difficult choice to calculate it accurately. Okay, so please note that what I said is it is very difficult to measure delta G or calculate delta G accurately. Okay, so I will come to this point. Next slide. Okay, so here you see that delta G, which I was telling, is the free energy binding affinity difference, right? And this binding affinity difference is something which is called as the recognition in thermodynamic sense. Okay, so bigger the gap, you make one of the complex more than the other. Okay, depending on the gap. Okay, next slide, please. Okay, so this gap, this delta G, it is related to the error, because as you can see here that you can make more and more red complex if the red and the green level, they are come close together, whether if the delta G is small. And Linus Pauling already worked it in a different completely different area where he has applied, and the bottom line is that this gap, if the gap is bigger, delta G, then you see the probability of making error exponentially comes down. That means you are populating less and less red level. So here are a few numbers which is given. The delta G1 is 9. That means the probability of making error is 0.18. So if it is 10 kilocalorie, you see the probability of making the red complex is 10 to the power minus 8. You just plug the number in the equation, P error is equal to a exponential of minus delta G by KT. So delta G clearly control the population, thus control the accuracy of a process. Okay. Next slide. So is it really important that can somebody play with this delta G gap? The answer is yes. So here is a cartoon in the bottom, so which is a ribosome which is holding a tRNA, P-side tRNA with its polypeptide chain, where the red color represent the mRNA. Okay. Now the job of this complex, so you see this ribosome is going to select the tRNA from the cell, right? And the tRNA, there are a pool of tRNAs, different types of tRNAs. They will go and bind to the A site of the ribosome. With the mRNA, it is going to make the base pair. So here the job is to select the correct tRNA, which will form the exact base pairing in the A site, rejecting the other tRNAs. So here you can again think of the correct complex as a green one and the red one as the incorrect, which comes from any other tRNAs, which does not make complementary base pairing. Now it turned out that there are drugs, okay, like one of the drugs is called streptomycin, which binds to this ribosome, okay, small subunit, that means this bottom small area, and it goes and bind, and what it does, it is kind of reduce the delta G. So then what happens is it goes and bind to the microbe ribosome, and delta G is small. So then you see probability of making red level increases. So the ribosome will make the protein of the bacteria, but it will going to make wrong protein because your delta G is now small. So the point is that there are a few examples here that you see the mechanism of this error, okay, of this antibiotic viomycin inside from the fidelity of mechanism of process. So this making right protein rely on this delta G's, okay, and the drugs can tune them. Next slide, please. Okay, so the conclusion what I told you so far is that delta G is significantly altered the probability of making error. Okay, now you note here that delta G is not infinity. So if it is infinity, then your probability is 0. So there is always a chance that you can make mistake. Okay? And delta G is definitely the strength of the recognition, okay, that we wanted to compute. So next slide, please. Okay. Now the question is, what is the role of MD, okay, in all this business? So the role is as follows. So if you can run molecular dynamics simulation and you can calculate the delta G binding, which is the absolute binding affinity, or you can calculate delta G binding, but not computing the delta G binding. So what I wanted to say is that you don't compute the binding, absolute binding affinity, but in some different way, you can estimate the delta G, okay, which is the relative binding affinity. You remember delta G is the gap between the red and the green level, okay, that you can estimate, but without estimating directly to delta G bind, that means absolute binding affinity. And I will come to it that this, as I said earlier also, this delta G binding, absolute binding affinity is the holy grail, and it is very difficult to accurately estimate. But the point is, you can estimate them using molecular dynamics, but it is less accurate related to the delta G, okay, which you get it from a different route. Next slide, please. Yeah. So the question is, how can you estimate delta G or delta G's? Okay. So so what you need is you need a structure, okay, and the structure usually you get it from X-ray, cryo-EM, NMR. So you know the position of the atoms that makes this biomolecule, so which is represented here. Now, if the structure is given to you, it is expected that that is probably a minima in the potential energy hypersurface. Note here that this is the excess is energy, this is not the free energy which I have drawn. Okay, think about it, this is an energy, and in the energy landscape, your complex is some kind of a minima. Right? Now, if you start in the minima, okay, and you tell the system that you have to maintain one temperature, that means all the atoms need to move, because temperature is nothing but the movement. And if you know what is the law of motion, then you can generate different structures around that minima. Okay? So once you generate different structure around that minima, so using statistical mechanics and the structures that you have generated, combining those two, you can get the information of free energies. Okay? So that way you can understand that you can generate more and more structures which essentially will give you the thermodynamic properties of free energies. Okay, and that can be done by statistical mechanics. Next slide, please. Okay. Now, what is the laws of motion? Okay, so there are two laws of motions. One is the classical mechanics, one is quantum mechanics. So we are going to discuss about this classical mechanics part, which we are going to use and let the biomolecular atoms move respecting the classical equation of motion, the Newton's law. Next slide. Okay. Now you see, what I told you so far, that experiments could be actually divided into two different, broadly classified into two different groups. One is structural part, which gives you the position of the atoms of your biomolecule, X-ray, NMR, cryo-EM, and another one is your biochemical studies. They will tell you the rate constants, binding affinities, and so on and so forth. But when these two groups, okay, when you do an experiment in the lab, you don't know what structures actually it is going through. Okay, what are the different minimas actually it is passing. Whereas in the left-hand side, the structural one, they don't have any idea about what is the kinetic constants of the thermodynamic parameters. Okay, so basically this approach of starting from structures and you run MD and link it to biochemical analysis like free energies. Okay, so basically it is creating a bridge between the structural and the biochemical studies. So this is very important. Next slide. Okay, now let me give you a basic introduction of the MD. Next slide, please. Okay. So as I said that, okay, suppose you see now the whole biomolecule is represented with one, this gray ball here, and it is in this energy landscape. So as you can see in the left hand side, if it is, then it is going to feel a force towards the bottom of the well. Okay, and also if it is on the right hand side, then it will feel a force in the left hand side towards the bottom. So the force you can compute. So if you know the energy landscape, right? So you can compute the derivative of the energy which gives you the force. So as long as you know the energy landscape, okay, you can compute the force. Next, please. Next, yeah. So if I know ER, then maybe R is something which you can think of as a relative position. So the whole biomolecule is connected with different molecules, different atoms, covalently and also non-covalently, and all this internal coordinate is represented by R. Okay, so where the structure you started with, okay, if you at that point, if you know the energy landscape, you can compute the force where the whole thing will, how the R is going to change. Okay, the internal coordinate is going to change. So there is absolutely no problem in calculating the force as long as you know the R. So once you know the force, then you see force is equal to mass into acceleration. So you can calculate the acceleration by force divided by m. So m is not a problem because you know all the what your biomolecule is made up of. So you know the mass. So you can calculate the acceleration. Now once you know the acceleration, okay, then what you can do is, if you believe that your force is constant, okay, during that time, then you can get new velocities and new positions from this acceleration and the previous position and previous velocities. Okay, so that is crucial. And this is done by some numerical integration. Okay, so then you get a new position RT. Okay, and from RT, that means now you're at RT, you can again compute the force. So you go back and calculate the new force and new acceleration, and that you feed to get new velocities and time, that is V2t and R2t. From 2t you keep on going, 3t, 4t. So what you get is a trajectory, okay, and trajectory that will give you different R's, okay, that means different relative positions. So you are basically generating different structures, okay, around this minima. So the point here is that you need to know ER. Okay, and you need to know from where you are going to compute your position, that is RT0, and also you need to know VT0. Okay, it may be that your molecule is everything is not moving. So in that case, your initial velocities will be 0. But if you pump in energy, suppose temperature, then all of them are moving, and you have to assign particular velocity corresponding to that temperature. So as long as you know these three things, ER, that means the energy landscape, the initial position and the initial velocities, you can keep on generating structures. So next slide, please. Okay. Now, if you look into this problem, so as you can see that if you go to the bottom of the well, okay, then your derivative of that position at the bottom, okay, if you take the derivative, this is 0, right? In the minima or maxima, the derivative of the energy is 0. So if you are at the minima, but you do not use temperature, okay, then what will happen is your system will come and stop there and it will not move. Okay. So this is precisely done when you do energy minimization. So suppose you start with the structure from your X-ray or cryo-EM, but you see they are not very sure about where the position of the atoms are because the density was not good. So the model they have created was not great. There is overlap or something. So what you do is you minimize the energies so that you come to the bottom and get something which is at the bottom. And once you get there, then you assign temperature and then you run molecular dynamics simulations. Okay, so if you do energy minimization, molecular mechanics, it will come to the bottom and it will not move. Okay, so thank God that nothing is at 0 Kelvin. So even if it is at the bottom, it will start fluctuating because it has to maintain the temperature that you are interested in. Okay, so keep moving around around the minima, and that will be used for for computing free energies. Next slide, please. Okay. Now the accuracy of this whole business of molecular dynamics simulation entirely depends on this correctness of this ER, because if your energy, how the energy will vary with your relative coordinates, if that is wrong, okay, then definitely you are calculating the force wrong. So acceleration will be wrong. So everything will be wrong. So there is one error causing point is your ER, and another point is when you are using you are getting the new velocities and new position VT and RT. Actually you are doing numerical integration, okay, and then in that numerical integration you basically do a Taylor series expansion. So the point here is that there is again one portion of error, because when you do a Taylor series expansion, you are neglecting the higher order terms, okay, that is in t, okay, in time. So Absolutely, there is no problem in calculating force, calculating acceleration. So the problem is in your numerical integration part, accuracy problem, and most importantly in the ER. Okay. Now the question is what time scale that you are going to use, okay, for updating your velocity and R. Now, as you can see here, as I said that you are using Taylor series. Okay, so your calculation, first of all, what you want is you want your force to be constant. Okay, when you update your velocities and R. So that's why you have to use a very small t, so that the force is not changing within that time, so that you get the velocity and new velocity and time. So that's why you need to choose the time scale of the slowest, the fastest degrees of freedom, whatever is there in your system, you should choose it much slower than that. Now, if you think about this biomolecule, your CH stretching is occurring around like 10 femtosecond time step. So if you want to see CH stretching, so it is better actually use 1 femtosecond, so that it's very small. Okay, and for each of the point, your force is constant. But if you fix that CH bonds, if you are not interested in seeing the CH vibration, then you can use 2 femtosecond. Okay. So the choice of this T is not something which is innocent. Okay. So it has something to do with the accuracy of your biomolecular simulation. So next slide, please. Okay, now you see these three guys, left hand side, Martin Karplus, Michael Levitt and Arya Waschel, what they did is they have provided how you represent this ER. Okay, the biomolecule is connected with different kinds of, so if I take a protein, it is an amino acid, so they are connected with different sizes, balls, they're connected together, and they have, you know, given a prescription by which you can describe the energies. And that is something which is called the force field. Okay, so it's this. So they got the Nobel Prize 2013, but there are other people who also, you know, developed force field, like OPLS force field by Will Jorgensen, Amber force field Peter Pullman, Gromos force field Herman Berenson. So all of these guys, they actually worked to you know, give you a force field where you can describe the ER. Okay, fine. So once you know ER and you can do it, and you can generate snapshot, and you can you know, assign one temperature T. Okay, and for that temperature, what you are going to do is you are going to give the velocities, initial velocities. So I will going to talk about it. So next slide, please. Okay, so just a quick overview. So this is a force field ER. So I would like to draw your attention in the green part. So you see, suppose you have a sodium ion, so that you want to model. So the sodium ion is going to interact with another sodium ion, right? So they will be interacting with non-covalently, and that non-covalent term is given into two parts. What is Lennard-Jones potential at the top, where you see R to the power 12 by R to the power 6 dependence. And whereas the Coulombic interaction is modeled by Q1 Q sodium, and then another Q sodium divided by R is the relative distances. Okay, so then you see, if I tell you what the epsilon is and the R0 is and Q1 and Qj is, you can compute the energies. Okay, as you can vary R. Okay, so the parameters which is given for Na plus is the epsilon, R0, Q's, okay, Q. So now you see, it may not be a sodium ion, but maybe it is a water molecule. So then what happens is the water molecule, they are also connected by bonds, like, so you see that O is connected to hydrogen, to hydrogens. So along with this oxygen, which you have to specify epsilon and R0Q, you need to specify again some kind of an energy function that will describe these bonds, and that is given in this half, the first term, half kb b minus b0 square, where b0 is your equilibrium distance, and theta 0 again you can put some you know, bending motion, so when the angle changes, so you can put dihedral angles. So as long as your sodium plus, and if you think of it has an internal structure, it is not a sodium ion, so then you have to give extra terms to the force field which describe, you know, different bonded terms. Okay, so the point here is that these ER, so that means if I tell you this constant, this B0 KB k theta, theta 0, k phi, which you get from the maybe structures given to you, X-ray structures. You know what is the distance, you know what is the angle, then you do some spectroscopy, you know this, bond strength. Okay, so then these things, if I give you, then you know how the whole molecule will behave. So if you stretch one water molecule, how the energy will go up, so you can compute the forces very easily. Okay, so the point, the bottom line is that your force field is the identity of your molecule. Okay, so next slide. Okay, so let me just spend a little bit of time that how you are going to. So there is no problem in the R0 because you get it from the X-ray structures. You know where the R0s are, and ER, that means the force field, is also given developed by these people. Now the question is, how can I assign velocities, initial velocities? Okay, so maybe it is in a minima, you minimize it, you get a minima, and then you want to initialize the simulation. Now once you give a temperature, so suppose 310 Kelvin, so people know that at 310 Kelvin, okay, in a box of ping pong balls, okay, very small, they will get a distribution for 310 Kelvin. So that is called Maxwell-Boltzmann distribution. So what you can do is, that Maxwell-Boltzmann distribution, you follow that, you randomly give velocity to all of your biomolecules, including water, so that, okay, it is represent the temperature T. Okay, 310 Kelvin, and then when it will move, it will redistribute the velocity, and it will give you correctly distributed velocities. Okay, because they wanted to move, but you see they cannot move anywhere because they are in a well. So that ER is going to restrict their motion around the well. They cannot go anywhere. Okay. But obviously, if the wells are connected, there are multiple wells and they are connected with a small activation barrier, then you can go from one well to another well, and you can give structures from different wells. Okay. Next slide. Okay. Now, this is precisely what has been used in 1977, the first biomolecular simulation done by Martin Karplus. So it is a very small protein that is called Bovium pancreatis trypsin inhibitor, so small protein. And you see that they run this simulation for 3.2 picosecond, and 1 picosecond is 10 to the power minus 12 seconds. So very, very small time they could run it. And I guess they don't have used explicit water. So water is considered to be a kind of dielectric. So your computational power is even less. So you tell these atoms to move in a dielectric. Okay. And that small tiny simulation shows some kind of a motion at the terminal, so which is highlighted by this red circle. So they see some kind of a movement, okay, and it has been kind of reported. And the good feat is that people can now allow molecules to move in the computer, okay, respecting Newton's law. Next slide, please. Okay, now the fine. So one small protein in a vacuum or continuum dielectric you can run. So, but the question is that if I take a small stretch of amino acid, okay, and then run molecular dynamics simulation, can I get the structure that people have obtained experimentally? Okay, so that is the question. Is it possible to reproduce experimentally determined structure? So next slide. And the answer is yes. Okay, so there was a video which I cannot show you. So in the left hand side there are two different colors. So the blue is your simulation and red is your X-ray. So what the X-ray structure, people identify these structures, and whereas what they did, the paper is 2011, okay, D. Shaw, I guess. So what they did is that they took this long stretches, the small stretches of the amino acid, and then they let them run at a particular temperature, and they start interacting, and they get to a minima, okay, which gives you which is completely the X-ray structure which is reproduced, experimental structure is reproduced. So that is a great news. So next thing. Okay, so here is another example where what they did is, so this molecule which is in the cyan color sticks in the left hand side bottom, so that's a drug molecule and this is a protein. So they keep that drug molecule a bit far away and they run a very long molecular dynamics simulations, and what they did is they saw that this molecule goes and find its pocket. So that's again a great thing to see. Now next slide. Okay, so before I discuss this, so let me tell you that once you run this molecular dynamics simulation, like one stretch and it goes and fold to its particular structure, so then you really know that, you see, this protein folding, that is kind of not fully solved, but what it is going to tell you is that the pathway of going to the minima is not random. It is not randomly accessing any structure. So you start from one point and there is a very well-defined directed way of going to the minima. Okay? So it's a directed motion to the minima. Okay. Now the question is how to estimate the delta G. So our popular approach for doing this is, one of the approaches called alchemical free energy calculation. So I'm going to discuss about it. So I'm going to bypass, okay, the calculation of delta G binding, which is a difficult choice. And I'll tell you why. So next slide. Okay. So we are going to, so here is a cycle that is given to you in the right hand side. So you see these different arrows, okay. The vertical arms of this cycle, the horizontal arms of the cycle represent the transformation of this ligand L1 to L2 when it is bound to the protein or it is free in water, when top and bottom, below. Okay. Whereas the vertical arms correspond to the ligand L1 binding to the protein, okay, or L2 binding to the protein. So the vertical arms are the real binding event, and the free energy cost associated with that is the delta G binding, which is the binding affinity that I have discussed. Whereas the horizontal part is a transformation of the ligand, which you cannot realize experimentally, because it has no meaning, right? Something L1 is bound to it and you are changing the L1 into L2. Okay, but the good thing is that this is a thermodynamic cycle. So if I start from the left hand side and I do a cycle and come back to the same point, the total free energy change is 0, right? So then if you add this all free energy changes, delta G comp, which is the transformation of the ligand 1 to L2 bound to protein, then you reverse the sign of this black arrow in the right hand side. So if you add them up and you take it 0, you immediately see that you get 2 delta G comp minus delta G free is nothing but delta G L2 minus delta G L1 binding. So it is the difference between the binding affinities of the two ligands. So the computation of this delta G comp and delta G free, although it does not make any sense because this is something alchemical, you cannot realize it experimentally, but it gives you a quantity which is very much physical, which is the delta G, or the gap between your green and the red level. Remember? Okay. So here what I'm telling you is that you can bypass the computation of this vertical arms, which is the delta G bind. Okay. So next slide please. So you can estimate this, okay, so you can estimate this delta G with high accuracy. The reason is that as I said, it is much easily you can do it. So I'll tell you why. Okay. So here is an example. I am interested to know that the protein, okay, maybe it is selective for potassium but not sodium. So maybe 2 different ions. So how can I compute this delta G comp and delta G free? Now I told you that the description, the atoms or the molecule is described by its force field, right? So what you can do is you can change the force field, okay, from potassium to the sodium slowly, very slowly, and you can keep on computing the free energies. Okay, so then that way you can get the delta G comp. So the point here is that the K plus is represented by epsilon and R0, and obviously you have a Q, right, charge, but the charge is same for sodium and potassium, so I'm not interested to that. So this force field is taken from Chan 36. So you see epsilon and R0 for potassium and sodium. So the idea is that suppose you have a structure where the potassium is bound to the protein, okay? Then what you do is you run simulation, and then you again you change this potassium epsilon and R0, you change it slightly, okay, and you again run simulations. Then again you change it to more close to potassium, and then you run simulations. So you slowly you use strata which connect potassium to sodium parameters. Okay, and that stratification is something which is called perturbation. Okay, and for when you run simulations for your different strata that connects your sodium to potassium, so from these structures one can use, free energy perturbation or thermodynamic integration method, which will connect free energies for these individual small steps. Okay, so next slide please. Okay, so in the left hand side, as I said, so what I have to do is I have to change this potassium to sodium, and I describe a parameter called lambda. Okay, so if I change the parameter lambda, it will change the parameters from the potassium to the sodium, so which is given in the form of the table in the left hand side. Okay. Now, so if you do it in the presence of the protein, that means this complex, or free in potassium in water, whatever you do, the whole philosophy is very simple. So as I said, there are two flavors. So A and B, you can think of two different lambdas, maybe 0.9 lambda and 0.8 lambda. So if you go from 0.9 to 0.8, so A and B, the delta G associated with it is like minus kBT, the temperature you're working with a log and an average. So this average is something called the ensemble average. Now note here, the averaging is done with respect to A. Okay, so the meaning here is that I can compute the free energy going from 0.9 to 0.8, okay, of my parameters. So what I have to do is I have to take the 0.9 simulation structures, and there I can use that to compute the free energy change from 0.9 to 0.8. Okay, so you have to just use the energy values corresponding to 0.8 and 0.9. Okay. So the point is, this is very interesting. You don't need any kind of simulation software. So if you are good at any language, so C, Perl, or whatever language you are familiar with, what you do is you run simulations. They should be very close, and then you take the structures and you just maybe a scripting language, awk or Perl, maybe set or whatever, you just take those structures and you calculate this delta G for these tiny steps. So once you've done it, you can sum it up and you get the free energy change for the process. Okay. So next slide. So there is another one, thermodynamic integration, that is the same principle. Basically, you average over the previous lambda. Okay. Now the key points I want to highlight. So if you wanted to use this technique, bypassing the delta G point, first of all, you have to do this very, very slowly. That means you need to have a lot of stratas. You have more lambdas that connect from 1 to 0. And if you have the lambdas are very much separated, so you can say that, okay, fine. So if I run lambda is equal to 1, can I connect to lambda equal to 0? Okay, there is absolutely no problem, because the equation that I have shown you, it will allow you to do that. And that is true provided you run the simulation of lambda 0 to 1 for infinite time. Okay, so for finite simulations, okay, you are unsure about ergodicity. So what you have to do is you have to use the stratification. Okay, so stratification is the must. So once you are in a finite limit, you have to make these lambdas very, very close, and you can only extract the tiny delta G's between the two lambdas. Okay, that's it. And as I said, that from lambda A to lambda B, so A to B, 0.9 to 0.8. So when you run 0.9 simulation, when you get structures, the first part of your trajectory you should throw. Okay. And that has something to do with the Hamiltonian lag. So but the point is that once you run simulation of 0.9, okay, you generate structure, first part of it you throw it, and the next part of the simulations that you use for calculating those averages. Okay, so these are the two things, key things one should do in order to make it correct. Okay. So the advantage of this is twofold. Number one is that you see I am subtracting delta G comp minus delta G free. Okay. So then what you are getting is the free energy difference between the green and the red level. So you are actually ignoring the free state. The free state is subtracted, okay, when you do this subtraction. So this conformational change associated with the protein which was kind of large P, that you don't have to take into account because it will be cancelled out because you are doing a relative binding studies. Okay, so there are two things. One is that the force field cancellation. So even if your force field is not great, ER is not very good, but you are doing almost like same mistake when you compute the delta G comp and delta G free. So when you subtract it, most of the force field error will be cancelled in that process. So that is one thing which is good, and that's why I said you can measure it with a high accuracy within 1 kilocalorie error bar per mole error bar. And also you don't have to consider the conformational change associated with the protein, and that makes this calculation really problematic. So I told you that this is the holy grail, the computing of the vertical arms. The reason is that you will be limited by conformational change, so you will be limited by the sampling and the convergence issues, and that is why it is difficult. Next slide. Next slide, please. Yeah. So I hope now I have been able to convince you that delta G absolute binding free energy is limited by large conformational change and sampling and the convergence issue. Because you see, you need to have the lambdas to be close, or else you cannot compute. And when there is a large conformational change, then it's very difficult. Okay. Next slide. Next slide, please. Okay, now so let me show you some application. I will not take much time, just yeah. So you see here, these bars, whatever you're seeing like this blue, these are all delta G's, and the way they have computed is exactly by using the cycle, which is called the thermodynamic cycle. So once you use the thermodynamic cycle, you can calculate that how a protein actually recognize the right DNA from the wrong DNA. So what you have to do is, at the left hand side, you mutate the DNA to some other DNA. You do it in complex with the protein, you do it in complex with the, where it is free in water, and then you take the differences. It tells you that how it is recognizing the right thing with respect to the wrong thing in a thermodynamic sense, and that you can use it for different purposes. It is more general. You can look how a protein recognizes correct RNA, how a protein is recognizing a protein, how protein is recognizing DNA. So all this recognition business, as long as you are good at the structure of your complex, then you can do this exercise and you can get this relative binding affinities of the recognition. You can compute and you can understand it in a much better fashion and directly link your free energies to the three-dimensional structures. So next slide. So the limitations in the brief, whatever I told you, see, when you use this harmonic force field, okay, never ever try to explain some chemistry, because there is no bond breaking. If you remember, we use KB B minus B0 square. So that means if you stretch the bond B, it will again come back. So there is no bond breaking. Again, it is a fixed charge force field. So the sodium, which in it is come to phosphate, or sodium in some some water molecule, the charges are always same. They don't polarize, so you don't have an explicit polarizability. And you see, the sampling is absolute requirement, because if you don't sample, the averaging that you have done to compute the free energies, that doesn't make sense. Because see, we are using statistical mechanics which rely on probability, and probability is more and more accurate as you have more and more sampling. Okay, so if you don't have more sample, your probability is bad. Okay, so then everything is bad. So you need to sample. Then you have time scale. Okay, so how long you are going to sample. So that is some computational cost. Then the convergence issues, because when you do a forward delta G calculation, you have to compute it backward, because we know that for these free energies, okay, there is something called microscopic reversibility. So if you compute delta G AB, and if you compute a delta G BA, that should be same. Okay, so the microscopic reversibility should be respected. So that is something what one should note. But overall, okay, so if you look now, we have enough computational power to run simulation longer time. So sampling is not that a big issue, okay, and the time scale. And also if you run longer and longer, your convergence issue is also kind of solved. Okay. But the main problem, okay, is lies in the description of the molecule, that is the force field. Okay, so the force field is really a big issue still now. So my suggestion to all of you is that you see this machinery is it has no brain. So if you want to use this, use your own. Okay, so this is a caution. So next slide, please. So the take home is that delta G is relatively easy to compute because you are kind of bypassing the conformational change of the protein in response to ligand binding. And also MD simulation, there are sorts of errors, so force field. So your numerical integration, as I said, time step. And also you see the alchemical free energy calculation, delta G comp and delta G free. You need lot of stratification, you need more sampling. You should think about the Hamiltonian lag, throwing of your initial part of the trajectories. And it is possible to make testable predictions. So if you do that, suppose you have two different drugs, L1 and L2, and you know the delta G, and it tells you that clearly that delta G is favoring L1. So then you can understand why L1 is better, and then you can tell the experimental guys to kind of synthesize and try it. Okay, and certainly you can bridge the structures with the biochemical data. So next slide. So I would like to conclude my talk by quoting Richard Feynman, and which say that everything that the living things do can be understood in terms of the jiggling and wiggling of atoms. So jiggling means small fluctuation, that means you are in a one minima and you are accessing different structures, whereas the large conformational changes where you go from one minima to another minima, which is called wiggling. So if you can model them, if you can relevant structure, you understood pretty good. Okay, so almost everything according to Richard Feynman. Okay, so next slide. Next slide, please. Okay. So acknowledgement part. So I thank Dr. Hemant Srivastava for inviting me and the whole MMTT 2021 team. And this is my group. So whichever I have shown in the application part, this is done by these people. So I thank also DST and IIT Guwahati. BIF facility that is supported by again DBT, Government of India, and Paramission Supercomputing Facility IIT Guwahati. So thank you very much.
Thank you, Dr. Satpathy, for a nice talk.
Thank you.
I am sure that it was very, very helpful for all the people who want to do simulation. Now session is open for the question and answer. If anyone have any question, they are welcome to ask.
Hello.
Yeah.
Yeah, sir. As you said, that means if you have two different drugs like L1 and L2, and like we are calculating the relative binding free energy, and that can give us like L1 is better than L2. Means can we also estimate like during docking we can get two different conformations for one particular drug molecule? That is possible, and means after calculating the docking score is not it is very much near to each other, means you cannot differentiate between. Can these relative binding free energy can tell us some story, and means if we are getting for one particular conformation the relative binding free energy is very good, and for another it is very bad, can we consider that okay, this one particular conformation is it could be a biological conformation or the bioactive conformation, and the another is not the bioactive conformation? Or is there any other method to justify these which conformation will be the bioactive or which conformation will not be the bioactive?
Okay, so let me answer all of your question one by one. Okay, so the first thing is that, as you say, docking. Okay?
Yeah.
So in the docking, what they want to do is computing the vertical arm. So what you do is you get a pose. So you have a protein and the molecule, the ligand is bound to it. Okay.
Yeah.
And what you want to do is you want to compute that how much is the strength. So in the docking, what they do is they use semi-empirical technique. Okay.
Yeah.
So they don't care about the unbound state. So you get a structure somewhere, somebody has given you the structure, and for this structure, what you do is you just separate them and try to compute the strength. And the computation of the strength is completely semi-empirical. That means you know what it looks like, and you have some data associated with it. It is in some environment. So if it is like this hydrophobic environment, how much contribution you are going to take. So that way you just try to crudely estimate it. So but note, what you want to do is by computing the vertical arms. Okay, yeah. And that is certainly not the story, because this is not the one conformation that is going to give you your binding affinity. The binding affinity comes from the unbound state, which you are completely ignoring.
Yeah.
Okay, So that is one thing. So the first thing is that the estimation of your delta G affinity using docking, first of all, is very, very bad. Okay, sorry to say. So it is good that if there are million compounds and you quickly wanted to, somehow you want to reduce your number of molecules. So you semi-empirically kind of fit and kind of do that. Now the second point is that, so I think Hemant is going to discuss about this, that MMPBS method, which is again the vertical lumps that you're going to do it, but it is slightly better because you are taking multiple conformation from your trajectory. And again, you are now forgetting your, what state the undissociated state is, the free state is. But that is, again, it is not that great because you are completely ignoring the unbound state. Okay? Now, as you said, delta G, it is good to compute. So you have to remember L1 and L2. You have two different ligands. Yeah. But if L1 and L2 are very different, okay, so L1 is like a very small molecule, maybe a benzene, and whereas your L2 is kind of fullerene. I mean, just an example, a very big molecule. So then in principle, the theory is not restricting you to do that. You can probably do it. But again, you have to do it so slowly and so nicely. You have to connect these two states. So it is again computationally very, very expensive. So as long your L1 and L2 are very close, like you have benzene and you have a benzene with CS3. So you want to see which one is better. I think for that one, these things are very useful.
Yeah. But if we can explain like which group is giving the better activity, means we can correlate that with the biological data. But if the two molecules are very entirely different, then I think delta G will not probably give us a better result.
You can do it. See, you can definitely do it. But what I'm saying is that you need to do a more and more stratification.
Okay.
That is what I'm saying. You can do. You have to do it really slowly. So there are two things. If your structure is wrong, suppose the complex with L1, that structure is wrong. So the problem is that you see in molecular dynamics simulation, you can create one structure and start running. Yeah. But maybe that is a wrong structure and you are in a minima which is completely not accessed by the process. So you are in a minima where you are stuck, that is not the real structure, but you keep on simulating your finite simulation. So you will, and that well is quite deep, so you cannot cross it. So you will get structures which is complete, and from there you wanted to compute some delta G's which are completely meaningless. So the good idea is that you have some very good idea about the structure that you have started with. Okay, so you know what L1 goes and bind, but you don't know L2, but you know L2 is very similar to L1. So then if you do this kind of techniques, okay, then it will really tell you something sensible. And you really understand what thing is causing this delta G larger, because you have enormous control. You can change, you can play with the electrostatics, you can play with the van der Waals, you can do many things, and you can see what is tuning what. You got my point? Yeah. You can change the protein. You can see how this delta G is tuned by all these things. But you have to very do it nicely. Okay. Is that answered your question or?
Yeah. Yes. Yes. Thank you, sir.
Okay. So yeah. Anyone have a question?
Hello, sir.
Yeah. Hi.
Please introduce yourself.
I am Anamika Shukla from Lucknow, BBAU. Sir, you told us that identity of a molecule is force field. So I just want to ask, in MD, we have a set of force field to choose, but does it depend on the molecules that we are going to deal with, or a different set of force field used with some different type of molecule can lead to various unexpected outcomes or something like that?
Okay, so you see, the question you have asked me, then I have to talk about the force field. Okay, now what I show you is that I have to describe a molecule, like water molecule. So that description, how I'm going to describe in terms of energies. So I have to feed you some constants, right? So that will tell you that if you stretch the water molecule, what will happen, how the energy will go rise or less. Now, different force field have a slightly different philosophy. Philosophy in the sense, maybe there is a one dihedral angle and you want to represent the dihedral angle. So you may use multiple cosine functions to to model that dihedral. Okay, so maybe charm 36 is using 3 or 4 functions, cosine functions. Amber may be using 10 functions. Okay, so they have completely slightly different philosophy altogether. The point is, this force field has been designed to reproduce experimental data. So suppose some molecules, I know the solvation data. So what you do is you describe this molecule with this constant, and you describe the water in like that, and then you run the simulation and try to extract structural quantity or your experimental numbers like solvation free energies, or maybe iron, how the water will be distributed around the iron, okay, the densities. So once you run it, if it is fitting well, okay, then you take that force field. Okay, this is fine. Then the idea is that when I suppose I have designed that for different amino acids, I have data, solvation data for different amino acids, and I run molecular dynamics simulation, and I am kind of tuning my parameters, epsilon, R0. Okay, I started with quantum chemical calculation, and then I modify it. So now they are matching with the experiment. So then I consider it that this is my force field. Okay, so then what you should not do, you should never do, is that mixed force field. So suppose if you are using charm 36, use charm 36 for your water, your protein and everything, because they have been done, the force field optimization done in a completely same way that has been done for all the other molecules. So if you mix force field, okay, they use different philosophies. Okay, so never mix the force field. That is the first thing. Second thing is the computation of the free energies, delta G's. Okay, so these delta G of binding affinities, as I said, this is very difficult to compute, and that is very heavily depends on your force field that you are using. Okay, so it is not guaranteed that if you use delta G, you estimate the delta G binding with amber, and you get delta G binding with say charm, you get a different number. This is not a surprise. You may get a different number. So it is always what you have to do is, it is better to do it as a relative terms, where you are kind of you did it with delta G L1, you computed with charm maybe delta G L2 if possible, vertical arms, you compute it, you take the difference, okay. Then you see if the two force fields are matching, and that you will see that they are matching quite nicely. So you see, absolute binding affinity calculations, that is something which is tricky, which we haven't discussed. You can do it with this free energy perturbation, alchemical transformation. It is possible, but that's a completely different thing. But take home is that, so always try to, when you try to see like this two different force field, don't compare the delta G's directly. So compare delta G's, so which you will see that it's much better, intelligent, I think, more knowledgeable approach to do. Okay, so never mix force field, never mix force field, and always compare force field with something delta G's. So that's the something.
Okay, so let us thank Dr. Satpathy for a nice and informative talk. This talk is available on YouTube, so if any of you have any more question, you can always send him. Thank you, Dr. Satpathy.
Okay, thank you.
Now, next talk will be given by Professor Sandeep Pal, and before that talk, we are going to have around 10 minutes break. We will start at 11:45 sharp with Professor Sandeep Pal. So, tea break. Welcome all. So in this session we have Professor Sandeep Pal. Professor Sandeep Pal is a professor in IIT Guwahati in Department of Chemistry. He did his PhD from IIT Kanpur under the supervision of Professor Amulendu Chandra. After his PhD, he moved to University of British Columbia, where he spent 2 years. And then he moved to University of North Carolina, where he spent 1 year. And after spending that time, he came back to IIT Guwahati and joined as assistant professor. Later on, he moved to the ladder and now he is a full professor. He has published several high-impact international journals, and all those papers are highly cited. Welcome, Professor Sandeep Pal. Now, this is your turn. Thank you.
Thank you, Hemant. Yeah. So first, good morning, everyone. And first, let me thank Hemant for his invitation and giving me an opportunity to interact with new PhD students or PhD students who are pursuing their PhDs in theoretical chemistry or molecular modeling or molecular dynamic simulation. So first, let me share my slides with you guys. So my slides are visible?
Yes, it is visible.
It is visible. Okay. So yeah, the title of my talk is Basic Idea of Molecular Modeling and its Application to Improve the Aqueous Solubility of Sparingly Soluble Drugs. So basically, I have divided my talk into two parts. The first part is maybe half an hour. I will discuss about very basic molecular modeling.
Keep it in the slide.
And sorry?
Presentation mode.
Sorry?
Presentation mode. Presentation mode.
Presentation mode. Yeah. It's fine now?
Yeah. Yeah.
Okay. So, and then I will discuss its application. Okay. Now, if we go back and see, okay, so this whole molecular dynamic simulation is based on classical mechanics. Okay. Rather, classical equation of motion introduced by Newton in 17th century. Okay. And in just a minute, please. Yeah. So, well, I'll just use my cursor. Okay. Yeah. Then in 19th century, Boltzmann he introduced statistical mechanics. Okay, you know. And in 19th century, Schrödinger came up with quantum mechanical equation of motion. We know this is time-dependent Schrödinger equations that we are all aware of. Now, if we look at the different milestones of molecular mechanics, we can see that, okay, it started in 1953, okay? So, by Metropolis. So, he first carried out Monte Carlo simulation of a liquid, and he modeled the liquid as hard spheres, okay? That's the simplest model one can think of. And then later in 1957, Wood, he did again MC simulation, Monte Carlo simulation with Lennard-Jones potential, which is a more realistic potential. Okay, and in 1957, first molecular dynamic simulation of a liquid hard sphere was done by Alder and Rahman. Okay, Rahman is from our India. In 1964, he did first MD simulation with Lennard-Jones potential. Okay, so again MD simulation with more realistic Lennard-Jones potential was done by Rahman in 1964. These are all liquid things, and if we go to complex system like proteins, okay, so we can see that, okay, in 1977, Martin Karplus and McCammon, they first studied MD simulation of protein, okay, and in Karplus also developed charm force field and MD program, and in 1984, Coleman did or generated or contributed to Amber general force field. And Carr and Pardinello in 1985 first did full quantum calculations. Okay, and Coleman in 1986, he first introduced us into hybrid quantum mechanical molecular mechanics simulations. Now the next thing is why do simulations? Okay, this is the basic question one can ask. Okay, so you see that in some cases it's very difficult to do the experiments like inside of star or weather forecast. It's too dangerous sometimes, like flight simulation or explosion simulation. Sometimes it's very expensive if you use very expensive chemicals, or very high pressure simulation or wind challenge simulation. And sometimes the time scale is so small that you cannot do the experiment. Okay, for that, this kind of blind system, we can carry out simulation. So simulation is a useful component or useful complement because it can replace experiment, it can provoke experiment, it can explain experiment, and aid in establishing intellectual property. Now, if we start with a real liquid, okay, so you perform experiments with the real liquid. Suppose you take water, okay, then you perform experiments, whatever experiments you want to do, and then you get experimental results of that liquid. Okay, now for that same liquid, suppose in water, you make models first, okay, and then you get, once you make the model, you get model liquids, right? Now for the model liquids, you carry out computer simulations, and then you get exact result for model. Remember, exact result for model, okay. It's not exact result, but it is exact results for model. Now, for same model liquids, you construct approximate theories, okay, and you make theoretical predictions. Okay, so what computer simulation does, it compares with the experimental results as well as it compares with the theoretical predictions. So it acts as a bridge between experiments and theory. So this is the beauty of simulation. So what is molecular modeling? Molecular modeling is concerned with the description of the atomic and molecular interactions that govern microscopic and macroscopic behaviors of physical systems. And what is it good for? The essence of molecular modeling resides in the connection between the macroscopic world and the microscopic world provided by the theory of statistical mechanics. So macroscopic observable, we see whatever, we do experiment and whatever we see, like solvation energy you can measure, and affinity between two proteins or distance between two atoms, etc., or conformation of a protein, those are macroscopic observables. And these observables, okay, so we can measure this observable or average observable properties over selected microscopic states. And what are the molecular modeling tools? You can do quantum mechanics. It deals with electronic structure of the molecules based on Schrodinger equation. It is accurate but applicable for very small systems that you know. Next comes classical molecular dynamics. Okay, it deals with empirical forces. Okay, as Professor Satpathy discussed. Okay, it deals with the empirical forces by solving the Newton equation of motion. As I mentioned that, okay, in classical molecular dynamics, we solve Newton's equation of motion. It's less accurate, but applicable for large systems, specifically for biomolecules. And then we have the modern, this mixed quantum classical QM/MM approach. As I just mentioned, that quantum mechanics, what it does, it gives electronic structure. It uses Schrodinger equation. It's accurate, but it's expensive. And it deals with very small number of atoms, 10 to 100 atoms, and for very small time steps or time scales like 10 to 100 picoseconds. In classical MD, okay, it's empirical forces. We solve Newton's equation of motion. It's less accurate, as I mentioned. It's very fast, okay, and we can deal with large number of atoms. Sometimes in millions of atoms we can deal with, okay. And typically we run with 10 to up to millisecond we can run nowadays. And then we have mixed quantum classical approaches. I'm not discussing that. Now, what is classical molecular dynamics? Okay. So simulation, as we mentioned, simulation of explicit particles we can consider. We can consider atoms, we can consider ions, we can consider full molecules, everything we can consider. Particles interact via relatively simple analytical potential functions. So they interact in very simple manner. I will discuss that in another few slides. And then Newton's equation of motion are integrated for all particles simultaneously. This is very important. Simultaneously we solve or you integrate the Newton's equation of motion for all particles. And we can use millions of particles and up to millisecond we can go. Okay, so we can use for large time scale. Another few important properties or aspects of classical molecular dynamics is we can change temperature, we can change pressure. We are able to treat comparatively large system for relatively long time, as I mentioned. It helps interpret the experiments and to provide alternative interpretation. Okay, we can go to molecular level and then we can see what is happening there. And we can test theories of intra and intermolecular interactions, obtain detailed molecular level information, and structure and dynamics. Both we can obtain from classical MD simulation compared to quantum calculation or molecular mechanics or Monte Carlo. Okay, so Monte Carlo, like Monte Carlo deals with only structures. So dynamical part is absent there. So we can improve our understanding through model building. Now this is basic statistical mechanics. Okay, so what is space and time averages? So a system, suppose a system contains n number of atoms. So for each atom we have 3n. We have 3 coordinate and three number of momenta. Okay, so for n number of atoms we will have 6n number of variables, and each combination of coordinates and momenta define a point in the 6n dimensional phase space equal tau n, and consider a property that can be ascribed as a function of the coordinates and momenta. Suppose the property we consider as a tau, and the average value of the property we can write a observable, or we can this time average or this ensemble average, and this is the time average. If we do the integration, we get the time average. And we know that ergodic hypothesis also. So it says that long time averages, the long time average is equal to the ensemble average in the limit of an infinite number of members in the ensemble. So in molecular dynamics, what it does, a molecular dynamics simulation generates a sequence of points in phase space connected in time. The result is a trajectory of all particles in the system as a function of time. And time averages and other properties can be calculated from this trajectory. So what we do, we run the simulation. Simulation is nothing but it generates a data point or position and velocity or position and momenta of all the atoms at different time steps. Okay. And motion of the system through phase space is governed by Hamiltonian equations of motion, where here H is Hamiltonian. And you know that Hamiltonian is sum of potential and kinetic energy and potential energy term, and kinetic energy term is determined by this expression. And then we have the Newton's equation of motion, and we solve the Newton's equation of motion F equals to ma. You know. Now, how do the particles they interact? Okay, the potential energy can be divided into interaction between pair, triplets, or quartet, like that. So 2-body, 3-body, 4-body interaction. The sum of that. So here I am showing 3 particles I, J, and K. If you see the first figure, okay, so I and J first they interact. We get V, which is function of Ri and Rj. And J and K, then they can interact. So V, R, J, K, and so on. So these are 2-body interaction terms. Okay, pair interaction terms. This is the first term in this expression. And then all these 3 particles, they can interact simultaneously. So we will get 3-body interaction term, and then many-body interaction terms will be there. Okay. So the total potential energy is the sum of the 2-body term, 3-body term, 4-body term, like this. Okay. But the beauty is the interaction energy is usually or largely dominated by the pair term. Only the two-body term is sufficient to get the interaction energy of our system. So we can truncate up to in the first term. So in simulation, what we do, in simulation, the true pair potential is usually replaced by an effective pair potential, which incorporate the average many-body effect in the first term. So basically, we tune the two-body term, okay, to get the effective pair potential. And these potential are usually parameterized to reproduce some experimental data that Professor Satpati described. I will also describe that part. So the total potential energy function, it consists of, if you see the expression, it consists of 6 terms, right? The first 4 terms are intramolecular terms, okay? And the last 2 terms, they are non-body interaction terms or intermolecular interaction terms, okay? So if you concentrate on the last 2 terms, what do you find? You find there is a short range part. The first part is Lennard-Jones part or short range interaction part, and the second part is Coulombic interaction part, which is long range part. And for a system with only 1,500 atoms, 10 to the 6th pair of interacting atoms are there. So we need to consider that many interactions for only 1,500 atoms. If you increase the number of atoms to millions, so it will increase like anything. Okay. So in modern days of computer, you cannot really calculate all those interactions. That's why we introduce a cutoff radius there. I will discuss that. What is cutoff? So as I mentioned, okay, so bonded term is like this intermolecular term. Okay, so this R naught is the equilibrium bond length. Okay, and then this is the term for bond interacting term, and then you have the angle term where theta naught is the equilibrium angle, and so on. And dihedral angle, improper angles are also defined like that. These are all textbook things. And in the non-bonded interaction, that is the very important thing. Okay, we have, as I said, we have a short range or Lennard-Jones potential term. So if you see the Lennard-Jones interaction potential term, sometimes it is called 12-6 potential, because you can see that it raised to the 12 and raised to the 6, the powers are 12 and 6. Okay, and why it is called short range? Because it dies up very quickly. Okay, it dies up very quickly, so that's why we call it a short range interaction term, Lennard-Jones potential, or use this one. And then we have that it can be plotted like this. Okay, so if you plot this Lennard-Jones term in VW versus R, you will get this kind of plot. Okay, so it consists of 2 terms. So one is attractive term, the minus 2 times or minus sigma by r to the 6. This is your attractive term, and sigma by r to the 12 is repulsive term. So when the distance between 2 particles is very large, when R is very large, the attractive part dominates over the repulsive part. So you get this kind of thing, negative value, and then the distance between 2 particles is very small. The repulsive part dominates over the attractive part. Okay, so you get this kind of sharp increase. Okay, that's why you get it passes through a minimum. Okay, and that minimum is epsilon. That epsilon is here in this in the expression, and sigma is also there. You can see the sigma here. Okay, in the expression. Okay, and then the electrostatic interaction. This is called Coulomb. We use Coulomb's law for that. And here Qi and Qj are the partial charges of atomic site I and J. And Rij is the distance between between them, and you have the dielectric vacuum and those terms that are there. So you are all aware of this thing. Okay, now in MD we actually use periodic boundary condition. Why we use this? Because a real system might contain about Avogadro number of particles, right? Suppose if you take a glass of water. Okay, so it will have Avogadro number in the order of Avogadro number of particles, right? But in modern days of computer, you cannot handle those many number of particles. Okay, so simulations may contain only 100 to million number of particles, which is much smaller than the Avogadro number of particles. Now, if you take a small cubical box of 10 angstrom edge length, okay, so you will find that half of the particles are present at the surface. Okay, but your goal is to calculate bulk property. Like suppose you take a glass of water, you want to calculate the bulk property of the liquid water. But if you take a small box like 10 angstrom edge and if you slowly introduce introduce the particle, water molecule there, you will have maybe 1,000 or 1,200 water molecules there, and you will find that half of the particles are present at the surface, not at the bulk. So your properties that you will be getting will be dominated by surface, not the bulk properties that you are looking for. Okay. So what we do, we use periodic boundary condition. We use a central cell. Okay, and we put or we incorporate the number of particles we wanted to put. Typically, we use 30,000 or 20,000 particles inside the main simulation box here, and then we replicate actually, and then we replicate the main simulation cell in all three directions, and there are no walls or no surface, and then once a particle, you can see the red particle here. Once it leaves this box to the next box, another particle from other side, it comes into the main simulation box to maintain the total number of particles fixed in the main simulation box. Okay. Some systems inherently Hello.
Yes. Please go ahead.
Okay. So something happened here actually. Okay. So some systems inherently contains a boundary, for instance, a liquid droplet or surface. So if you want to calculate surface property, so you have to be a little bit careful about applying your boundary conditions. Okay. But if you want to calculate bulk properties or liquid properties, you have to use periodic boundary conditions in all three directions X, Y, Z. Okay. Now in simulation we use minimum image convention. What it does, as I mentioned, so if you use only 1,500 atoms, essentially you need to consider 10 to the 6 number of interactions. So huge number of interactions you need to consider. But what here we do actually in minimum image convention is individual particle in the cell interacts with the closest image of the remaining particle in the assay. So red color is our reference particle, and it interacts with the images of this remaining particles. Okay, and in Fortran you can really introduce this minimum image convention by using this kind of code. I have given the code also. And as I mentioned, we use a cutoff, because we cannot deal with 10 to the 6 number of interactions. So what we do, we usually use 10 to 12 angstrom cutoff. Means we take a reference particle here, and from that reference particles, the number of particles which are present within that cutoff distance, only those interactions we consider here. Okay. So very commonly, minimum image and spherical cutoffs are used together. We usually use both minimum image convention as well as spherical cutoff. Now solvation. Okay. So suppose you want to simulate a protein molecule. Okay. So what do? You take a protein molecule and now you want to solve it. You can use real solvent, real in the sense real model solvent like water, or water alcohol mixture, or whatever you want. Okay, so those are called explicit solvent. Okay, so you can use the solvent molecules in real manner. Okay, or you can do implicit solvation also. So for that you have to adjust the effect of solvent, by using a screening constant. Okay, so mostly people use for atomistic simulation use explicit solvent. But for very, very large systems, sometimes they use implicit solvent. So here you can see that I have shown one right side, right bottom. Okay, you can see one figure. Okay, I have used one protein molecule solvated in water. Okay, so I am using real solvent here. As I mentioned, implicit solvation, you can use Poisson-Boltzmann equation, or you can use generalized Boltzmann equation. Okay, so these are all textbook things. I can just keep those things. And now the most important part is we need to integrate the Newton's equation of motion, as I said. So motion of all particles. So you have thousands or millions of particles in your system. So motion of all particles is coupled. Okay, so they are not independent. The motion of the particles, because they are interacting. Okay, so motion of all particles are not independent. Okay, so motion of all particles is coupled. Okay, that's why it's many body problem. And this is impossible to solve analytically. So you cannot solve it analytically. So you have to use numerical method to solve the Newton's equation of motion in simulation. Okay. So how do you do it? Okay. So first you expand in Taylor series. Okay. So R t plus delta t. T is at a particular time t. T means, and delta t is the time step you are using. So we use usually 1 femtosecond or 2 femtosecond time step. So delta t is the increment in the time step. Okay, so R t plus delta t, if we expand in Taylor series, we get this kind of expansion, right? That we are aware of. Similarly, velocity of that particle, of a particular particle, you can expand it like this. Okay, and acceleration like this, and so on. Okay. And the simplest method to truncate the series after the first order term. Okay, the simplest method is, okay, so you truncate after the first order term. Okay, so but it is not advisable. Okay, because the first order terms should not be used. Okay, it will give you a lot of error. Okay, then in 1968, Verlet came with one algorithm. Okay, here t plus RT plus delta t can represent it by RT and this term, and RT minus delta t means one time step back. Okay, so this is the expression. Okay, and if you rearrange them, you get like this. Okay, now it has also this disadvantage. So what is the disadvantage? The advantage is that the Verlet algorithm is a second order algorithm, not like the first one where we can truncate up to the first order. But the drawbacks of the Verlet algorithm is velocities do not appear as they have been eliminated by the addition. Okay, so we don't get velocities there. Okay, and it is not a self-starting algorithm, and a small term is added to the differences of two large terms. Okay, these are the drawbacks of Verlet algorithms. Then comes leapfrog method. Okay, so this is a leapfrog method, but again, in the disadvantages, velocities and positions not available at the same time. You can see in the leapfrog method the expression RT plus delta t, and V is t plus half delta t. So okay, so velocity and the position you don't get at the same time. That is the disadvantage of leapfrog algorithm. And we mostly use velocity Verlet, and these are the expressions for velocity Verlet method. It's solved in 2 steps with a force evaluation in between, and it yields the position, velocities, and acceleration at the same time. So this is the method we generally use in our MD simulation. So we use velocity Verlet method in our MD simulation method because of these 2 advantages. Means it's solved in 2 steps with a force evaluation in between, and it yields the, most importantly, it yields the positions, velocities, and accelerations at the same time. And apart from numerical differences, the 3 Verlet results is in the same trajectories, but there are disadvantages, as I said. Now the most important thing is how do you choose the delta t or the time step in simulation. Okay, how do you choose it? Typically, we need to use 1 order of magnitude lower than the period of fastest motion in the system. Okay, so if you concentrate on the example 2 given in the bottom, so you consider water molecule, the fastest motion is vibration of with stretching mode, right? And the period of vibration is typically 4000 centimeter inverse, and if you convert into time scale, you get 10 to the minus 14 second. Okay, so delta t you usually use 10 to the minus 15 second, or in femtosecond ranges, 1 femtosecond or 2 femtosecond you usually use time step. Okay, so in order to choose time step, you have to be very, very cautious here. So you should not use too small or should not use too big. Okay, there are disadvantages there. So in brief, what we do in simulation, okay, we calculate position at time t plus delta t or dt, and it is determined by the position at time t and t minus delta t, and by the acceleration at time. And the equation of motions are deterministic, that is the positions and velocities at time 0 determine the position and velocities at all other times t. Okay, so in MDM algorithm, what we do? Okay, so we calculate force at per time t, and then we update position, and then we again move, and then we again calculate force, and like this it goes on and on. Okay, so what are the steps in MD simulation? We first select interaction model. How do you select it? I will discuss. Professor Satpathy also discussed. Selection of boundary conditions. As I mentioned, if you want to calculate bulk properties, you should use periodic boundary conditions in all three directions. If you want to choose, if you want to calculate surface property, then you have to be very cautious. You have to modify the things, okay, accordingly. And select the initial conditions like position, velocities. Selection of ensemble, because the properties you are looking at, okay, so essentially it does not depend on ensemble, but sometimes you have to choose your right ensemble, okay, for your system. A selection of target temperature, density or pressure. Suppose you want to calculate 300 Kelvin, if you want to simulate something in 300 Kelvin, you use 300 Kelvin, or you want to 400 Kelvin, you can also do 400 Kelvin. Pressure 1 bar or 1000 bar or 10 to the minus 3 bar. So you can select temperature, you have to select temperature, pressure, etc. You have to select the indicator like velocity varlet or whatever you want to use, that you have to use. You have to use thermostat. If you want to keep the temperature of the system constant, you have to use thermostat. If you want to keep the pressure of your system constant, you have to use byrostat. And you have to perform simulation until equilibrium is reached. That is kind of your experience will tell, okay, whether the system has reached equilibration or not. It's property dependent. It depends on which property you are looking at. Accordingly, you have to run the simulation. We have to perform, then after doing the equilibration, you have to perform the production simulation to collect thermodynamic averages, okay, positions, velocities, etc. And then you have to analyze the result via post-processing. So essentially, you will get trajectories, okay, of different atomic sites at different times, and then you have to do the analyze the data to get your desired result. So how do you parameterize the force? Okay, so I highlighted in red this one. You concentrate on the red one. Okay, so usually we look at thermodynamic data, experimental data. We look at thermodynamic data, and then type of system we consider molecules in solution or mixture, and phase is condensed phase we usually consider. And type of property we look at for this experimental thing is heat of vaporization, density, free energy of solvation, etc. And we match with the experimental data, and accordingly we tune sigma, epsilon, and partial charges of different atomic sites. Okay, suppose you start with something sigma, epsilon, and partial charges of different atomic sites, and then you calculate heat of vaporization, and then you compare your result with the thermodynamic data. Okay, so if you find they're matching, closely matching, okay, so your data are mostly okay, you can go ahead. If they are not matching, they deviate a lot, then you have to tune those things, sigma, epsilon, and q, or partial charges. Okay, so that's how you do the parameterization in MD simulation. And these are the different softwares people usually use. You can use CHARM. In CHARM, you can use both. E stands for explicit, I stands for implicit. You can use both explicit and implicit solvent in CHARM. For all atomic simulation, you can use and unitary atom simulation. Unitary atom means suppose you have a methane or ethane rather. Okay, you have ethane. Ethane is CH3, then bond CH3, right? So that CH3, you have two CH3 part, and then you can model each CH3 unit as 4 different atomic sites, like C is 1 and then 3 hydrogen atoms. These are called all atom model, and unit atom means the whole CH3 unit you consider as one atom, and you have the parameter for that whole CH3. That is called unit atom. Okay, so in CHARM you can do both all atom and unitary atom. In amber, you can use both explicit solvent and implicit solvent and all atomic simulations. And bromos, you can use in vacuum or unitary atom model and explicit solvent, and bromax and MD all are there. Okay, so bromax is free, NMD is free, charm and amber they charge, but they charge reduced amount for academics. Okay, so you use whatever software you want to use. Minimization part. Okay, so here the little video I'm just showing you. I used high peptide molecules in water, and you can see that particles are moving. They are going from one end to another end, and you can see that yellow lines are like beta sheet. Therefore, they are forming and so on. Okay. Now come to application part. Okay, so the basic molecular dynamics simulation we just discussed, and we'll see how we can use this molecular dynamics simulation to improve the solubility of a sparingly soluble drug in water. Okay, and this work is done by Subodip. He is now doing postdoc in Germany. So how do we take drug? Okay, drug means tablet or capsules mainly. Okay, what do we take? We take a glass of water, right? And we take a pill. Okay, and then we take the pill and take the glass of water. Okay. So what is the most important thing here? The important criterion is the solubility of drug in water. Okay, the drug has to be soluble in water because most of the drug we take orally. Yeah. Okay, so the drug has to be soluble in water. That is the most important part. But the problem is the insolubility. The medicinal chemists who are developing the drugs, they are obsessed with affinity and selectivity of the drug. Okay, solubility they really don't really think of. Okay, so solubility can get left by way aside. And general tendency is that the new drugs are becoming more insoluble. Okay, so you do not consider the solubility of the drug before formulating it. Okay, so after developing the drug, okay, you find that, oh my God, the drug is insoluble in water. Okay, and then the formulators are they are going crazier. Okay, they are becoming mad now. Okay, so how do we overcome this problem? So we can overcome this problem by the use of hydrotrope. Okay, so many of you do not know hydrotrope. So I'll in couple of slides I'll show what is hydrotrope. Okay, so hydrotropes basically are a unique class of amphiphilic molecule. Amphiphilic molecule, you know, amphiphilic molecule means you have a molecule which has both hydrophobic as well as hydrophilic part. So hydrotropes are a unique class of amphiphilic molecules that can enhance the solubility of the insoluble or sparingly soluble molecules in water. Okay, so this term of hydrotrope and the concept hydrotropy, this phenomenon is called hydrotropy, and first reported by Newark, okay, in 1916. So 100 years ago, he gave this concept, okay. But the unfortunate thing is not many research work has been done so far, either experimentally or theoretically. Okay, so hydrotropes are classified into three categories like anionic, cationic, and non-ionic. Anionic we mostly use, okay, so and they are classified according to the charge on the head group. Okay, as I said, head group is amphiphilic and the tail part is hydrophobic for hydrotrope, and so the charge of the head group determines whether it is anionic or cationic or non-ionic. Okay, and we usually mostly use anionic. And some of the, here you can see some of the anionic hydrotropes. Okay, so we in our study we use the one I showed here in blue color. I just encirculated this one. Okay, you can see that, okay, it has a hydrophilic moiety here, SO3, right? Hydrophilic moiety SO3 minus, and it has a small hydrophobic tail part CH and CH3, right? Okay, so this is the hydrotrope we have used in our study. So what are the applications of hydrotropes? Hydrotropes have many practical applications as well as they have some biological importance too. Hydrotope additives find a wide application in the industries such as detergent formulation, healthcare, and household purposes because of their efficiencies in both aqueous and oil phases. They are also extensively used as solubilizing agents to solubilize drugs, biochemicals, dyes, and organic compounds. They have also been tested for extractive separation processes. Okay, so there are several in paper and pulp industries also. They are used. And hydrotopes can affect the formation of micelles and microemulsions. Okay. Now you can see that. Okay, so there are similarities and differences between hydrotope and surfactant, because in surfactant also you will find one hydrophilic head group and one hydrophobic tail group. Okay, but well-known surfactant possesses a long tail alkyl carbon. Okay, so in surfactant you usually have a very long tail hydrophobic chain, alkyl chain, usually 8 to 20 carbon atoms. Okay, whereas hydrotropes, they contain short alkyl chains. Okay, usually 0, no hydrophobic alkyl chains, or up to 4. Okay, up to 4 number of carbon atoms can be present in hydrotrope. Okay, so alkyl chains means 4 number of alkyl chains can be present in hydrotropes. Okay. So, but similarity is also there, as like surfactant forms micelle above its critical micelle concentration, CMC, above which the surfactant they form micelles. Hydrotopes also shows aggregation behavior above minimum hydrotope concentration, we call MHC. Okay, so hydrotope also they aggregate above a certain concentration, we call it minimum hydrotope concentration. Now, what is the mechanism of hydrotopy? Okay, so there are three possible mechanisms or three possible hypotheses proposed so far. One is complex formation between solute and hydrotope. So suppose this orange one is our solute and green one is our hydrotope, they form a complex like this. So this is one mechanism. Second mechanism is perturbation of tetrahedrally located water structure. If you know that in water, in pure water or liquid water, okay, so each water molecule is forming 4 hydrogen bonds with neighboring water molecules, right? Okay, and they form a network that is called tetrahedral network. Okay, so what these hydrotopes do, okay, they can also break that tetrahedral network of water, and then those water molecules are free and they can help in improving the solubility of the drug molecule. This is another hypothesis. And the third hypothesis is self-aggregation of hydrotope molecules similar to surfactants, and that helps to solubilize insoluble molecules. Okay, so these are the hydrotope molecules. Okay, they self-aggregate like this, and inside they encapsulate the drug molecule. Okay, now the problem is, how do hydrotopes aggregate? This is the common question. But again, it is not known. As I said, that this is not it's not explored so much. Okay, so how do the hydrotope aggregates? Still, we do not know. So, what is the goal in today's presentation? Our goal is to study or see how do hydrotopes aggregate in aqueous solution, and how do hydrotopes help to solubilize sparingly soluble drug molecules. Now, first we'll see how do hydrotopes aggregate in aqueous solution. Remember, over last century, a few experimental works have been done so far, but to our best knowledge, this is the first molecular dynamic simulation of hydrotope molecules we have carried out. Okay, so what we studied, we considered, as I said, we considered sodium cumene sulfonate. These are the atom numbering from 1, 2, 3, 4 to 9. So 9 carbon atoms are here, and SO3 is there, and Na+. And its appearance is white powder or liquid. Solubility is soluble in water. Stability, it is very stable. Biodegradability, it is biodegradable. Characteristics, excellent hydrotropic, viscosity reducing, pore pointing, depressing, anti-caking, and dispersing, emulsifying abilities, all are there. And toxicity, that is very important, because our goal is to improve the solubility of the drug by using hydrotropes. But if the hydrotope is itself toxic, then we cannot use it, right? And we have found, okay, no, toxicity is very low for humans for this drug, for this hydrotope. And it has several uses. How this is a simulation protocol. We have used SPC/E model of water. The students who are familiar with MD simulation, you know that there are several models of water, tip 3P, tip 4P, SPC, SPC/E, so on. Okay, so we have used SPC/E model for water, and charm general force field we have used for sodium cumene sulfonate, and we have used AMBER 12 software to calculate the simulation. And we have used periodic boundary condition in all three directions, X, Y, Z, and we have used Selk algorithm to constrain the covalent bonds between hydrogen and heavy atoms. And we have used Langevin thermostat to get the desired temperature. An initial configuration we prepared using PACMOL program. PACMOL is a very good software. You can use it. It's free to packing the molecule inside a box. It's very useful. And at first we use canonical ensemble. Okay, and we heated every system slowly from 0 Kelvin to desired temperature for 320 picosecond at 1 atmospheric pressure. And the systems then were equilibrated in MMTT ensemble at 1 atmospheric pressure for 5 nanoseconds. After that, we ran the production run for 40 nanoseconds in MMTT ensemble, and cutoff distance we have used 40 angstrom, and long range interactions we treated with particle mesh UART method. Those who are familiar with MD simulation, they know what is UART summation method. Okay, and in total we have considered 9 systems, and for each system we considered 4 different temperatures. Okay, so we level the system S0 to S8, and for every system, suppose S0, we consider 4 different temperatures: 298 Kelvin, 318 Kelvin, 338 Kelvin, and 358 Kelvin. And for S0, we have used 5 sodium cumene sulfonate molecule in 3600 water molecules, and this is the box volume in nanometer cube. Okay. And small m stands for molality here. Okay, and why we have used molality here? Because molality does not depend on temperature, but molarity does. Okay, so for each temperature we have the same molality here. Okay, because if you increase temperature, volume changes, so molarity will change, but molality will not change. Molality depends on weight. Okay. So molality does not change. That's why we have used molality here, because we consider 4 different temperatures here for each system. So for, as I mentioned, for system S0, we have used 5 sodium cumene sulfonate molecule dissolved in 3600 water. And if we then from S0 to S1, we increase number of sodium cumene sulfonate molecules, keeping number of water molecule fixed up to system S7. And you can see the molality of SCS increases. Okay, and system 8 we consider for only pure water without any hydrotrope is sodium cumene sulfonate. Okay, so we have used only 5. We use this one to get the density of water at different temperatures. Okay, these are the systems. Now we verified our result with experimental results. Okay, and we calculated few very basic thermodynamic quantities like apparent molar volume and partial molar volume. Okay. Now, if we plotted for all 4 temperatures, we plotted apparent molar volume and partial molar volume as a function of molality of sodium cumene sulfonate. Now, if you consider on 298K, which is shown in black, you can see that okay, it starts increasing. If we increase concentration of hydrotrope, it starts increasing up to this, and then okay, after that it almost stopped, and then here it is stopped, and there is not much change after here. Okay, so partial apparent molar volume increases up to 0.185 to be exact molality, and it stops around 0.37 molality. Okay, after that not much change, and the similar trend has been observed for all 4 temperatures. Okay, and inside in this figure you can see that we have shown the experimental partial apparent molar volume and partial molar volume, okay, for different temperature. You can see the trends are very similar. Our trend means the simulated result, the trend of the simulated result is very similar to that of experimental results, but the numerical value is slightly different. Okay, that is applicable for both apparent molar volume and partial molar volume. Okay, so our simulated results matches qualitatively well with the experimental findings. Okay, and then what we did here in the right hand side of the top figure, we plotted phi v naught. Okay, phi v naught is your apparent molar volume at infinite dilution for different temperatures, and we plotted apparent molar volume at infinite dilution temperature and infinite dilution for different temperature. If we plot it, we get a straight line like this. Okay, and how do we get this infinite dilution value? We can just extrapolate this data, and then we get the phi naught value here, and then we can plot it here. Okay, and then we further compared with different experimental findings. Suppose you consider the molar expansibility at infinite dilution, we can calculate, and experimental value is 0.146 and our calculated value is 0.2155. Yeah, there is a difference between experimental value and simulation value, but that is not that much. And you can see that if we differentiate this delta V naught by del T and we get a negative value, means a very small negative in fact. So yes, water structure breaks a little bit here. Okay. And then here the last few lines, if you concentrate, what we calculated here, we calculated the volumetric change on aggregation with respect to the number of carbon atoms, or delta Vm/n, and we get 0.719 for our hydrotrope, and for surfactant it is 0.78 to 0.91, 0.9 like that. Okay, so it's a little smaller than the surfactant. Then we calculated osmotic coefficient. Again, osmotic coefficient you can calculate very easily from activity coefficient. How do you calculate activity coefficient in simulation? It is like you calculate how many of them form clusters. Okay, how many of them aggregate and they form clusters. So you calculate total number of monomers, how many monomers are there, and then total number of molecules in solutions. And then if you take their ratio, you get activity coefficient. So once you get the activity coefficient, you can easily calculate osmotic coefficient. Okay, and this is our simulated value, this osmotic coefficient as a function of molality for different temperatures. You can see it starts decreasing if you keep on increasing molality of the solution. Then after that there is not much change. This is our simulated value, but this is the experimental value. And experimental value shows okay, it keeps on decreasing. So here we do not see not much matching between experimental and simulation value, in particular at higher concentration. Okay, lower concentration values, okay, our simulation values match well with the experimental value, but at higher concentration of hydrotrope, they don't match well. Then what we calculated? We calculated okay, average number of first cell water molecules. First cell means okay, so we usually calculate radial distribution function, and then from the first minimum we get the distance of the first cell. Okay, so the first cell radius value we get, and then we calculate the first cell water molecules around C7 and C8. If you remember the figure I shown you for our hydrotrope, C7 and C8 carbon atoms are hydrophobic. Okay, and we calculated now average number of water molecules around C7 and C8 for different concentrations and for different temperature. And if you consider only one temperature, suppose 298 K, if you consider, you can see you keep on decreasing. Okay, and in the parentheses I have shown some values. These values are like okay. So if you remember from S0 to S1. We have kept water number fixed, but we have increased the number of hydrogen molecules. Box volume increases, so that because of that actually, so you can get little increase or little change in the value. So that value is shown here. So this is the kind of expected value, and this is the real value we have got. And you can see that okay, around point your S3 system, the difference between the expected and the actual value is quite large. Okay, around 2 number of water molecule difference is there. Okay, And here for S7 system, difference is 6. 6 water molecule difference is there. Okay, and this trend is for other temperatures also this trend is there. Okay, and here we show the contour plots of solvent water density. Okay, so and for around C7 atom, and you can this green color is the water molecules. Okay, and for different time steps. Okay, this top panel or top 4 figures are for 298 Kelvin, and bottom 4 figures are for 358 Kelvin. And if you can see that as simulation progresses, okay, so you have less and less water molecules there. Okay, around C7 atoms. And you can see the clustering of sodium cumin sulfur hydrotop molecules through hydrophobic tail. Hydrophobic tail are in blue color. You can see that they're coming closer and closer, and they form aggregate. Okay. Then we calculated hydrogen bond properties. For hydrogen bond, actually for calculating average number of hydrogen bond, we usually use geometric definition that is well accepted in literature. So geometric definition of a hydrogen bond says, okay, so the donor acceptor distance has to be less than a cutoff value that you fix by yourself, and donor hydrogen acceptor, that angle has to be greater than 120 degree. Again, you can change this value 120 degree to something else according to your choice, and these two conditions are to be satisfied simultaneously. Okay, both conditions have to be satisfied. And here D and A represent donor and acceptor atoms, and each is the hydrogen atom. Okay. And here we show the SCS water and per SCS hydrogen bond, average number of hydrogen bonds, okay, for different temperature and for different system. If you see that for consider only 298 K suppose, and for system S0 we had the value of 4.74. And if we change from S0 to S7, there is hardly any change in the average number of SCS water hydrogen bonds. So SCS water hydrogen bond does not change if we increase the number of SCS molecules in our system. And these are the snapshots for different system. Again, the top panels are for 298 Kelvin and the bottom panel for 358 Kelvin. And you can see this is for S0 system. First one is for S0. S2, S4 and S7, and if you see the S4 system or S7 system, S4 system aggregate stars, S7 all of the molecules, almost all of the molecules are aggregated. Okay, I have not shown water molecules here, and the blue dots are ions here, or sodium ions. So water molecules are just left up for better visual clarity. And you can see in for 358 Kelvin also, a lot of aggregations are there, and even aggregation is more at 358 Kelvin. So in conclusion, self-aggregation of SCS molecule starts at 0.185 molar, as I mentioned, but above 0.375 molar concentration, the change in these quantities are not quite dramatic, suggesting MHC regions of SCS solutions. MHC means minimum hydrotrope concentration that I mentioned. An interaction between the hydrophobic tails of different SCS molecules which contributes to SCS aggregate formation. So SCS, they form aggregate in water. The effect of elevated temperature on this hydrogen process is also quite visible, and volume change accompanying the aggregation value indicates hydrotopes form more close packed structure than classical surfactant. Okay, so volume change is lower there, we have seen, than the classical surfactant. So in brief, they aggregate like this. Okay, but so far we have not introduced any drug in this system. We have just considered how SCS molecule or the hydrogen molecules they aggregate. Now we introduced, now we studied we applied these hydrotopes to solubilize a sparingly soluble drug molecule. For drug, we consider a glyceofulvin drug, which is a lipophilic, orally administered fungistatic antibiotic drug. And its water solubility, you can see it is very, very small, 8.64 milligram per liter at room temperature. Okay, and this is the structure of glyceofulvin molecule. And it is believed that it can also act as a potential anti-cancer drug. And glyceofulvin is one of the highly prescribed antifungal drugs, which with an annual worldwide business of 63.7 million USD, whereas its annual consumption is 85,000 kg. Okay, so it's very useful drug. And according to biopharmaceutics classification system, glyceofulvin falls into class 2 drugs that have low solubility and high permeability character. Okay, so it does not solve in water. Okay, that much. Okay, it's very sparingly soluble in water. And for this study, we considered 8 systems again, and they are labeled as S0 to S7. You can see we have used 8 drug molecules for each system, and we have used 24 hydrotrope molecules. Sodium cumin sulfonate again we have used for our study, as I mentioned. 24 molecules in cumin sulfonate we have used, and we have used different number of waters. Okay, we change number of water molecules. Actually, we decrease the number of water molecules from S0 to S7. Basically, we are increasing the molarity of SCS. Now we are considering molarity because we are considering only room temperature thing, only one temperature. So molarity we are using, molarity of the SCS we are increasing from 1.5 to 0.88. Okay. And as I mentioned, SCS they form clusters as we increase the concentration. And in the previous slide, I showed that okay, concentration increases from S0 to S7. So here we calculate the average number of clusters. So this bar diagram is in a bar diagram, and their probabilities. Okay. So suppose if you consider for S0 system, we have almost 60% probability of having monomers. Okay, they are not aggregated, and around 25% or 15% dimers are possible, and then 25% trimers are possible like this. And if you concentrate, suppose for S7 system, you can see that okay, 20 mers there is a considerable amount of probability having 20 mers, like 20% probability is having 20 mers, and then 40% is having probability 19 mer, and so on. There is very few monomers are there. Probability of forming monomer is very less for system S7. Okay. Now the numbers showing above the bar graph are like the number of particles present inside that aggregate. Okay, so suppose for system S0 we have a 20 mer here, and on the top of the bar graph, if you concentrate, you see that 2 is written there. Okay, means 2 drug molecules are there inside the hydrotrope. Similarly, for 19 mer, 4 drug molecules are there, and then in a 16 mer, 2 drug molecules are there, because we have used 8 drug molecules, so all 8s are here. Okay. Now we can see here we are showing that the density of water around the drug molecule for different system. Okay, so D is your highest concentrated system, S7. So pink color is the drug molecule here, and you can see the very less water density is there around the drug. And here we calculated the density of hydrotrope molecule around the drug. Okay, so you can see again we concentrate the most highly concentrated system D. Okay, figure D. If you concentrate, you can see that okay, you have a drug molecule pink color, and then high density of hydrotrope molecules around the drug molecule. Okay. Then we calculated water, drug, and then water hydrotrope hydrogen bonds. You can see the water hydrogen bond decreases slightly. Okay, it has to, because we have decreased the number of water molecules here, but the drug water molecule, that is the most important thing. And you can see that for system S0 we had on average 2.59 drug water hydrogen bonds per drug molecule. Okay, and if we keep on increasing the hydrotrope concentration, it decreases, and it decreases, and it is 1.56 for S7, and the expected number of hydrogen bond was 2.15. Okay, so water and the drug, the hydrogen bond number decreases. Okay, as we increase the concentration of hydrotropes. Then in the right figure, what we did here, actually we decomposed total drug and hydrotrope interactions into 2 parts: van der Waals part and electrostatic part. Okay, those are non-bonded interactions that I discussed. Okay, so we had 2 non-bonded interaction terms. One is van der Waals, another one is electrostatic interaction. So we decompose total interactions, drug and hydrotrope interaction, into van der Waals and electrostatic part. And then we added van der Waals and electrostatic, and we get the total interaction energy. Now you can see for every system, in the total interaction, total drag hydrotrope interaction, the van der Waals interaction dominates. Okay, so it is the van der Waals, or it was the van der Waals drag. It is the drag hydrotrope van der Waals interactions that plays a very important role in this kind of interaction. Next, we calculated Flory-Huggins interaction. Okay, interaction parameter. Okay, so why it is required? One of the most important drawback in simulation is we cannot calculate solubility of any molecule by classical MD simulation. Okay, directly we cannot measure solubility of anything in MD simulation. Okay, so we have to use something else. Okay, so that's why we calculated Flory-Huggins parameter, Flory-Huggins theory, and this theory actually was developed mainly for polymer solvation, but we have used modified Flory-Huggins theory. Okay, and this modified Flory-Huggins theory, it allows us to consider intermolecular interactions between the molecules that was not possible in original Flory-Huggins theory, as well as we can calculate Flory-Huggins interaction parameters Xi-FH. We call Xi-FH as a Flory-Huggins interaction parameter as concentration changes. Okay, so this concentration dependent Flory-Huggins parameter, as well as the interaction between the molecules are absent in original Flory-Huggins theory, but in modified Flory-Huggins theory we can actually calculate. Okay, and it says that okay, the Flory-Huggins interaction parameter is less than 0.5, then it is soluble. So how did we calculate Flory-Huggins interaction parameter? Okay, we calculated Flory-Huggins parameter by calculating V reference, delta E mix and RT. R is as usual gas constant and T is the absolute temperature. And what is V reference? V reference is the smaller volume between SCS and drag. So we have SCS and drag, the molar volume which is having the molar volume, we will use that. And delta E mix, we can replace delta E mix by delta H mix or enthalpy of mixing. Okay, because we do not have much volume change in our system. Okay, and delta H mixing you can calculate from this equation. Okay. Now, in order to calculate Flory-Huggins interaction parameter, we consider two additional systems. So in first system, we consider 24 hydrotopes, so 24 hydrotope SCS molecule and one drag molecule, and in 24:1 ratio. In second mixer, we use 24 hydrotope molecules in two drag molecules. Okay, imperial is 1 ratio. And at first, what we did vacuum simulation for 12 nanoseconds at 298 Kelvin room temperature, because to get the prior compaction. Okay, if you have water, it will not be. You cannot compact them properly. Okay, so for that we use vacuum simulation for 12 nanoseconds, and then we added water molecules, and then we did the normal simulation for 20 nanoseconds. And now you can see, okay, so in figure A, B, C, okay, these are all for vacuum simulation for three different time intervals. Okay. Now you can see that, okay, so pink color, as I said, pink colors are drag molecules. You can see that pink drag molecules are outside of this. The SCS molecules they aggregate, and the drag molecules they are outside of this thing. Okay, outside of that aggregate. Okay. So and then if we introduce water, now we concentrate on figure D, E, and F. As we introduce water, now you can see that okay, drag molecules go inside. Okay, so you have the hydrophobic or you have the hydrotrope aggregates here outside, and inside you have the drag molecules. Okay, so drag molecules they get encapsulated by these hydrotrope molecules, and then for these hydrotrope molecules, these hydrophobic part are pointing inside. Okay, and hydrophilic parts are this red color. You can see the red colors. They are pointing outside. They are making favorable contact with the outside water. Okay, and the hydrophobic tail of the hydrotropes they interact favorably with the drag molecules here. Okay, And then we calculated delta H mixing as well as Flory-Higgins interaction parameter for these two systems. And you can see that, okay, the Flory-Higgins interaction is much negative, very large negative values. Okay, so they say, okay, now they are miscible, okay, and they are soluble. Okay, so in brief, what it does actually, okay, so in brief, hydroatom molecules, they first aggregate, okay, and then they encapsulate the drug molecules inside their aggregates, and their hydrophilic parts, they are pointing towards outside towards water molecules to make favorable contact with the water molecules. So in conclusion, the self-aggregation of HCS molecules through its hydrophobic tail above minimum hydrogen concentration causes the formation of micellar-like frameworks. Interestingly, though, the drug molecule glycofulvin possesses both polar and non-polar group, it prefers to get encapsulated inside the hydrophobic core of HCS aggregates. So although the glycofulvin it has a small polar part, but it still prefers to go inside the hydrophobic core region of HCS aggregate. And the total decomposition of the decomposition of total HCS drug interaction energy into van der Waals and electrostatic components suggests that the former plays a major role in this interaction. And the calculated Flory-Higgins interaction parameter values gives a strong indication of the mixing ability of hydrotop HCS and glycofulvin drug molecules. And this is okay. This is not a present group. Present group. This Sijita has left. Again, he has gone to Germany for postdoc, and few new members have joined. But it's quite little old picture. And this is our campus. And thank you for your attention.
Thank you, Professor Pal, for such a nice and informative talk. Now session is open for the questions. Your voice is very low. You please ask again. Okay. In the meantime, I will read what somebody is writing in the chat. They are saying in molecular dynamic simulation, you apply and solve Newton's equation of motion for a set of atoms to obtain real dynamic information. Why can't this same method be used to determine atomistic mechanism by which chemical process occur?
Okay, chemical process usually in classical molecular dynamics simulation you cannot really deal with chemical processes because chemical processes involve bond breaking and bond formation, right? But here in classical molecular dynamics simulation, bond breaking and bond formation is not possible. Is not actually possible. So you cannot really do.
Okay. Any other question?
Hello, sir.
Yes.
Anvesh Pandey.
Yes.
Sir, I have one very basic question. Sir, I have seen manuscripts. They have done simulation for like 10 nanoseconds, 50 nanoseconds, sometimes 100, and even there are some manuscripts they have done simulation for more than around 1000 nanoseconds. So, is there anything like what is the minimum simulation time that one has to do?
Yeah, that's what I mentioned actually. Yeah, it's very important question actually. That's what I mentioned. Okay, it depends on the properties you are looking at. Okay, suppose you want to calculate density. Okay, yes. So you need very short simulation run. Suppose you want to see the conformational changes of a protein molecule. You need to run very big simulation, very large simulation. Okay, it depends on the type of the system you are considering.
Yes, but
okay,
sir,
suppose it comes through the experiment, comes through your experience as well as from literature survey. Okay.
Yes, sir. So suppose we did a 10 nanosecond or 20 nanosecond simulation and the system has already converged, like there is no the deviation in the RMSD or RMSF is already converged within the required range. Right. So should we do more longer simulation so as to make sure if we...
So my advisable is suppose if you are considering suppose 3 simulations or 5 simulations, you run at least one simulation longer time steps. So for that, suppose in 20 nanosecond, okay, the conformation or RMSD does not change, suppose. In most of the cases also, we also see in up to 100 nanosecond, there is no change in the RMSD values. Okay.
Yes, sir. Yes, sir.
So it fluctuates around 2 angstrom or 1.5 angstroms. Okay.
Yeah, that is acceptable range.
After that, okay, it starts breaking. Suppose the protein starts breaking, and RMSD changes.
Yeah.
So that what 10 or 20 nanosecond does not guarantee you that okay, you have reached equilibrium.
Yes, sir. Okay, sir. Yes, sir. Yes, sir.
Got it?
Yes, sir. Thank you, sir. Thank you, sir. Thank
Next
you,
person is asking in chat how to do loop modeling.
I did not understand what is loop modeling.
So I also did not. So maybe we will skip this question.
Can you elaborate what is loop modeling so that maybe the term is new to
Chat
me?
box in the chat box. So
Yeah, I have seen that one, but only written how to do loop modeling.
yeah.
So what is loop modeling? Could you please just explain a little bit so that I can entitle some light or like shed some light on that?
Sir, probably he is asking in the protein region how to model the loops which are missed in the X-ray crystallography.
No, usually loop modeling, whatever you do, actually, if you consider a protein, okay, or if you consider a DNA, you have the loop region there, right?
Yeah.
Okay, and then what you do, you have to choose a force field, and that force field will take care of all the atomic sites of your system. Okay, so you are not it's not advisable to change the force field parameters of that protein on your own. Okay, because those are parameterized by calculating different thermodynamic quantities and matching with the experimental values. Okay. Suppose again I am telling you, suppose you use mostly, suppose you are using a biomolecule, you are using amber. I will suggest you to use solvent as tip 3P water, not SPCE or SPC, because the parameters or the force field parameters developed for a particular protein in amber or charm, for those parameters, the parameterized were done using tip 3P water. Okay, so if you use SPC water or SPCE water, okay, so you might get different result. Okay.
The next question is Ramachandran plot is sufficient for result analysis of modeling of protein?
Oh, okay. It's one of the tool, I would say. Okay, one of the tool, but you need to consider several things there. Okay.
So any other question? If anyone want to ask? If not, then let's thank Professor Sandeep Pal for his very nice lecture and interactions. Thank you.
Thank you, Hemant. Thank you, everyone. Bye.
So now we will be having a lunch break, and immediately after the lunch break, Professor Etienne Derard will be giving a talk and hands-on demo on QMMM. Yes, hello.
Hello, sir.
Yeah.
Sir, I have one question.
Professor Sandeep Pal already left, so you can send me a message. Sandeep, you are there? No, he left. You should have asked him in the when it was asked that you asked the question. No, why you are asking now? You can send this by email, and we will get back to you. Okay? Because I don't want to answer question on his behalf because it is based on his presentation. So you send him an email. So after the lunch break, we will be having the talk by Professor Etienne Derard, and he will be explaining QMMM calculations, and then it will be hands-on. So see you at 2:15. Till then, we are going for lunch. Thank you. Hi, Etienne.
Hello, Eman, how are you?
Fine, fine. How are you?
Fine, you see, I'm ready for the talk.
Very good. Thank you so much. So everything is working. Your video is very nice. So beautiful background. And your voice is
Yeah,
very
the background because I'm at home, so I think it's better. Is there anything special I should know about the meeting or what we are doing?
So this is a talk followed by hands-on, and you can just continue after a 2-3 minute break if you want, or 5 minutes break if you want after your talk. The same link and same everything will be available. Only thing is that whoever is inside the meeting, they will be able to do whatever you will instruct them to do. Others who are watching online or on YouTube, they will just listen to you.
Okay. No. I have not planned anything on a computer simply because it's very hard to have access to all the software needed. So it's simply
on YouTube, and then you can. This recording will be available on YouTube, and in future also, all these participants, if they want, they can see this video and continue whenever it is required. That is absolutely fine. We have another 13 minutes because this is lunch break. So approximately 13 minutes are there. But whoever are inside, they can listen to our conversation, and they are seeing you. So, but
I'm in touch with on WhatsApp with Simon. Just I'm here online, but perhaps send me a message when I need to start everything. Perhaps I will try something because the slider on PowerPoint, and I will put how do I?
In the presentation, there is a right-hand side. If you go extreme right, there is a tab share and mute all. So you can click on share. If you go to completely right side.
You know, I'm never using a fichier right side. Up.
Suman,
you can find them.
Yeah, Suman is coming and he will guide you where to click. Yes, he will do it from there.
Sir, in your screen on my.
Yeah. Okay. I need to.
Yes, it is coming.
Yeah, your presentation is visible now.
Your presentation is visible.
You can see
presentation mode so that we can see the next slide if it is going or not.
1 second.
Presentation mode.
Does it work?
Not yet, but it should work now. Not yet. You are using a Mac or a Windows?
Yes, I'm using a Mac. 1 second.
For sharing, you open that the slide mode.
1 second, I'm going back to. Whoop, up, up, up. Okay, what do I? Okay. Now, do you see something?
In the slide mode.
Okay. I have a bad network. Do you hear me? Do you see me at the moment?
Yeah. We can see you. We can see your slide, but not in the presentation mode. What you should do is you close your presentation, open it, keep it in the slide mode, and then come to Teams and then share your screen.
Okay, so, I will
close this, open the presentation, keep it in the slide mode, then you come to Teams.
Okay.
Now you share your presentation.
Okay. And now?
Yeah, absolutely fine now.
Okay.
Go to the next slide so that we make, yeah, everything is working fine now.
Okay.
Very good, thank you.
You're welcome, but sorry, this is the first time I'm using Teams for teaching. We are using Zoom here. So,
okay. So in India, Microsoft Teams is very popular.
Mm-hmm. So
this Office 365 license, which includes Microsoft Word and many other apps and all. But anyway, so how is everything, Etienne? Long time.
It's complicated. We during last spring we were at home for 2 months. After that, in summer it was better. We started to teach a little bit with the students in September, October, and since October we are teaching online only, with no practical teaching. So for chemistry, it's really terrible. That's complicated.
Your picture so that you can run some calculations whenever it is required.
Yeah, no problem. I can go to the lab when I want. In fact, the rule in the lab that we will need to have only 50% of the people, but so I can. I'm going whenever I want. For example, I will go this afternoon after this talk. Yeah, that's.
And your students?
Sorry.
Your students, your scholars, PhD students, and whoever is there.
That's okay. Basically, they can work from home. The same story. They are coming to the lab from time to time. We discuss mainly online. But we get in touch sometimes also by virtual meeting. Still okay for the moment, but I'm fearing that we will see the consequences in 6 months or so because it's completely, we are losing, we are not in touch with some undergrads which are really lost in the middle of nowhere. So this is the main problem, the new one, let's say.
Good. So I hope that this year will be better for all of us.
Oh, yeah, but they are starting to vaccinate everyone here. In fact, today they are starting all the people above 75. In 2 weeks, it will be above 60. And in February, they are planning to vaccinate all the teachers. So we'll see what's going on. It's a little bit slow. But
So we have 5 minutes to start. Still this lunch break is going on. Earlier I thought that I will invite all of you to Guwahati and then we will have this workshop, but then it was impossible. But you are muted. So you are muted, Etienne.
for activity.
Good. So Good afternoon, everyone. Welcome to the post-lunch session of Day 4 of MMTT 2021 International Workshop. In this session, I am very pleased to introduce my colleague and friend, Professor Etienne Derat from Sorbonne University, France. He is an associate professor of theoretical and computational chemistry, and his research focus on understanding mechanistic pathways in organometallic and/or radical chemistry. He has done a lot of work on cytochrome P450 by hybrid QMMM calculations, which you have heard a few talks. During the first part of his career, he worked on the catalytic mechanism of catalytic reactions and demonstrating its selectivity, and several other research topics. He was involved in several CH activation and so many other research areas. He has published very high impact international journal papers, including 9 Angevity Chemie papers and 13 JACS papers. So this is all very, very difficult task which he has done. He has more than 75 publications and they are cited more than 3,000 times. He has given so many lectures and so many presentations around the globe. And he got several research grants, awards, and several other things. So let's welcome Professor Etienne Derat. Thank you. Now you can start the talk.
Thank you, Eman. Thank you for the invitation and welcome everyone. So let's start. First of all, I will summarize quickly what we will discuss this morning. What I plan is the following. First, something like 40 slides on hybrid method devoted to QM, QM prime, and QMMM. And after that, a few examples, in fact, 3 selected examples. And in between these two parts, there will be a few slides on summarizing the key point you need to know before starting QMMM calculation. I'm not a big expert of the Microsoft Teams software, so don't hesitate to interrupt me by switching out your mic or some stuff like that. Anyway, we have time, so it should be okay. And I see, from what I see, we are around, I don't know. I don't find the information how many people there is at the moment, so we will see. Okay. First, this slide is summarizing a general problem when we are doing simulation. We can start. Do you see my pointer on the blue square?
Yes.
Yeah, okay, perfect. So you can perform calculation, as you know, by doing quantum mechanics calculation, but in that case, the time on the length of the system will be around 10⁻⁹ meter, and you can perform, or perhaps sometime nowadays now, because this picture is quite old, a few nanoseconds of calculation. On the other side, in red, when you are interested on macroscopic system, the size of the system will be an order of 1 millimeter, and the time will be in seconds. So we need to bridge these two parts, and there is several solutions to do that. One of them being to use coarse grain method. I will not discuss that. But one of the main methods that we can use is molecular mechanics, or for some system, semi-empirical quantum mechanics, in which the most time-consuming calculation are replaced by pre-calculated stuff. But the question, in order to reach a bigger system, is that can we merge molecular dynamics with quantum mechanics? The answer is obviously yes, as you know, and we will see how now. So the main idea for that is to split the system in different parts. The key part of the system will be described by quantum mechanics. The surrounding part will be described by a simpler method in terms of time of calculation, usually a force field, but it can be also a lower quality quantum mechanical methods, so semi-empirical, or for example, the DFTB or this kind of stuff. And around that, most of the time now, we add a solvation scheme in order to take into account all the environmental effects. So I don't know if you see it very well because it's red on red. It's not very clever from mine. But the big question is how do you manage the border between the QM and the MM part? This is the most important question in this area of research. Okay, so outlook of this presentation. I will start with some historical aspect. I like to start with that because science is not evolving by continuously, but step by step, and I will show you how in this field. After that, we will move to the two big methods in QMMM, either additive or subtractive. After that, we'll discuss the methods to manage the layers. We will see a few examples on practical aspects at the end. So, historically, everything is starting a very, very long time ago, in fact. It's starting in the 50s and 60s, because already at that time, people were knowing that there will be a problem of computation, even with the computer of that time. They were knowing that it will be impossible to treat a full system quantum mechanically. So, for example, the first one to do a coarse grain calculation was referenced in 1925. For all this work, they gained the Nobel Prize, as you know, and you have to remember that it was not direct. First of all, calculation, for example, in 1968, they first published a force field for cycloalkane, and after that they moved to protein, but it was not a direct story. And it's important to understand why they were starting on alkane, because they were also just after that working on alkyne. And in fact, the first QMMM calculation was not done on a protein but on alkyne, in order to separate sigma bond from pi bond. The sigma bond were described by molecular force field, and the pi bond by a quantum semi-empirical method. So, after this first calculation, so in 1972, they moved to do what we call now QMMM, so most of the time, so studying a protein. It was in 1976 in a journal of molecular biology. So the idea of mixing force field and quantum mechanics is clearly not new. It has now something like 50 years old. And the main equation is this one. The potential of the system can be split between a potential on classical system, a potential on the quantum system, so the area treated by quantum mechanics calculation, and a potential describing the interaction between the two parts. The choice of the first system, lysosym, it's not by chance, and I will take 1 minute to explain why they chose that. This is a key system in enzymology, a key system targeted by antibiotics. You know, this is a very critical problem even in our days. Since 1 year we have a problem with the virus, but as you can see, we have now a vaccine. It's okay, no problem. It's nearly solved, let's say, after 1 year of amazing science. But now we still are facing the problem of antibiotics. And even 50 years ago, it was already anticipated because there was already some resistance in the time. And the key reaction when you are dealing with antibiotics is breaking the CO bond on a sugar. And Warshel and Lewitt were discussing that. And by doing these calculations, they have been showing that the main result is that the enzyme is controlling this reaction by electrostatic interaction and not only by steric interaction. You have to remember that at that time, the main idea behind the way enzymes were working was induced fit. So the substrate is going into the pocket and you induce a fit, and because of the steric interaction in the pocket, there is a reaction. they move this picture towards electrostatic interaction by doing this calculation. Okay, so it was in 1976, and for a few years nothing was done in this field. When I say nothing was done, it's not entirely true. In fact, everybody realized at that time that the main problem was not the force field part. It was understood that to improve the force field, it's just a matter of time, but not of thinking. You can improve a force field simply by adding more terms and adding qualitatively better empirical data. But the main problem was contour mechanics. So basically, during the beginning of the 80s, everybody was working on R3 fork, on MP2, MP3, MP4 couplet cluster in order to improve the algorithm on the computer. And when it was done, at the turning point of the 90s, suddenly everybody moved back to QMMF because at that time, contour mechanics algorithms were pretty good. It was possible to do calculation, and thus it was possible to treat suddenly more interesting system. And thus, what was done at this turning point was to incorporate a semi-empirical method within a force field molecular dynamics code. It was done by Martin Field, essentially, who was at that time a postdoc in the group of Martin Karplus. And at that time already, their main problem was not at all to implement that in a molecular dynamics code, but they realized that the main problem was to manage the boundary between the QM and the MM part. So suddenly there was plenty of work done on the treatment of this problem. So the boundary between the QM and the MM part. So we are in the 90s, and you have to realize that the DFT implementation in the main program used by all the people who were doing QM calculation, DFT was only implemented in Gaussian in 1992. Before that, it was only possible to do R3 fork MP2 and stuff like that. Nearly the same time, clusters were introduced, so 2 years later, same area of time, the Intel Pentium was introduced, a cheaper processor. And because of all that, we moved from computer able to calculate gigaflops to teraflops in a dozen of years. Of course, in our day, we are more above petaflops. What does it mean? In fact, to move from gigaflop to teraflops for us, computational chemists, it means that the main change is that we move from 5 atoms to 50 atoms. Why is it so important? This point, and why is it related to QMMF? Because 50 atoms, suddenly this is the size of an active site in an enzyme or for a catalyst. Of course, with some cuts, for example, you replace the tertiobutyl group by a methyl and stuff like that. But you can have a model, a good model, because you can always model a system with 5 atoms, but in that case, you cut a lot, and it's a very small system, and you will lose a lot of information. But basically, with 50 atoms, you can do a lot of stuff. You can treat all the catalysts on most of the enzymes. But, this is a big but, there is still an environmental problem, meaning that you don't take into account everything on the ligands. As I said, for example, you skip the tertiobutyl by replacing them with methyl, and you don't describe the 20 molecules of solvent around the catalytic site. And thus, because you have these 50 atoms available for your calculation, you go back to the usual solution in that case, which is to divide and conquer. as I said previously, the easiest solution is to, in this divide and conquer scheme, is to do the calculation, is to use an additive model. So the energy of the total system will be the energy of the QM part plus the energy of the MM part plus the energy at the boundary. Of course, this boundary term, there is many ways to calculate it, so there is many flavor of the scheme. But there is another idea which was introduced by Keiji Murukuma, and which is known now as Onium, and I will now discuss for a few minutes about this subtractive model. Okay. So the idea is that we want this point in black label 4. So for the real system at a high level of calculation, we want the energy in order to compute the potential energy surface and gain access to TS and so on and so on. So this is what we want, point 4. But we are only able to calculate the real system at a low level of calculation. But we are also able to calculate the model system at the low level. Of course, it's very easy. It will take 1 second of calculation. But the model system, we can calculate it also at the high level of calculation. So the difference in energy between the point number 2 and the point number 1, we will say that we add it to point number 3 here. And that's why it's called a subtractive method. It's because we add the difference between point 2 and 1 to point 3. But we can do the same also with a 3 level. In that case, the idea is that we can calculate 6 easily, so the real system at low level. We have an intermediate model where we can calculate the energy at the low level and also at the medium level of calculation. So we add the difference 5 minus 3. And the small model of the system is characterized by a medium calculation and a high level of calculation, and we add again the difference between point 4 and 2. So the total energy of the system will be 6 plus the difference between 5 and 3 plus the difference between 4 and 2. So this is a 3-layer Onium idea. There is different flavor of Onium, and that's why I entitled my talk QM prime or QMMM, because in fact, the Onium idea started not for QMMM calculation, but for QM high-level calculation. In other words, by IMOMO. The main idea was that you have, let's say, a system of 100 atoms, and you can easily calculate that at the R3-Fock level. And the key part of this 100 atoms, where the part where you are breaking and creating bonds, you need a couplet cluster. So as you know, for doing a couplet cluster with a huge basis set, you can do that up to 10 atoms. And thus, you will use Onium in order to incorporate surrounding effect on the couplet cluster calculation. So in that case, it's called IMOMO. It's described in some publication back in the middle of the 19th. And basically, the equation is the same where whatever flavor you are using, either IMOMO, so QM prime, or QMMM calculation. It's always the same idea, adding a difference to an energy that you can easily calculate. Okay. So here are the equation for the Onium 3 levels. I talked already a little bit about that. In fact, it's relatively interesting to use Onium 3 level in the following two schemes, either when you have 2 layers of quantum calculation plus 1 layer of molecular mechanics, or 3 layers of quantum calculation, for example, couplet cluster, MP2, R3-Fock, or couplet cluster, R3-Fock on semi-empirical calculation on layer 3. Okay, there is a big question about onium. What is the quality of this calculation? Okay, I will go back a little bit here. First, what we assume in fact that in order to obtain this target, that this value is equal to this value and this value is equal to this value. In other words, that here we have a perfect square. So basically, the group of Keiji Murokuma introduced the S-value test, which is saying basically that the onium calculation that you did, you will compute one time the target energy here and calculate the difference between the target energy and the real value that you did one time. So you will think, oh, but it's impossible to do that for enzyme. Indeed, it's totally impossible. In fact, this idea of onium, you have to remember that it was created in order to gain access to a catalytic system at the beginning, so doing QM prime calculation. But basically, when you have a system of, let's say, 300, 400 atoms, for example, you can try to do an S-value test one time. It will be a very long calculation, but you will know what will be the error of your onium calculation. As I said, the main idea of doing onium calculation was at the beginning to gain access to high-level complete cluster calculation, in fact. And because of that, there has been the group of Murokuma introduce what we call G2 extrapolation. G2 is a way to obtain high-quality thermodynamics data, which is very useful for checking experimental data related to thermodynamics. And the idea here is that not only you do an extrapolation on the low-level, high-level quantum mechanical calculation on the size of the system, but you add also an extrapolation on the basis set, taking into account the same idea of onium, meaning that this difference between the small and the large basis set on the difference between the large and the small basis set on the real system is the same on the large and the small basis set on the low level system. So basically, this difference is the same here, and you can do an extrapolation. A few examples of this idea here for a Diels-Alder reaction. Experimentally, we know the value, for example, for dimerization of beta-diene. It's 23.7 plus minus 0.2. If you do the calculation at the R34 level, the barrier is 48, totally wrong, you can't forget it. If you do the calculation at the MP2 level, the barrier is 18 kcal, way too low by 5 kcal. If you do a composite calculation, so QM prime, coupling couplet cluster on MP2, you obtain a value of 27.2, so above by a few kcal. But this calculation was done with a double zeta basis set at the couplet cluster level. So there is a problem with the couplet cluster calculation. So in order to improve it, you use the idea of extrapolation. And in that case, you obtain a value of 23.5, so exactly the experimental value within the range of error of experiments. And this is nearly all the same for all what they calculate. Sometimes it's even interesting, as you can see, it's a little bit lower, so we can even guess that there could be a problem with the experiment sometimes. It's an old article and I didn't update, but Jan Martin shows that there is sometimes error in the experimental data. Okay. So what I described for the past 20 minutes is mainly the subtractive methods in which you add a difference to a reference point. And now let's go back to the additive method, in which the energy is a sum of an MM part and a QM part plus the interaction between the two parts. This interaction between the two parts can be divided in three components mainly. A component which is related to the bonding between the QM and the MM part, a component which is related to the van der Waals interaction between the QM and the MM part, and a component which is related to the electrostatic interaction between the QM and the MM part. Because I don't see you, I will make a small break. Is everyone okay? No problem. I don't see if there is question also. I need to check that. No. Okay. Okay, so treatment of this electrostatic part. There is a few ways to do that. You can first consider that there is no interaction. And it will be called mechanical embedding, meaning that the MM part will be interacting with the QM part only by imposing constraint on the position. You can think it's not interesting at all, because we all know that an atom will have some electrostatic effect on the surrounding part, but it could be very interesting to do mechanical calculation like that. Because if you skip all the electrostatic part, by deduction you will know what is the electrostatic effect. So basically, the mechanical embedding, it's a silly calculation most of the time, except if you do it at the same time with the electrostatic embedding. So if the MM part is polarizing the QM part. Because in that case, by calculating the difference between electrostatic embedding and mechanical embedding, you will know the influence from an electrostatic point of view of the MM part on the QM part. And there is a third way to do the calculation. Of course, the MM part can polarize the QM part, but the QM part can polarize the MM part. In that case, you need to use not a classical force field like a charm, amber, or gromos, but you need to use a polarizable force field. It's much more computationally expensive by a factor of 10, but it can be really interesting because when you are dealing with an active site of an enzyme, of course, there is usually a huge polarization induced by the active site. Oh, since we are talking about mechanical embedding, electrostatic embedding, and so on, I need to give you another point of view about QMMM calculation. When you are doing a QMMM calculation of an enzyme, you are dealing with a system of, let's say, at minimum 10,000 atoms, meaning that there is a lot of potential problem. The main one being the conformation of this huge system. You need to sample a lot. I guess all of you have done a molecular dynamics in your life, and as you know, for big systems there is a lot of potential conformation. I'm sorry, I will make a small break because I don't know if you hear me well, because there is a garbage truck in the street. This is the joy of teaching at home. Oops. We'll wait a few more 30 seconds that the truck is moving. Okay, better. Okay, so the key sentence is here. With a small model, it is generally possible to identify all structural change in the model during a reaction and make certain that these changes are not artifacts of the model. So the point of view of the Swedish school of QMMM is that, okay, it can be interesting to do QMMM calculation because you introduce everything around the active site, but it's also interesting to do a small calculation with what they call a cluster model. So something which is reduced to the smallest calculations that you will do using only QM calculation in order to identify the everything coming from the surrounding part around the active site. There has been a lot of discussion about that.
Hello, sir. Hello, sir.
Yes, go ahead.
Hello, sir.
Yes. Go ahead.
Sir, you are muted, sir. Can you please unmute, sir? Unmute yourself, sir.
Etienne, you are muted. Now it's fine.
It's fine. Since when I was mute?
You are good.
What's going on? Did you hear what I said on this slide?
Yes, sir.
Okay.
You are audible.
Okay, thank you. So mechanical embedding. I said I talked already about that. So it allows to treat steric effect in an efficient manner. It allows you to know what is the impact of steric interaction on your calculation. It allows you to calculate by making difference what is the electrostatic component on your active site. But the main problem with this kind of calculation is that the charges are the same throughout the reaction. It's really problematic because, as you know, when you are doing a chemical reaction, there is a huge modification of some charges on the active site. Second way to do that, electrostatic embedding. In that case, the interaction between the QM and the MM part will be calculated in the following manner. So QG are the MM point charge, QR are the nuclear charges of the QM atom, and of course you calculate the interaction between the molecular mechanics point charge, and you calculate the interaction between the QM atoms and the MM atoms in that case. The main problem is that you need to calculate also the gradient, so the difference of this equation. And because here most of the time you have above 10,000 atoms, so 10,000 point charge, doing the gradient of the point charge is becoming, in fact, the main component of your calculation. To give you an example, if you do a QM calculation with DFT on a classical force field, so let's say charm, on a system of around 150 atoms, the DFT calculation with modern software will take a few minutes, let's say between 3, 4, 5 minutes, but the gradient of the point charge will take around 20 minutes. So 4 more times than the DFT calculation. So you will have 5 minutes DFT, 20 minutes calculation of the point charge gradient, and 1 second of molecular mechanics calculation. Total time for one step of the optimization, 25 minutes, which is quite long for something which is very basic mathematics. But anyway, there is no choice. Okay, let's compare the molecular, the mechanical embedding and the electrostatic embedding, the advantage and disadvantage. Molecular mechanical embedding, the treatment from a computational point of view is very simple. Electrostatic embedding, it's you need to include one electron Hamiltonian in the QM part, so it will modify your Hamiltonian. On the other hand, you have to remember also that the MM parameters are not modified in that case, it's interesting because the electronic structure of the QMC atoms will be adjusted to the position of the MM atoms. Disadvantage of the two schemes, so the molecular mechanical embedding. Disadvantage for the electrostatic embedding, more computational effort, and you need to construct, before doing the calculation, a very good representation of the point charge surrounding the QM atoms. This is a very important disadvantage. If you think a little bit, for classical force fields, so charm and amber, were designed to represent classical proteins, so beta barrels, alpha sheet, and stuff like that. But they were never designed to represent active site reacting, meaning that sometimes it can be important to readjust the point charge of the residue surrounding the active site in order to take into account the polarization induced by the active site. Of course, this part is very time consuming if you need to recalculate all the point charge of a force field, because the calculation in itself is not very long, but you need after that to check it, and it can take some time. Another disadvantage of the mechanical embedding is, of course, that you ignore everything, all the perturbation coming from the atoms surrounding the active site, so you lose a lot of information. So in order to solve all this problem, there is an obvious idea, which is to use polarizable embedding. It's straightforward to get the idea, but after that, it's another story. Because if you need to recalculate in an iterative manner the charges on the MM part, it must be done in an iterative scheme or by using Lagrangian interaction, meaning that basically, to simplify the idea, what you need to do is a kind of R34 calculation at each step of your QMMM calculation on the MM part. So of course it will be quite time consuming, and thus you cannot do that all the time, and that's why even in our days, after more than 30 years of work on QMMM calculation, most of the calculation are done only with electrostatic embedding, because a factor of 10 in time of calculation, as I said a few minutes ago, one step of calculation is around half an hour. So if you multiply by 10, it's a huge time of calculation, and you cannot afford it for most of the problem. Okay. So I sum up the two first parts. Additive versus subtractive, mechanical embedding, electrostatic embedding, polarizable embedding. But at the moment, we never discuss how we cut between the QM part and the MM part. So let's imagine a system like that. You have a QM part constituted by these atoms, and the MM part is constituted by these atoms. In fact, in that case, you need to break between atom Q1 and atom M1. So there is several problems in that case, because I will introduce something. Of course, you need to take into account the valence of the atoms. So basically, one of the solutions that you can have is to introduce a link atom. So Q1 will be linked now by covalent bond to a link atom. On the molecular mechanics atom M1 will be linked also to a link atom. There is a problem with it. If you imagine that this link atom is an hydrogen, on this Q1 M1 bond is a standard CC bond, so 1.5 angstrom. The link atom here will be at 1 angstrom of Q1, thus at 0.5 angstrom of M1. And if you think a little bit, you will have a point charge associated to the hydrogen atoms, to the hydrogen atom L, here very close to the M1 atoms. Because these 2 point charge will interact too strongly, we need to do something. And the usual idea is to replace the charge on the M1 atom by 0, and to redistribute the original charge of M1 on M2, M2 on M2 here. This is why it's said Q divided by 3. So the original charge on M1 is divided in 3 and redistributed on the 3 M2 here. Okay, so this is a first idea. But there is also other idea that we can do. So I treat the first one, the link atom scheme. We introduce an additional atomic center, usually a hydrogen atom, but it can be something else. It can be a chloride, a fluoride if you want, or even a virtual atom that never exists with a special basis set fitted in order to represent the electronic structure of your stem. There is other idea. You can use a special boundary atom, so in order to represent in a better manner the electronic character of the MM moiety and the QM character of the QM. And there is a third idea which has been deeply explored by many groups around 20 years ago, which is to introduce localized orbital. So, as you know, we can reconstruct canonical orbital in order to make hybrid orbitals. For example, you have a carbon sp3, and you know exactly what is the kind of orbitals that you have around a carbon sp3. You localize, you freeze one of them, and instead of introducing a link atom, you freeze the electron in these hybrid orbitals. This is a very nice idea because you gain immediately all the electrostatic components, all the quantum mechanical components. The main problem is that you need to pre-compute this localized orbital. And I will explain why this is problematic, because basically when you do it, it takes 1 year to make this cut. Because you have to check plenty of stuff, you have to check this is stable in many calculations. So, on one hand, you have the link atom, you cut, you introduce an hydrogen, and you run the calculation, it's a 10-minute works. And on the other other hand, you have the localized orbital with high-quality quantum mechanical calculation, but it takes 1 year. So you know exactly what everybody is doing now. Everybody is using link atom. I talk a little bit about the next slide. So because you need to remove the charge at the interface in order to reduce the polarization. There is several ideas to do that. I talk about shifting of the point charge, but there is another idea, simply deleting everything and not replacing by anything. Deleting the one electron integrals in the QM part in order to reduce the overpolarization, or blurring the point charge by representing them by a Gaussian distribution. Also, this last idea is quite time-consuming because you need to ascertain that the Gaussian distribution is correct, but this is the best solution from a qualitative point of view. Frozen orbitals. There is two ways to freeze the orbitals. Either you can freeze the orbitals on the QM part or on the MM part. If you freeze the orbitals on the QM part, it means that Q1 cannot be taken into account in the reactive part of your QM part. Therefore, most of the people still doing frozen orbital calculation for QMMM are using the freezing on the MM part. This is much more stable. On the other hand, qualitatively, it's better to freeze on the QM part. But as I said, this is very, very time-consuming. There is another idea in order to manage the boundary between the QM and the MM part. I guess all of you did a QM calculation, and sometimes we use not only basis set, but we use effective potential for representing the core electrons. So let's translate this idea to the boundary between QM and MM. So if we use, if you want to calculate a big system like that, with the core atoms where the reaction is happening, surrounded by many groups around, but these groups are all the same. For example, this is a water molecule. You will divide the system in two parts. The core system will still be described by QM, but all this water molecule will be described now by an effective fragment potential. So potential representing the orbitals of the fragment, but everything is pre-calculated. This is a very nice idea because you maintain the quality of the quantum calculation, but there is a penalty, which is that you need to pre-calculate the potential before starting the calculation. This potential usually is divided in three parts: Coulombic potential, induction, and repulsion, with the classical equation for representing these three parts. As usual, it takes a lot of time to generate this potential. When you have done it for one type of fragment, if you slightly modify the fragment, you have to redo it again. So this is very interesting when you are always using the same fragment, but otherwise this is very, very time-consuming. If you think a little bit, in fact, this idea of effective fragment potential is the same than doing a semi-empirical calculation because in fact, you replace all the integrals needed to calculate this in a fully quantum mechanical manner by one electron integrals on all the fragments. So basically, it's like pre-computing your fragment as a semi-empirical level, which is totally dedicated to your system. Okay. So now we have been discussing nearly everything regarding the QMMM calculation, the classical QMMM calculation. There is a last type of QMMM calculation, which is EVB, empirical valence bond. The idea in that case is that the calculations that you will do will be 100% molecular mechanics, except for one stuff. The core of the system, what is important, will be pre-calculated, represented by an empirical Hamiltonian. Meaning, for example, that you want to create the bond between this oxygen and this carbon, and you will pre-calculate the component in the force field associated to the formation of this bond. How you will calculate that? By quantum mechanical calculation, and you will introduce the result of this calculation into the force field. So this is very interesting because you introduce high quality quantum mechanical data into the force field. Of course, you have to do that all the time when you are dealing with a new system. So at the beginning, this is very time consuming, but after that, you can gain access to the dynamical component of your system. So basically, the reaction is quite simple and can be easily described by the force field. For example, this is a simple SN2 reaction, and you introduce the SN2 parameter into your force field, you will gain access quickly to the dynamical component of your SN2 reaction. Unlike that, for example, you can calculate quite a lot of transition state and see the influence of, for example, mutation around the active site. So this is a good way to calculate the influence of mutation on a specific enzymatic reaction. For example, if you do a standard QM calculation with mutation, it will take you 1 or 2 months in order to obtain the transition state. And if you use EVB, it will take you 2 days. So basically, in that case, meaning in 60 days, you can do something like 30 mutation calculation in the same time that you will do standard QMMM calculation. Okay. If you don't mind, I will do a small break because I'm talking for 45 minutes or so. So it's on.
Okay, let's have a 5 minutes break and then we will continue.
5 minutes break. And I will drink water. I'm coming back.
Sure.
Ça va. Ouais. Okay. Will we start?
Yes, please.
Okay. Okay. Something on the side of QMMM calculation, something more recent than what I discussed before, on which is called adaptative QMMM. So for the moment, we discuss what is the scheme for doing QMMM calculation, how you break between the QM and the MM part. But now imagine that you have a water molecule which is entering the active site during the reaction. Meaning that, for example, you start with 5 water molecules at the beginning, and at the end of the reaction, you have 6 in the active site. How do you deal with that? So the idea is to have an adaptative QMMM scheme in which there is a QM region which will deal with the water molecule entering or leaving the QM area. And it's in fact much more complicated technically because as you know, the QM calculation must be stable, meaning that the total number of electrons in the QM calculation must always be the same. So you need to readjust the QM calculation in order to take that into account. There is a few ideas in the literature regarding that, which is summarized here. First idea is to base the QM managed area on a distance cutoff. So meaning that all the water molecules between 2 radius are taken into account in the QM calculation. There is a problem associated with that, which is the number of water molecules in this area is not stable. Thus, you can imagine that you replace the radius by a total number of water molecules. In that case, the QM part is much more stable. And because, as I said, what is important is the total number of electrons. And if you are doing a QMM calculation with DFT, in that case, what matters is the total density. So in that case, you take into account the total density of the QM system. Another idea, which is technically more challenging, is to use a buffer method, meaning that, for example, a water molecule is at the same time in the MM part in the QM tab and slowly buffered when it's moving. This is the most promising adaptative QMMM, but technically to implement that in a code, it's very, very complicated. As you notice also, I was always talking about water molecule, but we can imagine doing that on many other stuff. At the moment, most of the adaptative QMMM scheme is done, has been tested with water molecule. So basically, it's still there is still a lot of research going in that direction. Okay, let's move to the technical part of this talk. How to build a reactive center for doing a QMM calculation. First point, on the most important one, don't start a project if you don't have experimental data. And when I'm saying experimental data, I'm not saying only a protein structure coming from X-ray or synchrotron. You need a lot of data because you need a lot of reference points in order to check the quality of your calculation. So basically, before starting a project, you start by 1 month reading the literature on that kind of enzyme. After that, if you have a lot of data, you know that it's possible to understand in a better way this specific enzymatic reaction. You move to the technical part. Okay. How do you cut between the QM and the MM part? First rule is there is a specific interaction in the active site. You include this specific interaction in the QM part. For example, I don't know, let's imagine that it's well known experimentally that an arginine is very important for the reaction, and this arginine is doing hydrogen bonding with a lysine. You include this hydrogen bonding. It will cost you a lot because I know an arginine on a lysine is something like 20 atoms. So basically, if you have already something like 70 atoms in your QM part, you add 20 more, you are around 100. The cost of the QM calculation is higher, of course. But basically, it's not a good idea to dismiss this specific hydrogen bond. You always cut unpolarized bond between the QM and the MM part. And if you think a little bit, it's complicated because in an enzyme there is a lot of amide bond which are polarized, so you cannot cut the peptidic bond. You need to find something else to cut. So basically, it means that each time you are nearby the peptidic bond, but you need to include it fully. Meaning that if you have something like 10 residue in your active site, you will increase by 10 amide the size of your QM part. So 4 point, is it similar to QM models? As I said before, when I was talking about the SIGBIND point of view of doing calculation on cluster models, when you have divided your system into QM and MM part, the best idea is probably to start by doing QM only calculation to check if what you did is stable. There is no strange behavior. For that, you use your favorite QM software. You fix some atoms by you optimize it a couple of times. You see if there is no, for example, CH bond going crazy and stuff like that. 5th point, you need to set. Yep, sorry. A question?
Yes, sir.
Yep.
Yeah, please tell your name and then ask the question.
Hello?
Go ahead. Who is that? Go ahead. Or you fix the problem and then you can come back. Let Etienne continue.
Yeah. Okay. Building the reactive center, last point. You need to check that you can afford the computational cost. When I'm saying afford, I don't mean that you are paying. Most of the time, we don't pay for the computational resources. But when I say affording the cost, meaning that, for example, your active system is constituted by a number of QM atoms where you can search for a transition state. For example, you can do a scan, and the scan will take 1 week. And if you need to restart, you will have sufficient time to do the scan again in the future. A selected example on how cutting an active site taken from my research. Long time ago, we were working on Oswaldish peroxidase, and it's an AIM enzyme, okay. And there is a very important experimental data which is saying that the enzyme is existing at two spin state at the same time, doublet and quartet. So basically, from experimental data, we know that there is no energy difference between doublet and quartet. So when you do you divide the first system, which is model 1, here very tiny, something like 50 atoms. And as you can see, at the best quality, the quartet is a little bit more stable. If you introduce step-by-step residue around this model 1, sometimes the doublet is more stable, the quartet more stable. Here at 6, we are nearly close to 0. 7, which is here, you are again, the quartet is more stable. On 8, 9, 10, it's okay. 10 is the biggest one we were able to do at that time. But as you can see, 6 is probably the best one. And what is important to notice is that there is a guanidinium close to it. 7 was not so good, but 8, which is 6 and 7, is perfect. So meaning that the guanidinium is extremely important. So basically, at the end, we choose to use model 8 based on this calculation. So basically, before starting the real subject, I did 10 models, meaning that I have to deal with the technical problem of 10 models before going to the interesting part of the calculation. You cannot skip that, and that's why I was saying that experimental data are needed. Without this key experimental data, probably I would have selected, for example, I don't know, model 5. And model 5 is totally wrong, in fact. So, it's always a discussion between experiments and computation when you are doing QM calculation on enzymatic system. Okay. In order to check if you have understood what I was discussing since 1 hour, a question. Okay, it's a proposition on this enzyme. It's called DMSO reductase. DMSO reductase is in fact reducing DMSO, the solvent, to sulfur ether, which is less problematic inside the cell, and so it's working like that. You start from molybdenum 4, you replace the water molecule by DMSO, then the oxygen is transferred from DMSO to molybdenum, so molybdenum is oxidized from 4 to 6. Then you release the sulfur ether, you obtain this oxomolybdenum 6, which is reduced by an electron and a proton to molybdenum 5, a first time and a second time reduction by an electron plus a proton is coming, and you obtain water and you restart the catalytic cycle. Here you can see on this side, this is the active site, which is very complex. My question to all of you is, tell me where you will cut the system for doing a QMMM calculation.
You can write in the chat box if you are not able to come to the mic.
Yeah, so to talk, Hello? Don't be shy.
So,
Eman? There is no technical problem? You hear me well?
yeah, there is no technical problem. It looks like these people are hesitant to ask the question. Looks like.
Yeah. Let's go.
So we'll wait for 2 more minutes to go there.
Hello.
Yes.
Sir, I just want to ask a simple question.
Yes.
Sir, while doing QMMM, I most of the time I have charge problem in the boundaries. You told us about many type of methods to do that, but can we do it manually by changing some parameters in our input files for
Yes.
the boundaries?
I see what is your ID. Okay.
First, what is your name? The person who has asked the question.
Sir, my name is Anamika Shukla.
Oh.
Okay. Most of the time, it's better. We'll go back in my slide. It's better to use this kind of idea of moving the original charge on the neighbors or at the boundary. But in some cases, it's problematic also. I will give you an example. It's not related to enzyme, but to metal organic framework. So something in which, for example, here, it's a silicium atom and here an oxygen. So you cut here, the silicium is also interacting with, for example, an alkyl group, and the SEO bond is highly polarized. So if you remove the charge on the silicium atom and you add more charge on the oxygens, you will over-polarize the oxygen. So it's not a good idea. In that case, there is a solution, which is to add a dipole moment on M2, putting back the charge in direction of the M1 atom. Is that clear?
Okay. Okay, sir.
Okay. This is called the charge shift method.
Charge shift method. Okay.
Yes, this is the best one. In fact, it has proven to be very, very stable. I don't know a counterexample. In fact, I cannot find in my mind in the literature an example where this method has not been working correctly.
Okay, sir. So this method is can we apply it on Onium 3 also?
Onium 3. I'm sorry, I don't remember if this is implemented in Gaussian. But it's implemented in Games, and since Games has Onium, you can do it in Games. But in Gaussian, I don't remember, sorry.
Okay, okay. Okay, sir. Okay.
Something about technically doing QMMM calculations. There is a lot of QMMM softwares in our days, and most of the people doing QMMM calculation now are not using Gaussian for managing the QMMM part. Meaning that everyone prefers to use a superscript, to manage a QMMM calculation. There is a good reason for that. Basically, by doing calculation like that, it's easier to manage a very efficient software for each part. For example, instead of using Gaussian, you use a very fast quantum mechanical software, and you will use also a very fast molecular mechanics software. For example, you can select a GPU version of your software during the QM part. And like that, you can gain a factor of 10 or 30 quite easily. So, and also by using a script rather than a standalone software, when there is a new version, it's way easier to implement. Okay, let's go back to my question. Where do we cut here? Okay, I think I will help. Here, as we can see, the molybdenum is moving from oxidation state 4 to 6 and going back. All right, meaning that there is during the catalytic cycle 2 times 2 electron exchange, so very huge polarization. So clearly, we need to include the sulfur atom directly linked to the molybdenum atom. Do you agree with that? If you do that, it means that here we include also the double bond here, right? So you start to think, oh, I can cut here and here. But no, there is a problem, because look very carefully. If you cut here, you cut a CC bond. Usually it's not very polarized, but this is not the case here. This carbon here is linked to a nitrogen. On the other side, there is plenty of nitrogen and oxygen. So everything here is very polarized. And in fact, when you look at the structure of this ligand, it's totally flat, meaning that there is conjugation. It's not aromatic, but everything is conjugated by homoallelic interaction. So basically, you cannot cut here and here. You need to take into account everything here. The only bond which is slightly less polarized is this CC bond here, and you can cut here. So basically, this part you can put in the force field. And also, you can all recognize something coming from DNA. So basically, it's very well represented by force field, so no problem. So it's an example showing that you have to be really careful about cutting bonds. You have to check step by step what you are doing. You can try first to cut here and here, you do a calculation, and you will see that the system is not correctly maintained. For example, you will have this part of the ligand which is flat normally, which is becoming like that, like making a strong one. Okay. So technically, how do you do a QM calculation? Let's imagine that you have an idea in mind. You have a lot of experimental data, and especially you have X-ray structure. So you take them from the protein data bank. You select a good one, because usually for an enzyme there is multiple. First stuff to do is that you add missing groups, chain. Sometimes residue are missing, and of course the substrate you are interested in, because most of the time in an X-ray structure the normal substrate is not present because simply the enzyme is reacting on it. You do tiny modification on the active site, not too much, because if you start to do a lot of modification, you need to test them. You add hydrogen on the structure based on pK value. I will talk again about that because this is probably the most critical part, and I will show you example where we found in the group that it was the pK value were wrong. Then you solvate the protein with water or whatever you want, and you do that in an iterative manner, meaning that you solvate, you do an MD, you check if this is stable, you resolvate, you do an MD, you check if this is stable, and so on and so on. And from this, the last MD simulation, you take several snapshots. I will give an example at the next slide by selecting snapshot at 300, 400, 500 picoseconds, sorry, and you will see that there is a huge impact on the calculation. Here again, it's a story between a spin state. By the way, man, you know I took that from Sam Devisa. It's not my work, this is Sam. Anyway. Here, Sam took a snapshot at 100 picosecond, 150, 200, 250, 300. As you can see, in all cases, the ground state is a singlet, an open shell singlet, but a singlet. The triplet is just above, and the quintet is, in nearly all cases, clearly above around 50 kcal per mole, except in one case, the snapshot at 250 picoseconds, where the gap is around 12 kcal per mole. So, when you have this kind of stuff, you have a big problem, because clearly you need to do calculation on snapshot, the first one or the second one, which are nearly similar, and the fifth one is also nearly the same. So meaning you have it seems three family, here 100, 150 on 2 nanoseconds, but clearly 200 and 250 picoseconds are different. So you need to do more extensive calculation on that. And as I said, it could be problematic, because if you need 1 week to localize the transition state, if you have now 3 family, you will need 3 weeks of calculation, plus all the technical problem around that. And a story which was at the beginning only 1 month became quickly a 6 month project. That's the usual business in your field, but you have to take that into account when you start a project. Another problem, which is related to that, how do you know which snapshot is representative on the real activity of the enzyme? Is it this one or this one? So again, you need some experimental data. For example, you need to know that the quintet is never involved. But most of the time, you don't know that. So you have to do the calculation for both snapshots. And you need, at the end of your work, to be in touch with experimental guys in order to know if what you find is fitting well with the experiment. But that's another story. Okay, and now I will stop the teaching part and move to the research part. But before stopping that, I would like to remind you something that you probably know, which is that the best model is not the most complicated one, but the one that explains. Meaning that most of the time, it's better to simplify the system in order to understand it than to overcomplicate the system and you lose insight on it. It's very hard to do that because you never know before doing it what will be overcomplicated or oversimplified. So basically, it's a trial and error process, and it will take time, but there is no way doing it in another manner. Okay.
So now the session is open for some questions, if there are any, before we go.
Timing on the first part of my talk.
So if anyone have any question, they are welcome to ask now.
Hello?
So, hello, sir. Sir, I have a. Sir, hello. My name is Roli and I'm from BBU. Sir, I just want to ask a very basic question regarding how you use transition metal based active sites, as these are the most active sites. Metals are present in most of the enzymes. So how you parameterize metal for doing MD?
Okay. I see. Parameterization of the metallic center for molecular dynamics. I like to freeze. Okay, let me explain. Usually, you know if the behavior of the force that you use is right specific active site that you are working on. So the best way is usually to start by freezing everything. On after you check carefully that you are.
Is it audible?
But there is meaning. For example, if you are dealing with zinc, the charm force field is quite good now. There is even very good quality parameter for zinc. If you are dealing with iron, that's totally another story, of course, because in that case, depending of the structure of the enzyme around and how it's moving, the spin state of the iron can shift from, for example, doublet to quartet, and it will influence the geometry, and no force field can take that into account. So basically, the best advice in that area is be very careful. Do that step by step.
Okay, sir. Thank you, sir.
Any other? Yeah,
Hello.
please introduce yourself and then ask.
Yeah. Hello. Myself, I am Suyash Pant from Naipur, Kolkata. Sir, can QMMM can be implemented with molecular dynamics? To
Yes,
understand.
of course. Yes, of course. There is in fact no problem. Everything I discuss, we discuss just how to calculate the energy. You can do either geometric optimization or molecular dynamics. Usually, this part is managed by other part of the program, and that's why I was saying it's very interesting to use the script for managing the QMM calculation. It's because the script is giving the coordinates to another program which calculates the energy. So basically, for example, if I take, imagine that you want to do a Car-Parinello molecular dynamics, you send the coordinates to the Car-Parinello software, which is dealing with all the technical calculations, sending back to the QMMM part of the program, and so on in an iterative manner. After that, there is a big question, which is, for example, we can imagine that the time step of the MM and the QM part is not the same. Because in the QM part, you are dealing with electrons. So as you know, electrons are moving at the femtosecond, lower than the femtosecond time step. On the other round, for heavy atoms, you can go up to 5 femtoseconds. So there is tricks here in this area in order to manage the dynamics between the MM and the QM part. But it's usually managed by the molecular dynamics software.
Thank you, sir.
You're welcome.
Any other question? Okay, then we can continue with this hands-on if Etienne is ready.
Okay, so I talked a little bit about that. I was saying that pK are relatively important, and I will give you a couple of examples because most of the time, what I'm finding is that the key point in exploring the enzymatic mechanism is related to protons and water. So let's talk about an enzyme called nitric oxide synthase. And this enzyme is generating NO dot, which is a neurotransmitter, by oxidizing an arginine into alpha reaction. First, arginine is oxidized to hydroxyarginine, so you introduce NO bond here, and then you cut the CN bond here, and you generate NO dot. This is very important as we are speaking at the moment. Basically, our brain are using that for doing transmission. It's also used for by macrophage to destroy bacteria. So this is relatively important for every process. We know the structure of nitric oxide synthase. It's an M enzyme with iron here. The arginine is here. But there is a third component which is called H4B, which stands for tetrahydrobiopterin. And we know something experimentally very important, which is that there is a single electron on this H4B moiety during the reaction. We know that by EPR spectroscopy. And this is the experimental result on which we will rely to understand the reaction. So the reaction starts by incorporating dioxygen on the iron. And if you want to do an oxidation of the arginine substrate, you need to break the OO bond here. So when we were doing this calculation, we started by exploring this OO bond breaking. So first news is that when you introduce dioxygen on this iron center, the spin state is an open shell singlet. And if you break the OO bond, you end up with with a triplet at the end. But if you check where are the electrons, we notice that there is no single electrons on the on H4B, but everything was on the arginine. So if you do the calculation without proton, the calculation doesn't fit the experimental results. And we are sure of that also because the thermodynamics value are showing that this is an highly endothermic reaction by something like 22 kcal per mole. So we were surprised because if we look at the literature, everybody is saying that this oxidation is performed by dioxygen, and it's not working if you introduce H2O2. So, surprise. But nevertheless, we were continuing the calculation by adding one proton. And in that case, again, the starting point is open shell singlet, and the product of the reaction is a triplet. But again, there is no single electron on H4B, but still one single electron on the arginine substrate. So again, it seems that there is something wrong with this calculation. And we can also see that by the thermodynamic value. It's again endothermic, but it's reduced. It was 22 without proton. Now with one proton, we go down to 12 kcal. So we were thinking, okay, let's add a second proton. But if we do that, we end up by having H2O2 here in the active site. And experimentally, it doesn't work also. So there is a big problem. We know if we introduce H2O2 on NOS, it doesn't work. But let's look at the results. First of all, the spin state is totally different. Now it's a septuplet. And when you break the OO bond at the end, you obtain a triplet again, but this time there is one electron on H4. So it fits another experimental result, so the most important one. From the thermodynamic point of view, also now the reaction is exothermic, so it's possible to do. So the big question now is not how do you break the OO bond, but why H2O2 is not working and why it's working with dioxygen on protons, on two protons. And when we were doing this calculation, there is a last something to check also, the barrier for the OO bond breaking. It's something like 7 kcal per mole. Okay, it's doable, no problem. But the main problem now is the origin of the proton. Where does it come from? And we ended by doing molecular dynamics, level only. So you put first dioxygen in the active site. You start your molecular dynamics. At the beginning, there is no hydrogen bonding network toward dioxygen, but very quickly we see that the metallic center with dioxygen is polarizing the solvent, the water, and we see that there is suddenly a couple of water molecules in the channel, and after 300 picoseconds, we have a full complete hydrogen bonding networks, which allows protons to be introduced in the active site. So it was a nice story, and we published that, and 6 months later we received an email from an experimental biochemist guy who said, oh, I read your paper, and the story of the hydrogen bonding network is really interesting, so I decided to introduce a methyl group here. You see this is arginine, and you replace this hydrogen by a methyl group. If you do that, you have steric hydrogens in the channel, and the water network is disrupted, and indeed, in that case, nothing is not working anymore experimentally. So we did the calculation, we get the experimental proof after. But as you can see, this is the first story where and when dealing with proton was key to understand the mechanism. And you have to remember that we have at the beginning, in most cases, no idea where are the protons in an enzyme. So basically, we need to do that step by step, carefully to understand what's going on. I will skip this part. Just to go back to the conclusion of this research. Clearly, it's possible to understand a catalytic, enzymatic catalytic cycle. It's not so easy, but we can check every step. It will help you to identify all the intermediates, and by identifying this intermediate, you can go back to the experimental level, saying that you have that, you have that, you have that. And the experimental researcher can check your hypothesis coming from calculation. And sometimes you have an experimental verification of your computational hypothesis. In that case, this is very nice. Okay, I talked already a little bit about horseradish peroxidase in order to introduce how to cut the QM part. But now I will show you a full story about horseradish peroxidase. So like nitric oxide synthase, HRP is a name enzyme with iron here in green in the center. It's an important enzyme involved in many processes. Basically, the main process done by this peroxidase is to repair the cell wall in many plants. For example, when a tree has a problem in some cells, this enzyme is repairing the double lipid layer by inserting a certain type of compound. By the way, this is what is giving the taste to radish, hence the name of radish peroxidase. This is also a very, very important enzyme because it's used in every ELISA test, so antibody antigen test, because there is a color modification due to the oxidation state of the iron center. We know this enzyme for a very long time because it's in huge concentration in radish, and because of that it was identified not as an enzyme, but a guy at the beginning of the 19th century was able to separate what was giving the color to radish, and thus we know how to deal with this enzyme, but of course the structure is more recent. Let's talk a little bit about techniques, how this calculation were done. It was done with ChemShell, which is not a program, a computer program, but a script written in TCL/TK, which interface the QM program on the MM program. For the QM part, we can use several ones. On the same for MM part, we can use Charm, Grobos, Gulp. The nice stuff of using a superscript is that you have the same input file for doing the calculation if you use Gaussian or Turbomol or Jaguar or whatever. The scheme which is used to do the calculation is an additive one, meaning that it's not a nonium calculation. Polarization, the MM part is polarizing the QM part, and we use the concept of charge shift, so we remove the charge close to the QM region and we add a dipole on the number. Technically, we start from a PDB, in that case 1HCH, we add the hydrogen, we minimize, we add the layer of 16 angstrom of water, we relax 8 angstrom of water plus the enzyme, we do an MD, and we have the snapshot for starting the QM calculation. The QM part is done with the classical bis-relip with a double zeta basis set at that time. The MM part is done with Charm 22 at that time with a modified parameter for iron. Okay, it looks like that. You have an iron oxo usually at the center. I'll show you that already, that there is an important arginine in the system plus an histidine. So this is the core of the system. What we know about the catalytic cycle is that you start from iron 3, you introduce H2O2, and this will oxidize the iron center to iron 4 and generate an iron oxo. This iron oxo is called compound 1, and this iron oxo will oxidize the substrate, generating a radical, plus what is called compound 2, and this compound 2 is again able to oxidize the substrate, generating a second radical. In fact, these two radical S dot will couple together to create a CC bond usually, and you go back to the resting state in which iron is at oxidation state 3. So one of the questions on this catalytic mechanism was, how do you move from iron 3 to iron 4 oxo? This is important because the iron oxo is a key species. It's a compound 1, it's an open state, open shell species, which has at the same time two spin states, either quartet or double. Okay, let's skip some technical part on that, because this is not what is interesting us this morning. So you start from iron 3, you introduce H2O2 here, and from here there is two solutions. Either you break at the same time the OO bond and the OH bond to go directly to compound 1. This is called the Polos-Crott mechanism, and it was in all the textbooks for 30 years. Or first you remove a proton on oxygen alpha here, you generate what is called in the literature compound 0, and then the base is giving back the proton on oxygen beta, and then you break the O bond and you generate compound 1. Okay, so the question is it a direct mechanism or a step-by-step mechanism involving a proton transfer? Okay, so I set up the QM calculation, and you do everything. Okay, this is a very complex figure, so I will summarize what's going on here. This is the hydrogen abstraction, proton abstraction, sorry, going on here. So moving from H2O2 to compound 0. There is a different conformation, but basically what we can see is that the barrier for doing that is between 14 and 18 kcal depending on the conformation. Okay, so keep that in mind, 14, 18 kcal per mole for doing the hydrogen transfer. Next step is to do the OO bond breaking. If we start from compound 0 here, the barrier is around 12 kcal per mole, 15.4 minus 3, so 12 kcal per mole. Okay. On the other side, on the right side, this is a direct mechanism, so transferring the proton and at the same time breaking the OO bond. The barrier in that case is around 40 kcal per mole, so we can forget this mechanism, it's totally wrong. And here starts the problem. I just said that the barrier for OO bond breaking is 12 kcal, and the barrier for proton transfer is around 15 kcal. The problem is that we know experimentally that the rate limiting step is the OO bond breaking. We know that because there has been some oxygen labeling with oxygen 18, and when you do that, you can calculate the KIA, kinetic isotopic effect, and go back to the kinetic value. And we know that the barrier is higher for OO bond breaking than for hydrogen transfer. So the calculations are wrong. We are missing something. What we are missing is that we didn't take into account a water molecule in the active site. I will go back a little bit. If you look here, on compound 1, which is crystallized, there is 4 water molecules in the active site. And when we did the calculation for the formation of compound 1, we didn't take into account any water molecule. But if you introduce again a water molecule, the barrier is going down from 12, 17 depending on the level of calculation to 1, 5 depending on the level of calculation. So basically, the proton transfer, the first proton transfer, is assisted by a water molecule. So summary of all that is that first, the textbook mechanism is wrong. The direct mechanism will never occur. The OO bond breaking is indeed the rate determining step, but to generate compound 0, you need absolutely a water molecule in the active site on top of H2O2. Otherwise, it will not work. So basically what I'm saying after these 2 examples is first, when you start calculation, you don't know if you have proton where they are. Second, here, you don't know if you need water more precisely, where are the water molecules. So you need to check the position of water molecule by doing, of course, molecular dynamics, but good quality ones, of course. So this is a first example of how tricky can be the setup of QMMM, because you have some ideas, but you need to check in an iterative manner with experimental results if it is working or not. This story of water molecule, we check that by doing metadynamics Carparinello. And indeed, the reactive conformation introducing a water molecule in the active site is found to be here. So basically, there is a resting state for the enzyme, which is called B1 here. And the reactive state is here, something like 2 kcal above B1, meaning that you have an enzyme which is stable, and in order to react, it must move towards the reactive conformation by introducing a water molecule in the active site. The big problem here is that on this slide, the time of calculation for doing that was 2 years on 500 processors. So basically, a tiny water molecule took us something like 3 years for 2 postdocs plus 500 processors. So it's a very tiny problem in the enzymatic reaction, but it costs a lot of money in time and computer. Conclusion, we know now for sure the mechanism of HRP. It's not a dry mechanism as written in most of the textbook. It's a wet mechanism in which you introduce a water molecule inside the active site. This is relatively interesting because, for example, there is another class of enzyme related to peroxidase which are called catalase, and catalase are working with a dry mechanism. So basically, we are thinking now that during the evolution process of enzyme, there has been a splitting between catalase and peroxidase, and probably peroxidase has evolved to take into account the water molecule. Are we okay? Everyone is good? You see my screen again? Sorry, I think I did a mistake, technical mistake.
You have unshared your screen, sir.
Sorry, my man?
You have unshared your screen, I think.
Yeah. My screen. Do you see again? Do you see the screen again?
Yeah, it is visible now.
Sorry. Okay. Longer story now. I will move to acetylcholinesterase, and we'll discuss that for something like half an hour. First, acknowledgement, but no, I will skip, you don't care. In order to move on mechanism on the function, sorry, of acetylcholinesterase. So acetylcholinesterase, it's an enzyme involved in neurotransmission. When you want to transmit motion of your muscle, your brain is sending acetylcholine through the neuron, and so acetylcholine is received by the cholinergic receptor, and you will move your finger, for example. But if the cholinergic receptor is continuously receiving acetylcholine, you will move always your finger like that, but not only your finger, you can move, you can shake. So basically, to stop the motion, you need to have an active acetylcholinesterase, which will break acetylcholine into two parts, choline and acetyl. If you don't do that, you shake, and because this is muscle, and as you know, your heart and poumon, c'est quoi en anglais? Poumon. No. Lung. And your lung also will collapse if they are not regulated by your cholinergic system. Okay, so basically this is a key enzyme for neurotransmission. That's very important, but unfortunately there is a problem. I will explain in 1 minute. Okay. This is acetylcholine. this is acetylcholinesterase. On the first reaction, which is done by acetylcholinesterase, is to do a transesterification. So basically here it's an ester, here it's an alcohol, and this alcohol is replacing this alcohol. So you generate a step deacylation, so water molecule is acting on this ester and giving back acetic acid on the starting point of the enzyme. The problem is with this serine residue, which can be inhibited by what we call nerve agents, so phosphoesterase with the phosphoester with a leaving group. And in that case, you create a PO bond. And as you know, the PO bond is a very strong bond, which cannot be easily broken. And thus, when acetylcholinesterase is inhibited by nerve agent, it doesn't work anymore. It's not able anymore to do A1 and A2 reaction, and you stop to break acetylcholine. And thus, all your muscles are doing that, including your heart, your lung, and you die. There is a way to reactivate acetylcholinesterase by introducing a very strong nucleophile. It's called an oxime, in that case an oximate because it's negatively charged. So the oxygen atom is very nucleophilic, will break the PO bond and regenerate acetylcholinesterase. Okay. So we became interested in this enzyme more than 10 years ago. And I will show you now what we discovered. But before that, I will show you the active site. The active site is a buried one, very buried, because the channel to enter the active site is more than 20 angstrom, meaning that the active site is highly protected, in fact. There is a lot of hydrophobic residue also, which is strange because the residue is positively charged. I can't explain all the story behind that, but it will take something like half an hour. Basically, what you need to know is that there is a lot of cation pi on CH pi interaction, which allows the substrate to enter this buried active site and the reaction to be done. The active site looks like that. There is basically three components. The first component is what we call a catalytic triad. So serine 203, histidine 447, and glutamate 334. These three residues are highly conserved. You can find them in all the acetylcholinesterase that we know for all the everywhere in nature. Second component is an oxyanionic oil. You can think that this is simply a Lewis acid way of for enzyme. Third component is the substrate, in that case the reactivator, the oximate, which is called 2-PAM. And I will introduce something now, which is glutamate, GLU202, and we will see this is very important for the catalytic activity of this enzyme. So first, let's discuss normal catalytic cycle. We start here with acetylcholine. Acetylcholine is entering the active site after the 20 angstrom channel. The oxygen of the serine is acting as a nucleophile doing a nucleophilic addition. At the same time, histidine is grabbing a proton, and you obtain a tetrahedric intermediate like that here. The oxyanionic oil, so the Lewis acid, is acting as a stabilizant for the O-, but you have the negative charge is going back, and you liberate this oxygen, which grabs the proton here, and you generate choline. Here you obtain, in that case, serine, which as at the beginning of alcohol, which is now an ester, but there is water in the active site. And one water molecule is acting as a nucleophile, acting on this carbonyl. The nitrogen of the ethylene is acting as a base, grabbing a proton. You obtain a tetrahedric intermediate here, and after that you do an elimination. You go back and you obtain acetic acid, and you go back to the starting point, and you restart, and so on, and so on, and so on. And you do that at a speed of 10⁻⁹ molecule per second, so working nearly at the diffusion limit.
Hello.
Yes.
Sir, how you are getting the exact confirmation for the these active site residues? Whether you are optimizing that using QM, or you are performing a simulation, MD simulation, and then clustering the snapshots and then trying for the QMMM calculations?
Oh, for this specific enzyme, acetylcholinesterase?
Yes, sir. Means how you perform the...
This enzyme, we don't need to do clustering, MD followed by clustering, simply because there is a huge number of X-ray structure.
Okay, means...
But it was crystallized now 30 years ago, and we have a very good idea of the protein motion because we have several loops moving in different X-ray structure.
Yeah.
So basically, when you have a lot of experimental information, you don't need to do this kind of stuff.
Okay. So,
but this is very specific. Otherwise, it's important to do it. It's because here for acetylcholinesterase, we know very well how the enzyme is moving.
Okay. Can QMMM also be used to answer the allosteric inhibition, like how the allosteric site is regulating the orthosteric catalytic site of a protein? Can we also answer these kind of questions from QMMM?
I don't have an example in mind, but yes, I don't see why we cannot. Most probably in that case, it would mean doing QMMM coupled to molecular dynamics. So much more expensive, as I said before.
Okay, thank you, sir.
For allosteric inhibition, the best way is to do EVB calculation.
What, sir? Can you please repeat?
At some point during my talk, 1 hour before, I was saying that the best idea is to do EVB calculation.
Yes, yes.
You know, okay. You remember, instead of doing full QMMM, you integrate QM calculation into the MM force field. It's faster. Okay. Why I'm saying that? It's because allostery is involving dynamical properties of the protein, so you need for sure to do dynamic.
Yes,
okay.
Yes.
So basically, it's better to have a very fast QMMM scheme in that case.
Okay.
Okay.
Yeah. Thank you, sir.
You're welcome. Okay. So we have now a picture of the normal catalytic cycle. Now let's have a look at the inhibition. So we start from here, the resting state of the enzyme. The phosphoester is entering the channel and going close to the serine. In that case, the oxygen is acting as a nucleophile. This nitrogen of histidine is grabbing a proton, and you liberate a leaving group. At the same time, the oxyanionic oil is stabilizing the oxygen here. You obtain this hypervalent intermediate, or you go directly to the tetrahedric intermediate. It depends on the leaving group. We will discuss that just after by doing QMMM calculation. And when you are here, when you are here, you cannot go back here because the PO bond here is a very strong one. So you have inhibited your enzyme. Oh, I will skip this slide because this is the same one that for HRP, Chemshell, Turbomol, Charm, active scheme, polarization of the QM part, the ZMM, and charge shift. Same story for the setup. You start from a PDB, you add the hydrogen, you add water, you relax, you do an MD, you select snapshot. So, we will just discuss now the taboo inhibition. So the last mechanism I show you. How does it work? Is there something special or not? So we selected among the bad guys, I'm saying the bad guys because this chemical compound are chemical warfare, so they can kill you in 1 second. And this is a very important problem, especially nowadays, because it's also basic chemistry, and everyone can synthesize these bad guys as long as you have some chemical plants in your country. There is good guys. The good guys are called the reactivators, so the oxime. Except that there is a tiny problem. You can see from the figure on the right that the percentage of reactivation is at most 20%. So basically, when you move your finger, imagine that there is 1 million acetylcholine esterase here. Only 20% of this million acetylcholine esterase in your finger will be reactivated by this method, meaning that you will die, even if someone is giving you the reactivator. Okay, so why is it so special with Tabun? Why is there so very low reactivation with Tabun? So we do the calculation first for the inhibition. So basically, let me remind you, the inhibition can go through either a direct pathway or a two-step pathway. And I will calculate the first transition state. So the oxygen of the serine will do a nucleophilic attack on the phosphorus, and the leaving group will depart. But there is a technical trick, a chemical trick. In fact, your tabun is exists in two enantiomers, R and S. On your enzyme is also a stereo-specific enzyme. So basically, you end up with four solutions. Either the phosphorus compound is S, and the cyano group, the leaving group, is in syn position regarding towards the oxygen, or you have an S, still an S phosphorus, or the cyano group is anti towards the oxygen atoms. On the two other solutions, R phosphorus on either departure in anti or departure in syn. So you end up with four possibilities to calculate. We did that. So the S-syn barrier is 8.5 kcal, the R-anti barrier is 12.8 kcal, the S-anti barrier is 12.5 kcal, and the R-syn is 17.4 kcal per mole. Surprise! As you can see, we move from a barrier to 8 to 17. We double. It's huge because it's a barrier in kcal per mole, and as you know, this is the Arrhenius equation. It's an exponential. So basically, what I'm saying is that the reaction will never be done on this stereo configuration, and also on this one, in fact, because moving from 8 to 12, 4 kcal more, it's an exponential 4, it's huge, it's a huge value kinetically speaking. So basically what I'm saying is that when tabun is inhibiting acetylcholinesterase, it's inhibiting by doing this reaction, except that there is a problem. It's not an SN2 reaction. It's an SN2 prime. The leaving group is on the same side as the nucleophilic group. And nobody never think about that. So basically, in enzyme, it seems to be normal when you are involving a phosphorus group that the subnucleic is going in syn and not the one that you learn when you were undergrads. It's not an SN2 reaction. Okay, fun. So summary of this first step. The inhibition reaction for acetylcholinesterase is a mechanism of addition elimination, except that the leaving group is in syn. And we can explain that very easily. It's because there is repulsion between the cyano group with the aromatic residue on the peripheric side. Okay. But I would like you to remind something based on that, is that basically everything which is written in biochemistry article, which is not proven by experiments, must be considered as an hypothesis and not as a truth. And especially because biologists are not chemists. So basically they are very, very poor level in chemistry most of the time. So they are writing stuff which are most of the time totally wrong. Okay. Second step of this story, how do we reactivate acetylcholinesterase? So we end up just now with this intermediate on which the oxygen of the key catalytic residue is inhibited by phosphorus. We introduce now a strong nucleophile and you do a nucleophilic substitution to regenerate the catalytic residue cell. Okay, so we have several mechanistic possibilities. How do we read that? We start from here. Either we have a direct SN2 reaction to end up here, or we have first an elimination, so the PO bond is broken, and second you generate the PO bond with the oxymate. Or we have first an addition of the oxymate on the phosphorus atom, and then an elimination. Yes, we can guess that the mechanism elimination followed by addition is probably wrong, but better to check, because there is a couple of enzymes in the literature which are known to work through this intermediate. So better to check all the hypotheses, even the strong one. Oh, now I will introduce something which is very important in science. I talked already a little bit about it because this is the main idea between QMM, behind QMM. This idea is that when you have a very complicated system to solve, you reduce it to something simple. And here, before starting the QMM calculation, it can be good to do QM-only calculation by taking only the minimal system and adding step by step all the components in the active site. So you take the basic QM model, and then you add glutamate, and then you add oxyaninicol, and then you add pi stacking interaction with benzene, phenol, or indole. And you do all the calculation. And as you can see, what the calculations are saying is that the oxyaninicol is favoring this reactivation, that the pi stacking interaction are stabilizing the product of the reaction, but the glutamate is disfavoring the reaction. Is disfavoring by quite a lot, in fact, something like 30-50 kcal per mole. And I have a question for you. Can you guess why this glutamate is disfavoring this reaction when you are doing a QM calculation only? Hello?
Yes, yes. Your voice is breaking a little.
So sorry?
No, yeah, please continue. It is fine now.
Go ahead, guys.
You can go ahead.
I'm talking?
You can go ahead. Yeah.
Okay.
Please.
So
Yeah.
let's explain. When you are doing a QM calculation, and when you introduce an anion, the electron associated to the anion is not at all stabilized. So it's totally normal. What we are showing here is that we need a QMMM calculation in order to take into account in a correct manner this anion. So basically, it's always a good idea to do model system. But when you do model system, you must always remind that you introduce also some errors, and sometimes these errors are huge. Here you see more than 30 kcal per mole. Okay, so let's move to QMMM. Okay, so first calculation. Your oxymate, your system with a catalytic triad, and I'll talk a little bit about that. Glutamate here, 202, is not protonated. Okay, as you can see here, I draw the figure in the same on the same shape than here. The scheme I draw with classical way of drawing a chemical structure. So basically, what the calculation are saying is that it's not a direct nucleophilic substitution. It's indeed an addition elimination with a barrier of around 19 kcal per mole on a product at 17 kcal per mole. So a product which is highly unstable. So there is a problem because we know that oxymates are able to reactivate this enzyme. So it should work. Here it doesn't work. It's an highly endothermic reaction. So we have been thinking a lot about that. And if there is glutamate 202 here, it's not by chance. Because if you look, there is something which is strange. The pKa of oxime is 7.7. So basically, it can be either protonated or unprotonated at the pH of blood. And the pKa of a normal glutamic acid is 4. So it should be unprotonated at pH 7. But when you look at the real pKa of glutamate 202, when I say real, it's not experimental one, it's calculated because it's very complicated to obtain the experimental one. Calculations are saying you that the pKa of this glutamic acid is 10. So basically, it must be protonated at pH 7. And this is a first surprise on something I want you to remind if you are dealing with enzyme all the time. The pKa of amino acid are always wrong inside an enzyme. And when I say it's wrong, here you see the delta pKa is 6. So thermodynamically speaking, 10 power 6. It's huge. It's 1 million. So basically, we need to protonate glutamate 202. Glutamate 202, if we protonate, now the potential energy surface looks like that. Starting point 0, we have a small intermediate after the addition here, and after that a barrier for elimination at 8.8, and the product is at minus 0.2 kcal per mole, so slightly exothermic, only a little bit. Meaning that the reaction for the reactivation of acetylcholinesterase is working only if glutamate 202 is protonated. Okay? Does it fit with the stories that I'm saying since the beginning? Remember, NOS, we were having a problem with proton. HRP, we were having a problem with water. Here again, we are having a problem with a proton. So basically what I'm saying is that for enzyme, main problem is not coming from understanding the mechanism. The global one is usually can be guessed, but the calculations are really helpful in order to gain tiny details which are highly important, in fact, to design, for example, a new inhibitor, a new drug targeting this specific enzyme. If you don't know that glutamate 202 is protonated, you cannot redesign something targeting this enzyme. Last question, how did glutamate 202 is catching this proton? Because, let's be frank, it's a buried glutamic acid. So we need to understand from where the proton is coming from. And what we discovered is that glutamate 202 can grab a proton from glutamate 450, which itself is interacting with glutamate 452, which is at the surface, so in direct contact with the water around the enzyme. Okay, I will skip this part. No, I will not skip, but I will say it quickly. This proton transfer is occurring in a different manner depending if the enzyme is inhibited by nerve agent or not. Meaning that, for example, as you can see, when it's inhibited, it's harder to protonate glutamate 202. And remember, it's harder to reactivate if glutamate is not protonated. So basically, it seems that there is a link between the protonation state of glutamate 202 and the speed of reactivation. So conclusion for this story, it was mandatory to do QMMM. You cannot do QM-only calculation here because otherwise there is a problem with an anion. We know also now that it's a mechanism by addition elimination, but there is only one transition state. Something critical is a protonation state of a residue called glutamate 202, and we found a new pathway for proton regulation within this enzyme. Okay, that's the end. I would like to thank you for listening. A few pictures from my university. Basically, my lab here is here in the middle. It's a big, ugly tower. I'm not lucky to have my lab inside the historical building. That's it.
Thank you, Professor Etienne, for your talk. And now session is open for the interaction.
Yeah, you are much welcome.
If anyone wants anything to ask now, they are welcome to ask. Otherwise, they can always send an email to Etienne, and he will be very happy to reply.
Yeah.
But it's better to have interaction. If you have any question now, you can always have it. If you want to do this based on this tutorial and based on this lecture, and then when you face a problem, you find a question, you are welcome to ask. We'll wait for 20 more seconds.
Right.
Somebody is sending in the chat box, not in this chat box, that they have a Schrodinger package and they want to do QMMM.
Yes.
So, I mean, I think it's a very basic question that why, how this would start.
Okay. Showing a package. Basically, so I'm guessing you have Maestro, and you have, I think I remember it's called QSite, the QMMM package for Schrödinger.
That is
yes. I've been using it one time 15 years ago, so. But anyway.
Not relevant for this talk actually, but anyway, that's okay.
Man, I didn't hear what you just said.
So that is not a very relevant question now.
It will be working. The only problem is that Maestro is hiding a lot of information to user. So basically, you can't do that, but you need specifically to be very, very careful with the Maestro graphical interface. I would never recommend to use only graphical interface to do QMMM calculation. Because this is one main point I said during this 2 hours and a half talk. There is a lot of tiny details which are very important. So basically, it's very helpful to have a nice graphical interface because you see stuff, but you must always remember to check the text file for errors. Something else? I don't hear you, Iman. I see that you are speaking, but no sound.
So if anyone wants, they can go to YouTube and revisit the lecture if they have missed some part or if there is some more detailed information they want. They are always welcome to visit our YouTube page, and it will be available. It is already available.
Nice.
So, yeah. Thank you so much, Professor Etienne, for your nice talk, and see you soon. Probably next time. Probably next time.
I hope to see you in Riyadh during. Let's hope I can come to India, or you can visit us in Europe in the next few months. That would be much nicer. Okay. Bye bye, everyone.
Bye. Bye bye.
Bye.
Bye bye. So now it is a break time. After this break, we will be having a talk by Dr. Syed Farhan Ahmed Pasha, followed by Thomas. So Farhan's talk will start at 5:30 p.m. So we have tea break, and you can fresh up, and then you can have something, and then you come back at 5:30. 5:30 p.m. sharp, we will start the next session. Thank you.
Now?
Yeah, now I can.
Okay.
Turn off
दोनों जब हम स्क्रीन पर रहेंगे ना तो एक फोटो ले लेना, दोनों स्क्रीन पे जब रहेंगे तब, ठीक है। थोड़ा सा ये हिल गया है शायद। We have 1 more minute to go. Good evening, participants. Welcome to the evening session of today's MMTT 2021 International Workshop. I am very happy to introduce my colleague, my friend, and a long-term collaborator, Dr. Syed Farhan Ahmed Pasha. He is online. He received his PhD in chemistry from MJPR University, India. His research interest includes quantum chemical study for molecular interaction and finding new applications of density functional theory-based simulations. He worked as visiting scientist at the Korea Institute of Science and Technology, Seoul, South Korea. During his stay at KIST, he developed several formalisms for macromolecular interactions using DFT and parameters. In January 2008, he moved to CAA Grenoble, France as a research engineer with Dr. Martin Field to work for an FP7 Marie Curie project where he realized conceptual DFT calculations for the protein-ligand interaction. Later in November 2009, Dr. Pasha joined IFP Energies, and then in Paris, he worked as visiting scientist, and he worked on QMMM approaches to design new catalyst. Dr. Pasha moved to KAUST Catalysis Center in November 2010 under a special agreement between KAUST and European ICE Consortium, and worked extensively in close collaboration with Professor Jean-Marie Basset. In May 2014, Dr. Pasha moved to SAVIC and started his industrial career to contribute his computational chemistry-based expertise to a wide range of industrial problems. Currently, he is a staff scientist and he works encompasses with theoretical and computational exploration of the structure and properties of inorganic and organic compounds, polymers, and catalysts. Additionally, coupling high-level molecular modeling with the advanced machine learning approaches is the new paradigm to expedite computational chemistry calculations and transform knowledge into value. He has published more than 50 research papers. They are highly cited. You can get his profile on Google and several other places. He has given several lectures and he received several awards. So let's welcome Dr. Pasha. Now this is your turn. Thank you for coming. You are muted.
Hemant, and welcome to all of you. Good afternoon. So please allow me to share my screen. Share back. Just a minute. Just a minute. Okay, is visible now, I guess? Hello?
Yes, it is visible now.
Okay, so it is a great pleasure to be part of this important workshop, MMTT 2021, and I'm honored to be one of the speakers in this important workshop. As a matter of 2 decades back when we started computational chemistry, I never thought that one day we will be addressing computational chemistry workshop by the computer only, but something happened in 2020 that we are forced to do things virtually, as I take as a positive. So, as we all know, computational chemistry and drug designing is a long-standing question and practice around the world. But before going deep into the drug designing part or molecular modeling part, I'm just giving a snapshot of the drug discovery process, which was used to be like 15 years, but because of the computational chemistry, it shrink to up to a few years. So when we are talking about computational chemistry, it is not just drug designing one target, but there are several phases where computational chemistry or simulation is deeply involved. From protein identification, protein structure refinement, and high throughput screening, virtual screening, and drug designing, finding new drug, and then enzymatic reaction mechanism. So there are variety of the topic which being addressed by the computational chemistry or molecular modeling, because I have been listening several talks by other eminent speakers during this workshop, and I have seen some questions which been raised about the validity and limitations and efficiency of the computational chemistry. So based on the learning, I heavily modified my presentation because I tried to cover the topic which been not extensively discussed. So it is a little bit off what I was supposed to talk about, but let's begin with. So research and motive options, because I must tell you, as Dr. Srivastava mentioned, I'm coming from industry, which is one of the largest chemical industry in the world. So my perspective regarding modeling or regarding research is more into practical approach rather than just being on numbers. So if we begin with the research motive, so finding what to do is a very important thing, and that proves that a person has a curiosity what to do. This is a very primary thing to be active in a research. But how to do, if someone is knowing how to do, is a researcher or is a scientist, that is very well. Otherwise, you cannot survive or stand in scientific community. But there is a third parameter, which is why to do, which is very important. What to do you can get from the community, how to do you can get from the peer, from literature, from other places. But why to do, this is a very important question that you have to ask yourself. And only you can answer this question. What is the motivation, what you want to achieve? This is one important thing, and the day a person knows is a successful scientist, not only in terms of science, but in terms of commercialization of the product also. So the other important thing is for the molecular modeling or for the computational chemistry, placing this skill set in the right place of the overall chain of the discovery. If we get an idea, so we have several options. It depends where we begin from. Directly, if after having idea, we have straightforward answer, quit and leave the idea. No, that we don't want. If we go to the lab without doing any research, background search, then success and lab, there's a between one thing stands, you pray, you may get success or you may not get success. But in a real world, the right approach will be study about the idea, consult with expert, and then realize or do the modeling or simulation or consult with modelers. After having all these three parameters passed, then go to the lab, of course, success will come. So I was listening address remark by Dr. Shastri at that time. I don't remember who asked, but someone asked this question: what is the chance of success? That's a very good question, and often in different platform I get this question. So I address that part differently. Like in ancient time, people were traveling by sea or by horse or something, but today we all use aeroplane as a commute medium. No one is asking what is the chance of success while flying instead of sailing a boat, because this is a modern technology and we have to. In the same way, we have to do molecular modeling. This will not just only reduce the time and cost, but it will increase the chance of success for sure. Okay, so within molecular modeling approach, there are different theories and different parameters being described by other scientists over 20 years. And to my knowledge, at least 6-7 people received Nobel Prize in computational chemistry itself in 2013 and 2008 and before that. There are several approaches, that's DFT modeling or topological modeling or molecular dynamics modeling or empirical modeling. I must emphasize here, nothing is good, nothing is bad. It depends on the problem. And one has to understand the problem, and then when we can choose, when we identify the real problem, then we can identify the real tool to address that problem. That is very important. My talk will be divided in two parts, something conceptual and then some case studies I will be presenting. So if we have to define the overall chemistry, in the simplest way, we can define whole chemistry in 4 different kinds of reaction, whether it's additive reaction or dissociative reaction or displacement reaction or exchange reaction. Reactions are all 4 kinds. And this is all like whether we give energy to the system or we gain energy from the system. So in a nutshell, what we can see, overall thermodynamics is the mother of overall the chemistry or overall the reaction. So it is very important to know the thermodynamics of the reaction, whether the reaction will be exothermic or endothermic. If we know that, then we can rightly address and we can place right condition to facilitate the reaction. But the key question is how to know what thermodynamics is all about said reaction. So there, molecular modeling or computational chemistry is greatly helpful to understand the thermodynamics and followed by the kinetics. After knowing thermodynamics and kinetics, then when we are coming in a molecular modeling field, so you will see a bunch of techniques like molecular mechanics, ab initio molecular dynamics, classical molecular dynamics, empirical modeling, QMMM modeling, DFT, and topological descriptor. So it is all chaos or confusing for a newcomer or for a fresh graduate. Everything is fascinating as well as confusing. So my message is never get frustrated or fascinated. Make a balance what you want to achieve. There are methods which is cost effective, but accuracy is compromised, and if you want to go for an accurate method, then the cost, you have to pay a little bit cost and as well as time. So it really depends on problems to problem, and more importantly, overall modeling is a method where realizing an absolute number is not our goal, but finding a trend and finding a comparison between 2 existing cases, one of the primary goals. Of course, you can produce in certain cases absolute number, but that should not be the primary goal. So as I said about overall thermodynamics or energetics, so to solve those energetics problem, one of the primary and the most important equation is Schrodinger wave equation, that what we solve by within DFT using computational chemistry. So here the computer is not doing any magic. Computer is just helping us to solve these complex equations, but computer is not solving any problem. The problem we have to solve as a human being, and it is our intelligence, our interpretation, and our result reading techniques that will solve the problem. So never get a misconception about computational chemistry that computer is solving problem, because often people who are not in the field, they thought that, okay, give the problem to the computer and computer will solve. Certainly there are problems computer can help up to a certain extent, but not in computational chemistry, because computational chemistry is for deep chemists, for hardcore chemists. Being excellent in computer science doesn't mean that one is going to be computational chemist. You have to be super good in chemistry and physics as well as in mathematics to understand or to address the problem. Here it is a very simple case how in DFT we deal the molecule, then I will go for the complex problem. So this is the simplest molecule, methane, and that what we feed to computer in terms of Cartesian coordinate, and after solving multiple equations and taking approximations, we get this kind of energetics like enthalpy, entropy, and 0-point energy at different temperature. Here in this case 298 K, that's room temperature, but we can get these value at any temperature. But this is a very simple molecule, and to address this simple molecule, we have to supply these 5-line coordinates. Think about if we have a protein with several thousands atoms, then the writing coordinate itself is a challenge. But our code writing or advanced tool made this possible. One can address those problems in a very sophisticated way. So after solving these equations, there will be certain parameter when we have to derive from those calculations, because these parameters will not directly come from those parameters. For that, we need an additional program. Many people have written, even we have written some program. By solving those equations, you can get an essential parameter that can guide or that can give a descriptor's value for a particular molecule, whether it is a ligand only or ligand protein or any two molecular system. The important thing is because we are talking about electronic parts, so the frontier orbital electronic density is one of the most important parameter that derives several other parameters in molecular system. But when I'm talking about a complex ligand protein complex system, then still these parameters are valid with a slight tweak, and we have to compute perturbation in hardness or electronegativity or the number of parameters that is coming under the framework of conceptual DFT. We can compute for a whole complex, and we can get the reactivity parameters. So let's talk about this modified descriptor. As I said, hardness is well defined for a molecular system, but if we have a ligand protein complex, then here just to be clear, CC means complex conformation, means ligand conformation in a complex form, because molecular conformation is very important parameter. I will be talking in a minute about molecular conformation. So if we keep a particular active conformation which is inside complex and outside complex, the difference that is that we calculated as delta eta or delta hardness, that is one of the important parameter. Similarly, for energy change in QM part of the ligand, because as I said, if we had to solve a complex problem which is a ligand and protein itself, so we cannot treat whole parameter within DFT or within QM. Of course we can do, but that is not advisable because that will take a huge amount of computational resources. So we split overall energetics into QM and MM part. I'm talking about the energy change of the active centers, that's QM part, and we can get the complexation energy difference as well as electrophilicity index of a complex within QMMM part. So there are a bunch of parameters. We have written several scripts to compute those parameters, so we can get after doing a QMMM calculation. But certainly, to do these kind of calculation, we need huge computational resources, which was used to be often challenged. However, we overcome with this challenge in recent time, and now we have sufficiently large computational facilities that can do such calculation in fraction of a day or less than a day. So what I was talking about, the difference in hardness, as I defined, like in terms of energy, free ligand has energy higher than the energy of a ligand inside the complex. So when the complexation takes place or complex formation takes place, always the energy should go negative inside the complex compared to free ligand. Then only this complex is stable, that we all know. And other way around, in terms of hardness, if we see, so hardness should increase inside the complex, then only a complex is stable and it will sustain. So in other words, if the total energy of a complex is higher than the energy of the ligand and plus energy of the receptor, means the complex is negative and the complex is stable. So that will define the binding of ligand with a protein. So in a way, we can compute not only for the protein ligand, but also a solvent layer around the protein and some salt to make a system neutral. And this is a parameter how MM energy or QM energy we are treating, and then how we can calculate the hardness within the core part, which is treated by QM part, inside the complex and outside the complex, and by their difference we can derive important parameter I will be presenting in a minute. As I said, this is not just QM or MM part or ligand and protein part, but the solvation energy and explicit solvent is also an important parameter. And using QMMM calculation, the advantage is we can treat a whole system soaked in a water box. And then the whole system become like several thousand atoms, which is which was used to be computationally limited, but not anymore. So there are a bunch of important parameters we can calculate and can relate with the molecular activity or reactivity. I'm not referring to only activity and reactivity in terms of QSAR kind of thing, which is very well practiced in academia as well as in industry, but more of transition state theory based reactivity parameters one can calculate. And for that, we need, as I said, a large computational infrastructure. Luckily, we are fortunate to have enough computational infrastructure. Our one machine Cray system has about 200,000 CPUs and 2 small clusters, which is up and running in our own facility in-house. So what I mean to say, computational power is one thing that enables us to address different problems. Then putting right problem is our duty. Only computational resources or advanced program cannot solve all the problem, but we have to figure it out what to do and why to do. Let's go to the case study. As I said, when we are talking about pharmacophore-based virtual screening, I don't want to go in detail what is pharmacophore because you have heard enough in previous talks, so I'll cut short that part. In nutshell, if we have a molecule which is active candidate for a drug target, so there are certain functional group, they are supposed to bind with the protein, or they are supposedly important for a drug activity. And if we superimpose 2 or 3 similar molecules, we will find similar group is at the same, they are at the same place. Like this case, there are like 10 different molecules, but they have similar group at the same place. So these are called pharmacophore. Why these pharmacophore are important? in ligand-based design, it doesn't mean why they are important, but we can relate. But in a protein-based or target-based design, it is important to look into the protein pocket. If any particular group, hydrophobic or electrostatic group, is existing at this place, a counterpart should exist in the protein pocket itself. If it is not existing, doesn't make any sense why this group exists there and why this group will make, or this particular group will make an impact on the activity. If this group exists there and a counterpart exists in the protein pocket, then only this makes sense, which is itself is a very big achievement, and it's talking about the molecular conformation. Because if we supply a drug candidate to the protein and it is not going in the right conformation, which means the active moiety is not reaching to the right place, and there should be some impact of this mismatch. If we are able to match these functionalities from ligand to protein, then the complex will form and it will have an impact on overall activity. So that's how the molecular conformation is important. But when, like 2 decades back, we started only ligand-based designing, and what was the option? Only to find a minimum energy conformer, because that was the easy choice to begin with. Recently, I realized this, not recently, but almost 8 years back, I took one molecule, and there are 3 different conformers. One is minimum energy, another is maximum energy, another is a DOC conformer. And I did QMMM calculation with molecular dynamics also, and calculated the parameters, all what I described. And here are the statistics. You can see with the noted biological activity, the correlation of the minimum energy conformer was only 0.38. Sorry, this was max, minimum was 0.38, and with the maximum energy, the correlation was 0.22, but the DOC conformer was 0.50. So this is a very important observation, that why the molecular docking or DOC conformer is important. Because if we start with a minimum energy conformer, we will mislead ourselves. In case there is no protein structure or target structure is known, then we have no choice. But if it is available, then certainly it is important to look into the DOC conformer. Now, when we start with DOC conformer, then there are certain other challenges I'm going to describe here. This is a very simple penicillin binding protein, which is a serine protease protein, and these are beta-lactam inhibitor. So when this ligand goes into the protein, what happened? This 4-membered ring will open, and this one proton will go away, and they will make a acylation reaction, something like this. This is known and is being crystallized in our own lab while I was working in France. So this was very well known, and the reaction takes place over this reaction center. So the periphery of the protein is not participating, although they have impact, but this is a covalent interaction. So in case of ligand-protein interaction, the multiple kind of interactions, I'm particularly referring all these things in terms of covalent interaction. So when this ring open and this oxygen attacks to this carbon, there will be a covalent bond, something like this, and the proton will transfer to the nitrogen, and this whole reaction is called acylation reaction, and this is a normal mechanism of antibiotics. Now, as I said, DOC conformer is important. So if we look at this whole chemistry, there's a deep chemistry involved. It's not just a computer programming, but a real chemistry involved where a bond breaking and bond formation takes place. So first and the most important requirement is to find the right conformer that this moiety should reach to this serine protease or this oxygen. Otherwise, if they are far away, there will be no interaction. So to do molecular docking, Often rigid docking is in practice, but there are flexible docking is also available, and QMMM docking is also available. There are covalent docking program. There are a bunch of AutoDock or Gold, Glide, FlexX are available. After getting a dock conformer, it is very important to derive some QM-based scoring to rank 2 or 3 or 10 different ligand, which one will be the better binder than others. To do so, we need huge computation again. I'm just giving a snapshot of the whole complex. So the protein, this region is a protein, and this is tiny part is the active center where the reaction takes place, and these are the solvent box as we supply to the system. So this is the QM zone of the whole system, and this whole bigger part is treated by MM. And the actual active center is over here. The ligand is there, and the serine protease active center is there, which will open this bond and make a covalent bond. So in this case, we treated up to 200 atom with DFT, and rest was with MM. And I'm not advocating any particular tool. You can use any QMMM tool, ChemShell, Dynamo, QSite, or Gnome, whatever you prefer. There are a number of tools, but it is important to look into the real chemistry, how it is happening, and right level of theory. Because if covalent interaction is not taken care of with DFT kind of parameter, and if we treat only with molecular mechanics, the outcome will not be that fruitful. So, at the very beginning, I said these particular ligand, if they will go inside the complex, what will happen? Is the snapshot of the previous complex what I was talking about, and we can see the complex, without complex, the ligand hardness was 0.05, while when it was in complex, the hardness increases to 0.09, which is very well following this trend. Of course, energy on negative, but the hardness parameter is also satisfying our complexation strategy. So it means the supplied ligand is a good candidate or good binder for this target. And I'm sorry to tell you, so this is a serine protease, and they polymerize this penicillin binding protein. They polymerize from this point one another, and that make bacterial cell wall. But once this antibacterial target, antibacterial drug is coming to the this oxygen, they make a covalent bond, and there is no place for polymerization. That's how the bacterial cell wall polymerization will stop, and this will behave as an antibacterial. So to stop polymerization of PBPs, we need to supply certain drugs, and how to choose the right drug, this is the way we treat. As you can see, this particular reaction involves several steps, and this is a new elastic band method used to derive this mechanism. Serine protease is coming, and bond breaking takes place, then a bond formation takes place, and proton transfer takes place. So there are several steps involved. Apparently, it looks like a one-step reaction, but it is not. There are at least 6 to 7 different steps, and we can picturize those steps in different snapshot, which are here. I'm not talking about energetics, but I'm just talking about different steps involved from this point to this point. Then there will be minima, maxima, and we can rank them. We can calculate those energies. After finding 3 different best candidates from our approach, then I did a little bit inverse design, that whether this pharmacophore theory fits into these parameters. So these out of 160 molecules which were identified, only 26 were synthesized and 3 were found active. And those active molecules, then I did inverse design to look for these pharmacophore based theory, whether they still valid and they have a common pharmacophore. And yes, there were similarity in these molecules. So that gives a strength to the ligand-based design. If we rightly treat and if we have right conformation, we can achieve a good guess for a drug candidate without doing extensive calculation. But for that, we have to be very cautious. So finding these kind of drug candidate is not only challenge in terms of computation, but as a source where we find these candidates. So there are a bunch of commercial and open source database where you can download these drug molecule based on certain criteria, and you can do search. But recently, these machine learning and statistical tools came into picture, and they are playing an important role to find new candidate or lead optimization, lead identification kind of thing. Of course, these database and these online tool one can use if someone doesn't want to do extensive calculation. So I am giving here as a reference some web-based tool one can practice. And these machine learning tools like RDKit is one of the best tool you can think about, is smiley-based molecular designing, and then certain property calculation, and then Python-based programming within Anaconda, or machine learning using scikit-learn or TensorFlow. So they are emerging as a prominent tool within drug discovery, not only drug discovery, but in the chemical discovery field. But I must emphasize, molecular modeling is not a nascent baby anymore. It is a very well practiced in industry, and being an industrial expert in molecular modeling, I haven't seen any industry which is far away or which who is not using computational chemistry in their real life or in real R&D, real world R&D. Here there are some drug molecule which discovered by the computer-aided drug design, because often people ask what it will give, is there any success story? So there are a bunch of success story. I just cover some important snapshots. So these molecules were discovered by using computer-aided drug discovery. There are more cases where computational chemistry discovered or help in genome or in bioinformatics related problems that I'm not listing. I'm just talking about drug discovery or drug designing part. So in my final remark, because of the time limitation, I have to limit myself. In the final remark, ligand-based design is good, or I can say best to begin with, but to address a real-life problem, it is important to address the overall problem with high level of theory, and as much as experimental or real value we can supply, our model will be much better. Hello. The most important part is computational chemistry is not solving any problem, but helping us to solve a problem. So it is our duty to find a real problem and address those problems. Computational chemistry based tool will help us solving those problems. So I think that much I have for today. If you have any question, you can take.
Yeah, the session is open now for the discussion. If anyone wants to ask any question, they are welcome.
Hello. I am Anamika Shukla. I saw your presentation. You said that docked conformers are very important, so we have to do docking. So I just want to ask a question that for studying molecular system, molecular dynamics is very important, but can we proceed to QMMM calculations without molecular dynamics? Can we analyze the system And so I just want to ask, can we do it without molecular dynamics?
Yes, you can do. So molecular dynamics is one part, but QMMM doesn't mean that you have to do the dynamics. QMMM itself is a potential. You can minimize the energy. So whether you minimize energy and study system in the minimum state or in a dynamic state is up to you. Often the protein is not very stable in particular conformation. So for protein interaction, for protein conformation, we do the dynamics. But that's not the sole requirement. Without doing dynamics, in a minimum energy state, you can compute all those parameters, but we can compute within the DFT framework for a small molecule.
Okay, so if we do that, the role of water molecules or the solvent present, can we neglect it for our system? Because generally we consider the solvent environment for high calculations.
So there are two kinds of water molecules. One is a reactive water molecule, another is solvent. If you are talking about reactive molecule, my advice will be consider those particular water molecule in QM zone. So they've been taken care. If they are reactive, they are breaking or hydrolysis or something happening, they will be taken care of by DFT. But if they are offside reactive zone and they've been treated with the parameters, then they will not take part in the reaction, but their impact or epsilon value or let's say dielectric constant will be there. You can do one more thing without putting explicit solvent. You can assign implicit solvent in QMMM itself. So that will heavily reduce the computation cost and still you'll have a fair amount of gas.
Okay, thank you, sir. Actually, I found very difficulties in using metals if any ligand metals are present in any ligand. So I just wanted to do QMMM after talking. That's why I asked. Thank you.
So no, I will address that point also. And this is a very well addressed problem of metal within QMMM. I am sure previous some speaker must have addressed this heme-based chemistry because I know some of the experts were talking about. But metal is not a problem. If you are treating with DFT, metal should be taken care. Only the force field based calculation puts some challenge in metal. If you put right force field for the metal, still it is good.
Okay, sir. Thank you, sir.
Okay, so any other question? If not, then let's thank Dr. Syed Farhan Ahmed Pasa for his informative talk. I am sure we all have learned so many things from this presentation. And if you have any questions, you are welcome to send him an email and you will be getting the reply immediately if he is in the office. Otherwise, he will reply as soon as possible.
Sure. Okay, thank you.
Next talk.
I can leave now, right?
If you want, you can stay for the talk. It's up to you. So the next talk will be given by Dr. Thomas Steinberg. He studied chemistry at University of Freiburg, Germany, and earned a diploma with distinction in physical chemistry. He completed his PhD thesis on computer simulation of protein-ligand interactions in 2005. He joined the developer team of the MBAL MD package which is very popular for doing molecular dynamic simulations and available to download. As a postdoc at Scripps Research Institute in San Diego and Rutgers University in New Jersey, the work focus was on efficient free energy calculation method and QMMM simulation of charge transfer. A very interesting work. And after returning to Germany in 2008, Thomas established a junior research group at KIT, and they are working on fast electron transfer phenomenon in DNA proteins. He joined Schrödinger in 2013, where he was responsible for the large-scale application of free energy calculation methods in pharmaceutical drug design. Since 2017, he heads the application science department for whole Europe and support customers in employing Schrödinger's drug discovery technology platform for their research. Let's welcome Thomas. And Thomas, now you can start your talk. Thanks for coming.
All right. Thank you very much for the invitation. Could you quickly confirm that the audio is okay and that you can see my slides in presentation mode?
Yes.
Okay, excellent. I would actually turn off my video because I'm joining you from my basement home office in lockdown conditions here, just to preserve bandwidth a little bit. As long as you can see the slides, I think that's the main part. Thanks a lot for the invitation, and I will spend my presentation to give some examples on how we at the software company Schrödinger think about applying molecular modeling for drug design methods, to summarize here a bit. As an outline, I brought a look at W-score, which is an advanced docking methodology that Schrödinger has very recently released. I would like to have a quick look at how we think that such tools should be combined into a theoretical molecular design platform to leverage them for maximum value. I would then try, time permitting, to outline the auto designer workflow. One way that compound ideation can be done in the context of such a platform, and hopefully be able to show some application examples of how this computational drug discovery at a really large scale can then perform. But let me start with W-score, which is a relatively self-contained and encapsulated part of my talk. It is a question of docking. And to introduce that, I would like to introduce you to what I would call the conservation law of computational chemistry. And that is, if you plot the methods that you're using on an axis of accuracy and an axis of speed and throughput, then what you'll see is that there's something like a frontier of either fast but inaccurate or slow but accurate methods. And depending on the task that you set yourself, you can move up and down this ladder of accuracy and throughput as needed. Now, of course, we all want to be super fast and accurate. So up here is the dream region of where computational chemistry would want to be, but if we're realistic, really isn't. There is always a trade-off in the amount of computational power that you need and the quality of the results that you get. If we are also honest, then this lower bottom here in the slow and not very accurate corner is what classical computational modeling looked like when people started doing it a couple of decades ago. Now, if we take a look at the methods that are very commonly used in computational chemistry, and the past talks, of course, have outlined quite a few of these, then we can build up this kind of frontier, starting with the very fast but quite inaccurate, let's call them rule of thumbs method. Yeah, rule of 5 for Lipinski, classical reals or panes filtering for unwanted functional groups. Easy to process billions of molecules with those, but of course, very, very approximate information that you're gaining from that. One step higher on the accuracy and computational cost ladder are QSAR and chemoinformatics methods. Those try to incorporate a lot more data, real chemical information, but they very often are based on easily computer processable fingerprints, 2D structures of your molecules. So if we want to take three-dimensional molecular structure into account, we probably will go to something like pharmacophore or rigid receptor ligand docking screenings. If we beyond that also want to take flexibility and the time axis of our system into account, we would have to move to something like molecular mechanics dynamics method. And now we're already talking about very, very expensive calculations that might take weeks to complete, not something you can do on large libraries of hundreds of thousands of compounds anymore. And of course, for the true purists in modeling, there's always high-level QM methods, which are accurate to this, often to the same level as an experimental measurement would be, but of course extremely costly, so they are only available for very small numbers of relatively small example molecules. And of course, this is not to disparage any of these methods. All of them clearly have their place. Sometimes the task that I set myself needs a very high throughput, not necessarily extremely accurate descriptor. Sometimes it needs to be as accurate as possible, and computational cost that is incurred by that has to be borne. Moving up and down this ladder skillfully for the right project that you're picking is one of the real skills of an experienced theoretical chemist. Now, what we, from a method development perspective, of course, want is to expand this frontier up to in this high accuracy, high throughput region. And I think on the QSAR level, a lot of that has been done with methods that I call deep learning QSAR, that very commonly are referred to AI methods. These have really expanded the knowledge-based modeling realm over the last couple of years quite tremendously to big data, to much higher accuracy methods. So a lot of progress has been made there. Likewise, in the area of molecular mechanics dynamics, Schrödinger especially have worked very hard to introduce free energy perturbation methods, molecular dynamics-based simulations, so true atomistic modeling into the drug discovery process, where we try to make expensive but highly accurate predictions of the properties of new molecules. And this has recently further been expanded to very high throughput artificial intelligence enabled free energy calculation methods. Of course, other fields are making progress as well, but you could wonder, maybe there's something missing in the middle here. So what would we have here? Somewhere on the intermediate level of accuracy, a high quality but still somewhat high throughput way of screening compounds that are potentially binding to my target receptor. And that's what I'm going to talk about first, and introducing an advanced docking methodology. Now, ligand docking, of course, is a very highly applied, widely used in the pharmaceutical industry as well as in academia, a very popular method. That is, if we're honest, really hard to improve on. If we look at the history of Schrödinger as a company, this is really what we started out with. Yeah, more than almost 20 years ago. Still, the most cited paper in Journal of Medicinal Chemistry is Schrödinger's GLIDE ligand docking tool. So this is something that people have worked on for apologies, for a very, very long time with a lot of effort. So docking is a hard problem, but we already have relatively good solutions to it available. So expanding on that is not going to be easy. Now, where would a potentially new docking method then sit? I have plotted here a very simple hypothetical virtual screening experiment. And I try to add the Indian number words that often greatly confuse me, just for fun here. So you could think about enumerating 100 million molecules. That is fairly straightforwardly done with chemoinformatics tools these days. Then we're filtering with relatively simple filter rules, unwanted, unstable chemistry, known toxicophores or so. We filter that down to maybe 5 million molecules. That is something that can be docked on a brute force level with ligand docking methods, leaving maybe 100,000 good-looking poses of novel molecules in my target. Going from these 100,000 then, at some point I need to either move to the high accuracy free energy calculations or look at buying compounds or look at synthesizing them. That is now a realm where we can probably deal with dozens or hundreds of molecules. So it seems like there's a step missing here. We go from 100,000 to 100. That's a few orders of magnitude too many to just pick. Of course, we can always pick randomly, but it would be really nice to have a more accurate method to reduce those 100,000 potential binders from the docked compounds to a few hundred molecules that are really promising to start either more expensive calculations or synthesis and purchase on. And that is what could be done by a second, more accurate, basically post-processing docking step. And that is what WScore is going to do. So introducing it quickly, WScore, as I said, it's a docking scoring function which tries to put a lot of effort into the accurate treatment of water molecules in a protein ligand complex. And it was developed with the goal to be the best-in-class empirical scoring function. So we're not moving to the realm of free energy calculation or dynamics here. It's still rigid receptor docking, but it is a scoring function that is much more elaborate than your typical ligand scoring function, because it puts a great emphasis on the elimination of false positives. That's a known problem in docking. The docking algorithms tend to generate false positives at a much higher rate than false negatives because they try to put everything into the binding pocket to see if it might possibly fit. And WScore was specifically trained to flag things or to flag structural motifs that should not occur in a real protein ligand complex and thereby kick out a lot of false positives. This represents, what I'm going to show you represents a very large R&D investment. This has been in development for more than 10 years with quite a lot of work that went into it. And WScore was only possible to develop because it heavily relies on already existing technology in the Schrödinger suite. For example, WaterMap, a method to properly represent where solvation sites are in a protein ligand binding site, sitemap to understand the characteristics of a specific binding site, where the polar, where the non-polar areas in it, MMTBSA to get an estimate of the binding energy based on a molecular mechanics force field, and a lot of other things. So maybe a very quick recap, docking 101. We're not moving beyond this, I would say, standard paradigm of what a ligand docking method is. It takes a rigid receptor, typically from an X-ray structure, could be a homology model or something else. It encodes this receptor structure in a way that makes it effective to screen this binding pocket, against molecules. The molecules are typically treated as flexible. So you can see here we've generated a lot of possible conformations of our ligand. And what docking programs do, one way or another, is to place all these many possible conformations of the molecule into a potential ligand binding pocket, and then come up with some kind of score that tells me, well, this placement of a molecule looks like a good binder or doesn't look like a good binder. It's important to remember that all of this is built with the emphasis on speed. Docking scoring functions have to be fast because we want to process millions of molecules with them. They don't try to calculate accurate binding affinities or things like that. They try to give you a quick indication this molecule could fit or it doesn't fit. Of course, under the hood, there's a lot of detail that goes into building a proper scoring function. I'm not going to go through the math here. The paper is full of all the details here of the various terms that go into it. The WScore scoring function contains the typical terms that you would expect from a scoring function: rewarding hydrogen bonds, rewarding salt bridges, exposed proper polar interactions, proper non-polar interactions, and a couple of more exotic ones. It also, in addition to what is commonly in a scoring function, tries to add proper strain estimates for both the protein and the ligand, partially from the MMTBSA calculations that are conducted. It tries to be very careful about desolvation and water penalties in such a system. And it adds a couple of additional terms that we found useful, all of which are described in much more detail in the paper. So what does a WScore docking workflow look like? As usual, we start with our prepared ligands transformed into chemically reasonable three-dimensional structures. Those are screened against what is called a WScore model. As I said, a WScore model encodes a particular receptor binding pocket, but by default, it is an ensemble docking method. So the assumption is that you have either multiple X-ray structures or multiple potential conformations of your binding pocket, all of which are combined, tried with an appropriate weighting algorithm to see how they contribute to the true conformational ensemble of that binding pocket. pocket. We have information about water molecules in those pockets, sitemap. If there's experimental binding data available, it is used to make sure that the model that was built makes sense. All of this goes into a W-score model. The ligands are then ensemble docked into these various structures. For the promising ones, more expensive MMTPSA calculations are being conducted. And then all of that information is combined into a final score that will then result in a hit list for the molecules. This is, as you can imagine, a slightly more expensive calculation as classical docking. A glide docking takes a few seconds for a molecule, and a W-score docking might take a few minutes. So we're not talking about something that can be done for millions and billions of molecules, but a method that can straightforwardly process tens of thousands or hundreds of thousands of compounds. I made a couple of pictures to outline the terms that are in the W-score scoring that are not typically in classical docking scoring function. Again, the first one, the W stands for water for good reason, is a proper treatment of desolvation. So whenever we place a molecule like the green one here into this cartoon binding pocket, we track potential solvation sites. So these are sites where in an empty pocket, water molecules would reside. And as the molecule binds, it has to displace those water molecules. And of course, the binding terms have to include the thermodynamic properties of those displaced water molecules. Were they stable, were they unstable? And that leads to both desolvation penalties of the protein. If I remove water molecules that bind to the protein surface without replacing them with something in the ligand that can take their place, such a hydroxyl group or so, there is a penalty incurred for the protein. Likewise, for the ligand, any charged or polar group in the ligand, like this pyridine nitrogen here, needs to either match a corresponding functional group in the protein, or it gets flagged as a desolvation penalty. And W-score is fairly strict about these kinds of desolvation penalties. A few of those are already sufficient to rank a ligand as probably not a binder. To balance that a little bit, the water sampling algorithm adds a little bit of flexibility. So if I have a water molecule that is being displaced by a ligand group, a non-polar ligand group in this case, so this would So for example, this donor and acceptor pair would normally not be beneficial because they're too far apart, but the algorithm recognizes that a water molecule might fit in between them and then be counted as a proper bridged interaction. On the flip side of that, we're also recognizing bridging water molecules that are not making the required numbers of interactions. So if the water molecule only is offered 2 hydrogen bond donors and acceptors, that is a very unstable or energetically high bridging water molecules. And something like that would also be flagged as not a beneficial pose by WaterMap. Furthermore, of course, that desolvation of backbone regions in the protein. In the backbone, there's typically very little flexibility that could alleviate something. So if a water molecule is removed and not replaced in the donors and acceptors requirements of the protein backbone, that is typically flagged as not a suitable pose. Likewise, as I said, W-score is by default an ensemble docking method. So we put in various protein conformations if they are available, and they are ranked relatively to each other by an estimate of their protein strain energy. Beyond that, there's a couple of more exotic interactions like pi-cation or cation-cation stacking that are included in the scoring function, and all of these together then make up the final W-score term. Now, how does this method perform? We are relatively early in validating it in real projects. We are heavily using it internally so far, but there's no real reporting publications on that. So what I'm showing here is, let's say, a standard set of target protein ligand complexes that is used to test docking scoring functions. And here we're comparing the performance of W-score versus Glide SP, our standard docking algorithm. in the early enrichment of docking sets of actives and decoys here. And what you can see is, in many cases, W-score improves on the results of Glide quite successfully, especially in cases where the ensemble approach is very needed, and where water molecules play a big role in binding. There are, of course, other cases where it adds very little value for being a much more expensive calculation. And overall, these numbers have already been superseded by the newer releases of our software. We get a little bit less than twice the enrichment that glide docking would give you. So again, not an order of magnitude improvement, because docking already is a relatively well-solved problem, but a doubling of enrichment factors, if it bears out in practice, would of course be an extremely valuable addition to the virtual screening toolkit. One problem with using this on known active decoy sets is that it is very often not all that clear that your decoys truly are decoys. What I bring here is an example from one of these training sets, where you see in green a computationally generated decoy molecule overlaid with, in silver, a known binder. Now, since a decoy molecule probably has never been made and tested, it's kind of hard to judge just from looking at the overlay. It's actually fairly possible that this molecule is ranked highly by the scoring function, not because it's a false positive, but because it would actually be a reasonable binder to this compound, to this target. So in summary, I introduced W-score, Schrödinger's new rigid receptor advanced docking module, a much harder scoring function than Glide, tries to be a little bit more physics or reality based in terms of how it decides what a good pose is and what not, shows quite nice improved enrichments compared to standard docking for the data sets that we have looked at. And the idea, of course, is that this more expensive docking methodology is to be used in conjunction with other methods in the screening cascade. So it's about to bridging the gap from the hundreds of thousands of molecules you might get as potential hits out of standard docking into the dozens and hundreds of methods that you can put into either experimental or more advanced modeling methods. So this concludes the first part of my presentation. For the next one, I would just want to talk a little bit about what we consider a molecular design platform. So something like W-score, in a way, it's a tool. It solves one problem. It places molecules in the binding pocket. That is a useful thing to do all of its own, but we think there's a lot more value in having this and many other pieces of technology integrated into a computational molecular design platform. And such a platform, in our opinion, might look a little bit like the following. The Schrödinger platform has three main components. The first one is a machine learning AI deep learning part. These are the extremely powerful novel methods that have come up in computational chemistry that allow you to look at or evaluate millions and billions of compounds very, very quickly. So this gives you the reach to explore vast chemical space, much, much more than experimentally possible in any sense. This has to be countered. All those vast numbers of molecules scored need to be scored accurately, and this is where the physics-based modeling comes in. This separates the data from the noise by being able to properly predict properties of molecules with an accuracy that in ideal cases approaches that of physical experiments. So this allows us to do precision design. It allows us to properly evaluate all the many compounds that come out of the fast machine learning, deep learning methods. Both of these legs of the design platform are very data hungry. They need and generate large amounts of computational data. And to make sense of that all, to transform that data into proper decision making, what we need is an enterprise informatics component. This is, I would say, the least scientific part of what I'm showing, but it is integral to get projects really running and to keep projects really progressing. This enterprise informatics component, it needs to allow you to collect, share, and analyze the data that has been generated by the various modeling approaches. In these days, of course, and in the case of Schrödinger, this enterprise informatics platform is called Live Design. It is a cloud computing, web-based, chemically aware spreadsheet. If you want to see, there's a front end here that tracks properties of molecules. So basically, you would have the whole inventory of chemistry within a project represented in what is called a life report here within that front end. And the nice thing about it is that it is very agnostic about the kinds of data that is being shared. This could be experimental assay data, it could be annotations, it could be structural data, it can be PDB structures, it can be images, it can be stored spectra, multi-parameter optimization. There's the eyeball here, there's a strong link to 3D structures. So at a single click, you can always bring up the 3D structure of a docked pulse in your web browser here. This is nice, easily usable and performant modern web technology that gets rid of having to update your Excel spreadsheets and PowerPoint presentations with related molecules, digging through various tables of when was that assay conducted, all of that present in a single spreadsheet, and importantly, always live and shareable with collaborators all over the world if they have the appropriate credentials. I just brought a couple of testimonials from some of our big customers who are starting to use these platforms quite intensely, and hopefully we'll be able to add many more to those in the future. Now, with this concept of a molecular design platform, what can we then do within it that is hard to do without having all that technology, all that infrastructure in place? AutoDesigner is one possible workflow that can be implemented in such a platform, and that is a workflow that I'll quickly show is supposed to solve, let's say, the design or the ideation problem. What I mean by that problem is that if you look at how traditionally drug design or molecular design is done, the famous make, test, analyze cycle of medicinal chemistry is that, well, we come up with some idea molecules. Those have to be modeled if they make sense, maybe synthesized, assayed, identified if our binding hypothesis is correct, and based on that SAR identification, we come up with new designs. That cycle can go through many such iterations, each of which typically is maybe a week or 3. Until we arrive at something that we consider our design problem is solved. The problem here is that design is very often the rate limiting step. If we don't come up with good molecules that fulfill our properties, then the whole cycle cannot advance. Yeah? Coming up with the right amounts of molecules is a key step in this process here. And hopefully, nobody who has ever done drug design needs to be convinced that that is a really hard thing to do. That is a really hard problem, because designing new molecules is a true multi-parameter optimization problem. So if we have our example chemist here who wants to make a change to a molecule, maybe I want to improve the potency for one of the targets, I also have to bear in mind maybe the potency for an off-target. But I also have to bear in mind a very large number of additional properties that all need to be fulfilled. They need to be non-toxic, they need to be soluble, they need to be permeable, they need to be synthesizable, they need to be within the IP range that I'm looking, they need to be outside of blocked competitor IP ranges, and so on. That is, of course, made a lot more difficult by the fact that all of these properties depend on each other. So if I change one thing, I probably change at least 5 other things here. So again, truly difficult multi-parameter optimization. And the problem here is, if I set humans to this task and put them under time pressure, there's a very strong tendency to just disregard everything that we don't have a good handle on, focus on the one few thing that we're good at, and just hope that the rest of the parameters will fall into place. And hope for the best is obviously not a very good strategy in drug design, because it usually leads to failures. So the question here is, can a computational workflow help with this multi-parameter optimization thing? The hope is, of course, that the Schrödinger platform can help here, because computational algorithms usually are very good at this kind of multi-parameter optimization. They basically are able to keep a lot more state in their head, metaphorically, than a chemist could ever be. So how is the design algorithm set up here? I would like to introduce quickly an analogy to chess playing. If you play against a modern chess app, A, you're probably going to lose because computers are really good at playing chess these days. And B, you're playing against a program that adapts its strategy to the part of the problem where it is currently at. So usually we would employ something like a knowledge-based approach in the early stages, because there's lots of literature on how you should do an opening, an attack, or a defense in chess. There's a middle part of the game where the amount of the probabilities, the amount of positions on the board explodes so hugely that you need some kind of fast heuristic tree search, because you can never be able to explore the entirety of the problem. You very quickly have to decide what is a promising path to think about more and which should be discarded immediately. Until you arrive at the end game, where you have not that many pieces on the map anymore, and these can be solved by brute force then. There you can just go through all possible options and find the best pathway forward. Now, surprisingly, a very similar approach is taken in what we're calling AutoDesigner here, which is a design tree algorithm to make to come up with new compounds. And like in the game here, like in the chess game, every single move is a change in a molecule. I'm adding or changing or removing a functional group. There's, of course, so many possible options here that I have to very quickly decide, is a given change a good basis for further changes, or does it not look like a good compound and should be discarded? And for this decision, we can put a lot of fast heuristics in place to make these kinds of choices. The first one is smart pattern searches. So we want, of course, stable chemistry. We want to avoid known medchem liabilities. We want molecules that are synthesizable. We want molecules that are amenable to further modeling methods that fulfill a couple of standard properties-based descriptors like rule of 5 and other ones. They need to be synthesizable. We want to exclude certain IP spaces. We need proper state predictions and so on. They need to dock. Many more things can be put in here, and that means that as this algorithm comes up with new molecules, 99.9% of those are more or less immediately discarded. We only keep the ones that look like they're fulfilling at least quite a lot of all of these properties for further analysis. A quick look at what these moves in chemical space really look like. Here are the 3 stages that we're applying. Given a starting molecule, there is a transformation stage, which is knowledge-based. So based on literature data, we have identified a large amount of matched molecular pairs, and those are applied swapping out, let's say, vaguely similar groups with each other. If something else might have been in that position, we will try it at that position. The second part, this is the heuristics, where we just try to get as much as possible reaction-based enumeration in. We identify breakable bonds in the molecule. For example, this aryl bond could be formed with a metal-catalyzed coupling, and we search commercially available building blocks for other groups that could have been introduced at this point. And then finally, the brute force endgame stage. The resulting molecules are decorated all over with a couple of decoration algorithms that just try to do nitrogen walking, methyl walking, halogen walking, and adding other small and simple bioisosteres or functional groups to there. All of this together is a way to generate millions and billions of molecules very quickly. And then the heuristics are used to quickly filter them down again to molecules that really look promising. For example, to feed into free energy perturbation calculations. I had FEP mention free energy perturbations to predict binding free energies of molecules a couple of times in my talk. I'm not going to talk about it quite specifically. This is something that Schrödinger has talked of at length at a lot of other places. It is a way to predict if a molecule is likely a good binder or not. It's a relatively expensive calculation based on molecular dynamics and so-called computational alchemy calculations. At this point, I would just want to sufficient to say it serves in the cases where it's properly applied as something like a virtual assay, where it can predict the affinity, the IC50 value that a molecule will have without having to synthesize it first. It's a very nice technology. Many presentations could be made about it, but here I just want to say this is one integral part of this design workflow, but here I'm more focusing on the ideation of new molecules. I'll skip this one. Of course, running these kinds of calculations, now this is truly applying molecular modeling at a massive scale. So we're talking about billions of molecules generated using tens of thousands of CPUs simultaneously for the enumeration and docking and prediction steps, where basically just storing the SMILE strings of all the molecules that you're considering is using gigabytes of storage, etc. So obviously not something that you would run on your laptop, but something that with modern technology, with cloud computing, is fairly straightforward to implement without having to think all that much about the high-performing requirements. But they are significant, and they, of course, need some professional support. I want to spend the remaining time I have on a couple of example results. So what does it look like if I really apply this methodology to a real project? And for that, I have one of our internal projects where we're starting out with a proprietary core that I can't disclose, but there's an R group in our molecule here that is pointing towards a specific group of subpockets in our protein of the CNS target. There's some activity there, but it certainly needs to be improved. And the question is, what should we change for this molecule to explore binding in either of these pockets? And of course, we don't want to go the traditional route where I would try to come up, test with ideas manually, so to say, but instead feed that problem into the AutoDesigner workflow and see what it spits out. So I put in my starting molecule. You can see illustrated here how it would come up with potentially alternative R groups here, score them based on our heuristics, then immediately reject most of these molecules and only the ones that look good. In the context of this project, we have decided to, on the fly, add a couple more models here because a very good clearance model came along in that project. So we wanted to make sure that we also generate molecules that have good predicted clearance properties. And as the algorithm runs to its end, it had looked at more than 500 million of ideas. Only about 100 or so came out looking good in all of the various parameters that we were looking at. And it came up with this new R group here that looked promising in pretty much all of these regards. At this point, of course, we have to go ahead and actually make this molecule to make sure that the predicted affinity is actually matched by the experimental one. The predicted clearance is matched by the experimentally measured. It fits all the other MPO properties that we have set for this project. And in the end, all of these came out as okay. And ultimately, the AutoDesigner workflow designed, and of course in exclamation points, because it didn't really design anything. I mean, it just applies the rules that the designers have put into it. But in the end, it generated a novel molecule that came out as potent, new IP with good ligand efficiency, fits all the CNS MPOs that we have used, and fits the clearance and fraction unbound models that we have put to it. So in a way, instead of going the traditional way where I start lead optimization with this molecule and then I come up with a lot of individual hypotheses and go after them one by one. Usually, if I change something, position B, I fall back to another step and have to solve problem A again. Instead of doing all of these steps manually, we replace them with an integral tree-based search algorithm that keeps all of the properties that we care about in mind simultaneously. Seeing that time has advanced a little bit, there's a couple more projects that I could have talked about, which I'll skip over. One thing that is nice that I wanted to point out is we went the way of doing a head-to-head comparison. How did the computer versus a pure human team perform here? So in this case, we looked at how many ideas were generated and tested by the MedChem team. How many ideas came out of the computer? Is there any matching or comparison between those? So in a specific project, AutoDesigner looked at 50 billion of ideas. The team came up with more than 4,000 compounds. And quite interestingly, every molecule that was made and tested that the design team came up with also was included in the AutoDesigner ideas that were made and tested. And that is quite stunningly something that came up in a couple of other projects as well. Whenever the AutoDesigner algorithm generated good molecules, it included all of the ideas that the design team had. Now, this is not to disparage our design teams, of course, we're working very hard, but it is kind of an indication that with pen and paper, even if you have really good ideas, really good knowledge and experience of the system, there is a limited number of chemistry that a human can come up with. And a properly encoded enumeration and ideation algorithm will always traverse a vastly bigger chemical space, and therefore hopefully have a much better chance to come up with novel and interesting compounds in there. Finally, of course, as always with computational methods, this is just the first step. Having that algorithm in place, we can, of course, try to make it smarter, because ultimately it always comes down as a decision-making algorithm. We have a molecule, should we transform it into derivative A or into derivative B? And I can put as much computational information into this decision as I want. So there's an obvious hook to add in a lot of additional knowledge-based, AI-based, experimentally based, or physics-based modeling as much as I want to make this algorithm more powerful and smarter. And that's hopefully something that we'll see a lot more examples for in the future. And with that, I would like to thank you for your attention. Hope audio and slides were nice and clear, and I'm of course happy to take some questions.
So the session is open for the questions, if any.
Any questions? I don't hear anything yet.
No, I think they are trying to turn on their mic, and also let's wait for 20 seconds.
Let me maybe also point out, of course, that if you have other questions or discussion, that either me or my colleague Shelvia from our Indian Schrödinger team should also be on the line. Always feel free to drop us a message. Simply write to help@schrödinger.com, and somebody will immediately get back to you on any types of questions.
Yes, so note down this email ID. This is available on the web also. So whoever is not able to ask question due to this mic issue, they can always send an email. But if you have any question, you are welcome to ask. You are welcome to interact in this session. We will be waiting for 20 more seconds. So if there are no questions, let's thank Dr. Thomas for his very nice presentation. And thank you, Dr. Thomas. It was very nice. I hope that all our participants, this video will be available on YouTube. They can watch it, they can rewatch it, and whenever they find a question, they will be writing you. Thank you for your time.
Thank you very much. Bye.
So this is the end of today's MMTT 2021 International Workshop session. Tomorrow, we will be having first talk by Dr. Nehar Ranjan Jaina. It will be followed by a hands-on session given by Dr. Harinder Singh from USA. He will be talking about introduction of chemistry.